Good morning. Takia Stevenson, DFO speaking. Before we get started, Dr. Goulart will be participating virtually and will not be chairing today's advisory committee meeting. Dr. Rebello will be the acting chairperson for today's meeting. I will now turn it over to Dr. Rebello.
Good morning and welcome. I would first like to remind everyone to please mute your line when you are not speaking. Also, a reminder to everyone to please silence your cell phones, smartphones, and any other devices if you have not already done so. For media and press, the FDA press contact is Amanda Hill. Her email address is currently displayed. My name is Elizabeth Rebello, and I will be chairing today's meeting. I will now call the December 4th, 2024 meeting of the Pharmacy Compounding Advisory Committee to order. We will start by going around the table and introducing ourselves by stating our names and affiliations. Those participating in all three topic sessions of this meeting will introduce themselves first. Those participating in specific topics of this meeting will be introduced at the start of their respective topic session. Panel members who will be in the CJC-1295 related bulk drug substances topic session will introduce themselves by stating their names and affiliations. We'll begin with that. Okay. We'll begin with Dr. Berman, the FDA.
No, so sorry.
Oh, okay. So now we'll go with introductions.
Marianne San Antonio, PCRT, FDA.
Good morning, Diva Shetty, Associate Director for Pharmacy Compounding Review Team, FDA.
Gail Bormel, Director of the Office of Compounding Quality and Compliance in FDA.
Office of Compounding Quality and Compliance, FDA.
Commander Kemia Sante, Team Lead, Office of Compounding Quality and Compliance, FDA.
Russ Westlake, Office of Pharmaceutical Quality, FDA.
Maitu, Senior Pharmaceutical Scientist, Office of Pharmaceutical Quality.
Todd Durham, Foundation Fighting Blindness.
Alan Veda, pharmacist retired from Institute for Safe Medication Practices.
Robin Bogner, University of Connecticut.
Brian Sarumaga, United States Pharmacopoeia.
Takia Stevenson, DFO, FDA.
Elizabeth Rebello, UT MD Anderson Cancer Center.
Kathy Guerra, Boston Children's Hospital.
Linda McElhaney, Elevance BioPlus Specialty Pharmacy.
Tim Fensky, National Association of Boards of Pharmacy.
Thomas Lupton, Director of Pharmacy Services, On-Demand Pharmaceuticals.
Danette Stas, Vice President, Regulatory Strategy, Jazz Pharmaceuticals, and Industry Rep.
I will now ask the online participants to introduce themselves. Dr. Kazel.
Gabrielle Cosell, Director of the Division of Compounding Policy and Outreach, FDA.
Dr. Ganley. Dr. Ganley, if you're speaking, you may be muted. Okay, we'll come back to Dr. Ganley. Dr. Goulart.
Dr. Padma Goulart, Duke University.
Dr. Gupta.
Dr. Anita Gupta, University of California, Riverside.
Panel members who will be in the CJC-1295 related bulk drug substances topic session will introduce themselves by stating their names and affiliations. We'll begin with Dr. Berman.
Kenneth Berman, MedStar Washington Hospital Center and MedStar Georgetown University.
Dr. Cook.
David Cook, Pediatric Endocrinology, Johns Hopkins University School of Medicine.
Dr. Jensen.
Dr. Kirk Jensen, Lieutenant Colonel, U.S. Air Force at Uniformed Services University.
For topics such as those being discussed at this meeting, there are often a variety of opinions, some which are quite strongly held. Our goal is that this meeting will be a fair and open forum for discussion of these issues and that the individuals can express their views without interruption. Thus, as a gentle reminder, individuals will be allowed to speak into the record only if recognized by the chairperson. We look forward to a productive meeting. In the spirit of the Federal Advisory Committee Act and the Government in the Sunshine Act, we ask that advisory committee members take care that their conversations about the topic at hand take place in the open forum of the meeting. We are aware that members of the media are anxious to speak with the FDA about these proceedings. However, the FDA will refrain from discussing the details of this meeting with the media until the be considered for inclusion on the list of qualified companies that may be used for compound drugs in accordance with Section 508 of the Federal Food and Drug Administration. Also known as 508 AOD 9604 is also required and the final alpha 1 that is
Good morning. Takia Stevenson speaking, DFO from the FDA. We do apologize for the technical difficulties. We have to take an unscheduled break. We will resume. I will hand it back to the chair to begin with the statement. Thank you.
Thank you. For topics such as those being discussed at this meeting, there are often a variety of opinions, some which are quite strongly held. Our goal is that this meeting will be a fair and open forum for discussion of these issues, and that individuals can express their views without interruption. Thus, as a general reminder, individuals will be allowed to speak into the record only if recognized by the chairperson. We look forward to a productive meeting. In the spirit of the Federal Advisory Committee Act and the Government in the Sunshine Act, we ask that advisory committee members take care that their conversations about this topic at hand take place in the open forum of the meeting. We are aware that members of the media are anxious to speak with FDA about these proceedings. However, the FDA will refrain from discussing the details of this meeting with the media until its conclusion. Also, the committee is reminded to please refrain from discussing the meeting topics during breaks or lunch. Today, we'll discuss the following bulk drug substances being considered for inclusion on the list of bulk drug substances that may be used to compound drugs in accordance with the Section 503A of the Federal Food and Drug and Cosmetic Act, also known as the 503A bulks list. AOD 9604 related bulk drug substances, CJC-1295 related bulk drug substances, and Thymosin alpha-1 related bulk drug substances. We note that all nominations for AOD 9604 related bulk drug substances, CJC-1295 related BDSs, and Thymosin alpha-1 related bulk drug substances have been withdrawn by the nominators, but FDA decided to evaluate these substances on its own initiative. For each of the substances, we will hear presentations from FDA, have the opportunity to ask clarifying questions, hold an open public hearing, and have committee discussion and voting. We have no nominators presenting today. The October 25th, 2024 Federal Register notice identified the uses FDA reviewed for each of the bulk drug substances being discussed at this meeting. Their uses reflect those for which adequate support was provided in a nomination. In certain circumstances, FDA may also review the substances in the context of unnominated or inadequately supported uses because, for example, such uses appear to be widespread, are intended to treat serious conditions or pose serious risks to patients. In addition, nominations and FDA evaluations for the bulk drug substances, which are included in the briefing documents posted on FDA's websites, identify the proposed and reviewed uses, dosage forms, and routes of administration. Thank you. Dr. Stevenson will read the conflict of interest statement for this meeting's 503A bulk lists topics.
Thank you. The Food and Drug Administration, FDA, is convening today's meeting of the Pharmacy Compounding Advisory Committee under the authority of the Federal Advisory Committee Act, FACA of 1972. With the exception of the National Association of Boards of Pharmacy, NABP, the United States Pharmacopoeia, USP, and the industry representatives, all members and temporary voting members of the committee are special government employees or regular federal employees from other agencies and are subject to federal conflict of interest laws and regulations. The following information on the status of this committee's compliance with federal ethics and conflict of interest laws covered by, but not limited to, those found at 18 USC Section 208 is being provided to participants in today's meeting and to the public. FDA has determined that members and temporary voting members of this committee are in compliance with federal ethics and conflict of interest laws. Under 18 USC Section 208, Congress has authorized FDA to grant waivers to special government employees and regular federal employees who have potential financial conflicts when it is determined that the agency's need for a special government employee's services outweighs their potential financial conflict of interest, or when the interest of a regular federal employee is not so substantial as to be deemed likely to affect the integrity of the services which the government may expect from the employee. Related to the discussions of today's meeting, members and temporary voting members of this committee have been screened for potential financial conflicts of interest of their own, as well as those imputed to them, including those of their spouses or minor children, and for purposes of 18 U.S.C. Section 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts, grants, CR-ADAs, teaching, speaking, writing, patents and royalties, and primary employment. Today's agenda involves discussion of the 3 bulk drug substances being considered for inclusion on the 503A bulks list. FDA will discuss the following bulk drug substances and the uses that FDA reviewed for each. 1, AOD 9604 related bulk drug substances, which are AOD 9604 acetate and AOD 9604 freebase for obesity. 2, CJC-1295 related bulk drug substances, which are CJC-1295 freebase, CJC-1295 acetate, CJC-1295 with drug affinity complex, abbreviated as DAC, freebase, CJC-1295 DAC acetate, and CJC-1295 DAC trifluoroacetate for growth hormone deficiency. 3, Thymosin alpha-1 related bulk drug substances, which are Thymosin alpha-1 acetate and Thymosin alpha-1 freebase for hepatitis B, hepatitis C, human immunodeficiency virus or HIV, coronavirus disease 2019 or COVID-19, depressed response to vaccinations, adjuvant to flu vaccines, malignant melanoma, hepatocellular carcinoma or HCC, non-small cell lung cancer, NSCLC, sepsis, infections after hematopoietic stem cell transplantation, or HSCT, chronic obstructive pulmonary disease, or COPD, myalgic encephalomyelitis and chronic fatigue syndrome, or ME/CFS. For nominated bulk drug substances, the nominators of these substances were invited to make a short presentation supporting the nomination. This is a particular matters meeting during which specific matters related to the 3 bulk drug substances will be discussed. Based on the agenda for today's meeting and all financial interests reported by the committee and temporary voting members, conflict of interest waivers have been issued in accordance with 18 U.S.C. Section 208 B3 to Drs. Padma Galore, Kathleen Gura, and Janet Lee. Dr. Galore is attending all topics. Her waiver involves stock holdings in a competing affected entity for all topics with an aggregate value between $25,000 and $50,000. Dr. Galore's waiver also involves stock holdings in a competing firm for the AOD 9604 and the CJC-1295 topics. The aggregate value of the stock is between $25,000 and $50,000. Dr. Gura is attending all topics, and her waiver involves 6 stock holdings. The first 2 stock holdings are in competing affected entities for all topics with an aggregate value between $50,000 and $75,000. The other 4 stock holdings are in competing firms. A competing firm for the Thymosin alpha-1 topic, a competing firm for the AOD 9604, and Thymosin alpha-1 topics, a competing firm for all 3 topics, a competing firm for the CJC-1295 and Thymosin alpha-1 topics. The aggregate value of each of the 4 stock holdings in competing firms is between $0 and $10,000. Dr. Janet Lee is attending the Thymosin alpha-1 topic. Her waiver involves stock holdings in 2 competing affected firms with aggregate value of between $25,000 and $50,000 and $0 and $55,000, excuse me. The waivers allow these individuals to participate fully in today's deliberations. FDA's reasons for issuing the waivers are described in the waiver documents, which are posted on FDA's website on the Advisory Committee meeting webpage, which can be found at www.fda.gov and by searching for December 4, 2024 PCAC. Copies of these waivers may also be obtained by submitting a written request to the agency's Freedom of Information Division at 5630 Fishers Lane, room 1035 in Rockville, Maryland, 20857, or requests may be sent via fax to 301-827-9267. To ensure transparency, we encourage all standing committee members and temporary voting members to disclose any public statements that they have made concerning the bulk drug substances at issue. We would like to note that Dr. Timothy Finsky is a representative member from the National Association of Boards of Pharmacy, NABP, and Dr. Brian Saramaga is a representative member from the United States Pharmacopoeia, USP. Section 102 of the Drug Quality and Security Act amended the Federal Food, Drug, and Cosmetic Act with respect to the Advisory Committee on Compounding to include representatives from the NABP and the USP. Their role is to provide the committee with the points of view of the NABP and USP. Unlike the other members of the committee, representative members are not appointed to the committee to provide their own individual judgment on the particular matters at issue. Instead, they serve as the voice of the NABP and USP, entities with a financial or other stake in the particular matters before the advisory committee. With respect to FDA's invited industry representatives, we would like to disclose that Dr. Thomas Lupton and Dr. Donette Stas are participating in this meeting as non-voting industry representatives, acting on behalf of regulated industry. Their role at this meeting is to represent industry in general and not any particular company. Dr. Lupton is employed by On Demand Pharmaceuticals and Dr. Stas is employed by Jazz Pharmaceuticals. We would like to remind members and temporary voting members that if the discussions involve any other bulk drug substances or firms not already on the agenda, for which an FDA participant has a personal or imputed financial interest, the participants need to exclude themselves from such involvement and their exclusion will be noted for the record. FDA encourages all participants to advise the committee of any financial relationships that they may have with the topics at issue. Thank you, and I'll turn it back to the chair.
We will now proceed with the FDA introductory remarks from Dr. Frances Gail Hormel.
Good morning, everyone. I'm Gail Bormel, the Director of the Office of Compounding Quality and Compliance, which is the FDA office primarily responsible for developing and implementing policies and compliance strategies addressing the quality of compounded drugs. I would like to formally welcome you to the 13th meeting of the Pharmacy Compounding Advisory Committee. As you've heard before, we will discuss 3 bulk drug substances that have been nominated for inclusion on the list of bulk drug substances that can be used in compounding human drug products under Section 503A of the Federal Food, Drug, and Cosmetic Act, which is also known as the 503A bulks list. As you've heard, the substances that will be discussed are 1, CJC-1295 related bulk drug substances, which includes the free base, the CJC-1295 acetate, the CJC-1295 with drug affinity complex, free base, CJC-1295 drug affinity complex acetate, and CJC-1295 drug affinity complex trifluoroacetate. The second bulk drug substance is AOD-9604 related bulk drug substances, which includes AOD-9604 acetate and AOD-9604 free base. And the third bulk drug substance is Thymosin alpha-1 related bulk drug substances, which includes Thymosin alpha-1 acetate and Thymosin alpha-1 free base. We have scheduled time for an open public hearing after FDA's presentation on each of the bulk drug substances. We are glad you are here today to participate in the Pharmacy Compounding Advisory Committee. We do value your input and expertise, both of which are critical to the success of this process. We look forward to a productive meeting and to continuing to work together. Thank you again for joining us.
We have Dr. Charles Ganley, who is able to join us virtually. Dr. Ganley, will you please introduce yourself?
Good morning. I'm Charlie Ganley. I'm the Director of Office of Specialty Medicine in the Office of New Drugs at FDA. Thank you.
Thank you. Slide 5, please. We will now proceed with the following 3 FDA presentations. Investigational New Drug Expanded Access presentation from Lori Bickle. Immunogenicity Risk of Compounded Peptides presentation from Dr. Daniela Vertelli. Bulk Drug Substances discussion presentation from Russell Westdyke.
Good morning. Next slide, please. I'm Lori Bickle, and I'm a regulatory counsel in CDER's Office of New Drugs Policy, and I have no conflicts to disclose. Next slide, please. This morning, we're going to look at 2 ways investigational drug and biological products can be used, either for research under an IND or for treatment through expanded access. This is to help inform advisory committee members and the public of ways in which an investigational drug or biological product can be studied or used to treat patients. First, I'll give a brief overview of the investigational new drug, or IND, submission requirements. This is needed before most investigational drugs can be studied in clinical trials. Then we'll move on to expanded access and how it differs from clinical trials, including the requirements for all expanded access and details about the 3 categories of expanded access. Finally, I'll take a quick look at some of the tools that FDA has developed to help patients and their physicians determine if expanded access is an appropriate option and to streamline the process if it is. Next slide, please. To start, we're talking about ways to use investigational drugs and biological products. Research on an investigational drug is usually done under an IND. To get to an approved drug, clinical trials provide evidence of the safety and effectiveness of the product, and to gather information about the drug that may lead to its eventual approval for commercial marketing and widespread use. Approval leads to the broadest availability for the product, with full labeling for patients and potential third-party reimbursement. However, a clinical trial isn't always an option. So in those cases, perhaps expanded access may be an avenue for treatment use using an investigational product if the appropriate conditions are met. This should be a pathway of last resort when other options are exhausted or unavailable. One key difference is that expanded access provides the investigational product for treatment use, not for research. Both of these pathways are distinct from the 503A and 503B compounding. Whether a product is or is not being studied under an IND is not a consideration in determining whether a bulk drug substance is appropriate for inclusion on the 503A bulks list. Next slide, please. I'll start with the IND for research, a clinical trial, using an investigational drug or biological product. However, all of the key content of the IND submissions covered here also apply to expanded access submissions. When I think about the elements for an IND submission, I break the components down into 3 categories. The first is information about the investigator conducting the study. This person may be a researcher in a large academic institution, or it may be a practicing physician within the community. Either way, basic information about the investigator must be submitted to make sure that they are qualified to conduct the research using a drug or biological product on humans. This information can be submitted on the forms that are shown on the slide. These forms are used for the information about the investigator, including all of their qualifications and their CV. Moving on to the second bucket of information is information about the drug product to be studied. What is its chemistry, manufacturing, and controls information? What is the product identity, purity? How will it be distributed during the trial? In some cases, a letter of authorization, or an LOA, may be used to reference information about the drug that is already on file with FDA within an existing IND. Next slide, please. Continuing with information about the drug is obviously basic information about the safety and efficacy of the drug. Is it reasonably safe at the dose and durations proposed? What clinical or non-clinical data do the sponsor have to justify the dose and duration they've proposed in the protocol? The final piece of information about the drug for the IND submission is about its efficacy. What is the sponsor's rationale to support the intended use of the drug during the proposed investigation? The third set of information included in the IND is the information about the patient and the proposed treatment, or the protocol for the investigation. This includes a description of the disease or condition being studied. What are the eligibility criteria for the trial? Information is needed about the clinical procedures and monitoring that will be in place to both evaluate the effect of the product and to minimize any potential risk to study participants. Finally, all INDs will need an informed consent form and IRB approval. These key content slides don't capture everything necessary for IND submissions, but I hope they've given you an idea of the types of information FDA requires and why before a clinical study using an investigational product can begin. Next slide, please. We're going to shift gears a little bit and focus on expanded access. In contrast to a clinical trial, which is primarily the use of an investigational drug for research purposes, expanded access is the use of an investigational drug or biologic for treatment. Expanded access is use of the product to treat a patient with a serious or immediately life-threatening disease or condition who does not have comparable or satisfactory alternate therapy. As I mentioned earlier, expanded access really is meant to be a last resort. Regardless, all expanded access requests and documentation must meet the requirements for an IND that I covered earlier. Next slide, please. Moving on to the basics of expanded access. The first thing I'd like to point out is actually the asterisk at the bottom of the slide. The sponsor or the manufacturer of the investigational drug must agree to provide it to the patient for the expanded access use. FDA cannot force a manufacturer to provide their product for expanded access. So once a manufacturer agrees to provide the product, there are three different types of expanded access under FDA's regulations. The first is individual. That's a single patient, which may involve the patient's treating physician. Individual patient access can also be emergency or non-emergency, depending on the situation. Under an emergency IND, the treatment use of the product can begin after getting the manufacturer's agreement and upon authorization by the FDA reviewing official. Often this can be done over the phone when treatment needs to begin immediately. The second type of expanded access is intermediate size population. There's no set number for intermediate size, but it's generally more than one and fewer than the number in a treatment IND or protocol. Finally, the third type is treatment use, which is typically larger and widespread. A treatment IND or protocol usually occurs either after phase 3 or compelling phase 2 data analysis. Now that we have the three types of expanded access, the next set of requirements apply to all three. Again, the patient must have a serious or immediately life-threatening disease or condition. There is no comparable or satisfactory alternative therapy. They aren't able to participate in a clinical trial. The risk-benefit analysis must show that the potential benefit justifies the potential risks. And finally, that providing expanded access will not interfere with the potential development for that expanded access use. Next slide, please. In 2009, FDA published the final rule on expanded access. In 2016, we released a question and answer guidance, which was revised in 2017. In 2022, FDA published a revised draft guidance for public comment with additional clarifications about the program. Public comments are being reviewed, and the guidance is in the process of being finalized. Next slide, please. I'd like to remind everyone at this point that all research done under an IND, as clinical trials and expanded access, come with the full range of human subject protections. These are the citations to the sections of the regulations that apply. Next slide, please. Since the regs were published in 2009, FDA continues to take steps to make sure the expanded access program is understood, that its criteria are known and followed so that the program is used appropriately and within its intended scope. These initiatives included creation of Form FDA 3926 in 2016. Prior to that, all INDs, including expanded access, had to use Forms 1571 and 1572, which are the forms for commercial INDs. At that time, FDA also updated the guidances and our website. FDA has also had an ongoing collaboration with the Reagan-Udall Foundation for the FDA to launch various tools to assist users in determining if expanded access is appropriate and help walk them through the process if it is. Additionally, FDA's Oncology Center of Excellence launched Project Facilitate in 2019, which is a program to help provide one-on-one assistance through the expanded access process. Next slide, please. Here's a screenshot from FDA's website. It's designed to be user-friendly with tabs for patients, physicians, industry, IRBs. I've also provided a link to a series of FDA-produced informational videos. Next slide, please. I also wanted to be sure to provide contact information for any questions that members of the committee or that the public may have about either INDs or expanded access. Next slide, please. Here are links to all of the regulations and the guidances that I've mentioned this morning. And next slide, please. Thank you for the opportunity to speak with you this morning.
Good morning. My name is Daniela Bertelli. I'm in the Office of Pharmaceutical Quality Research, and I'll be addressing the issue of peptide immunogenicity this morning. Next slide. I have no conflicts of interest to disclose. Thank you. Next slide. So today, as I said, we're going to be talking about product immunogenicity, describe the clinical immunogenicity concerns that exist for peptides, give a brief introduction to the mechanisms involved in generating an immune response, and then address specifically or discuss the immunogenicity-related concerns for compounded complex peptide products. Next slide. So immunogenicity is the unwanted development of an immune response, usually marked by development of antibodies for a therapeutic product. Now, as you can see on the right, development of antibodies is a complex process that involves multiple cells and signals. It is so regulated because their development can impact on patients' clinical status. So therapeutic peptides that can induce an unwanted antigen-specific response can impact on the safety and efficacy of the product. Next slide. The consequences, the clinical consequences can be none. Sometimes patients develop antibodies and nothing happens. They can be moderate. They can alter pharmacokinetics and pharmacodynamics. They can lead to loss of efficacy or sometimes even lead to toxicity if there's a drug accumulation. But they can also be severe. They can cause hypersensitivity or anaphylaxis, whether it's IgG or IgE-driven. They can cause immune complex diseases. The development of neutralizing antibodies can reduce the efficacy of the therapy, and the development of antibodies that would cross-neutralize the endogenous counterpart can be particularly complicated, as it can lead to a deficiency syndrome. Next slide. There are multiple factors that are going to impact on whether a product develops or induces an immune response. Some of them have to do with the patient, their genetic structure, their background immune competency, whether they have their underlying disease. But a lot of them have to do with the product itself, whether it's the API or the impurities that accompany that API. So it is important to understand the tolerance that the immune system has developed towards this peptide, and that has to do with the homology to self. The immune system is trained to ignore or accept peptides that are recognized as their own. But variants of those peptides can produce new epitopes that are recognized as different and thus can induce an immune response. The other type of impurities that can favor an immune response are things like aggregates, process-related impurities, contaminants, excipients, and leachates. And all of those can act as an adjuvant. And I'll clarify in my next slide, please. Next slide, please. There we go. So this is a little bit of a busy slide, but it's as you can see, if the idea is that as you introduce a peptide in a tissue, in this case we are modeling skin, what happens is that most of the peptide is going to go through the lymphatics to the lymph nodes, and there, depending on what the environment is, the peptide will be taken by cells that present the antigen to the immune cells or not. If it's not taken up, there will be no immune response. If it's taken up, the milieu in which this interaction occurs will determine whether there's development of antibodies or the body decides to just ignore the peptide. In the presence of impurities, what happens is that there's an increase in inflammation, and those antigen-presenting cells are getting activated and mature and are much better able to present antigen to those immune cells, those T cells and B cells, and induce the production of antibodies. And if those antibodies, again, are directed to the drug, they can induce changes in PK, reduce efficacy, but if they're also cross-react with the endogenous peptide, they can lead to a deficiency syndrome. Next slide. Because of these concerns, usually there is a certain level of characterization that happens that involves the assessment of their aggregation profile, the process-related assessment of process and product-related impurities, and those involve really complex methods for assessment, such as assessment of visible and subvisible particles, measurement of leachables and toxins, residual solvents, and innate immune response-modulated impurities, antigen uptake, and so forth. For product-related impurities, LCMS, MSMS, peptide mapping, and as well as in vitro assays that look at MHC binding or in T-cell activation are usually used. Next slide. In summary, the level of concerns with peptides is different than for small molecules. Peptide sequences can elicit an immune response, particularly if aggregated or present on scaffolding. Peptides administered via subcutaneous, intravenous, intramuscular, intradermal, inhalation, and intravitreal routes have greater immunogenic risk than oral or transrectal peptides. Product formulation is critical to the quality and stability of the peptide drug products. Formulation differences can modify peptide stability and immunogenicity. Peptide-related impurities may modify the target of the antibodies developed, and impurities or contaminants that activate immune cells may increase the immunogenicity of the API or result in an immune response that targets new sequences that may cross-react with endogenous counterparts. Next slide. Peptide-related impurities can be difficult to detect, analyze, and control because the impurities can have similar amino acid sequence to the peptide itself, necessitating advanced analytical techniques such as liquid chromatography, high-resolution mass spectrometry to detect, identify, and quantify the impurities. Impurities and contaminants can activate immune cells where the product is deposited, increasing the immunogenicity risk at trace levels, picograms or nanograms. Assessing the immunogenicity risk of immunomodulatory impurities in peptide requires complex in silico and in vitro studies. Mitigation of the immunogenicity risk of peptides requires sensitive assays and control of product and process-related impurities. Next slide. Here's an example of some of the studies generated in our own lab, looking at the risk of innate immune response modulating impurities in compounded in commercial samples. You can see on the left is a graph depicting the level of innate immune activation that we detected in cells exposed to 6 drug substance lots from a commercial vendor, and then 2 lots from a compounded vendor. As you can see, the level of innate immune activation was higher in those of the compounded vendor, but more importantly, when that drug substance was then filtered filter sterilized, the amount of innate immune activation achieved by one of these samples was reduced of the drug substance of the compounded samples, but one was not, indicating that there's different impurities, and those impurities may be differently purified with the use of usual purification methods. Next slide. In summary, product immunogenicity constitutes a risk for peptides, including compounded peptides, especially when delivered via certain routes of administration, which may result in significant risk of harm, including life-threatening reactions such as anaphylaxis. Control of impurities, including aggregates, can mitigate this risk, but requires sophisticated manufacturing and testing strategies. Thank you.
Good morning. Next slide, please. Good morning. I'm Russell Westdyke. I'm the Associate Director for Regulatory Affairs here at FDA. Next slide, please. I have no conflicts of interest to disclose. Next slide, please. So we're going to be talking a little bit today about bulk drug substances and active moieties, and you'll find that this is particularly relevant in all of the evaluations that we're going to be dealing with later on today. All of these presentations, whether it's CJC, AOD, or TA1, we're going to be handling multiple related bulk drug substances because it wasn't clear what, in fact, was being nominated or presented to us, or even the information contained within the nomination sometimes referenced multiple different substances. Despite the lack of clarity about which bulk drug substance was intended in the nomination, due to our concerns about safety, we decided to evaluate them all, present them to the committee. The goals of this presentation is to help you understand the regulatory definitions of bulk drug substance, BDSs, APIs, and active moieties. We want to explain how bulk drug substance differences have implications both for the products made with them and for the patients dosed with those products. And finally, provide some additional relevant background. Next slide, please. Okay, so we're going to begin with a thought experiment. For some of you, I apologize because you went through this last time, but some of you haven't, so allow me the chance to go through it again. Take a look at the screen and consider how many different bulk drug substances, APIs, and active moieties are present on that screen. And hopefully at the end of this presentation, you will understand why there are 6 different BDSs, 6 different APIs, and 2 different active moieties. And if a nominator proposes to manufacture or compound one of these things, or all 6, they would need to submit 6 different nominations, each supporting itself. You can't nominate diclofenac, but support it with diclofenac potassium information, or even naproxen sodium information. But that's in fact what we're going to be dealing with throughout the rest of this day. It's akin to, I'm nominating a COX-2 inhibitor, and here's all this information on aspirin. That's really not relevant in terms of the public health or the patients that are going to be dosed with these substances. So it's important to understand the differences and follow along with the presentations. Thank you. Next slide, please. Okay, so let's start with what do the regulations say about this? An active pharmaceutical ingredient is defined in the CFR in the portions that deal with compounding as being the same. Sorry, let me rephrase that again. Yes, an active pharmaceutical ingredient is defined as the same as a bulk drug substance. So then you ask me, okay, Russ, what is a bulk drug substance or what is an API? Sorry, I'm getting these in reverse. An API is defined in the regulations as the substance that's intended for incorporation into the dosage form, into the finished product, and is intended to furnish a pharmacological activity. In essence, an API is the aspirin that's in the aspirin tablet. And the BDS, that's the language we use for compounding, is the same thing. Next slide, please. From a practical standpoint, there can be differences in the API forms that are used. There can be various salt forms to a free base, and these tend to be chosen based on their pharmacological activity, based on their PK/PD characteristics, pharm tox profiles, et cetera, et cetera. And that selection is usually specific to the dosage form being studied. If I'm a formulator, for example, and I'm manufacturing an injectable product, I'm probably more concerned with the solubility of the API, and I'll select the salt form that aids in solubility. If I'm manufacturing a solid oral dosage form, I'm probably less concerned with solubility, more concerned with other characteristics, and so again, I might select a different salt form. Next slide, please. We've talked a little bit about active moiety, so I should spend a moment on that too. What is an active moiety? An active moiety is defined in the CFR as the molecule or ion excluding those appended portions of the molecule that cause it to be an ester or a salt. But if you're covalently bonding something to that structure, you've got a different active moiety. That's why, for example, although naproxen and diclofenac are not terribly unrelated, they are, in fact, different active moieties. And hopefully that helps you understand why, here in the example, we have 6 distinct BDSs and 2 different active moieties. Next slide, please. Why does this matter? Again, I'll start with the regulations. Okay, we've stated on a number of occasions that when a salt or an ester of an active moiety is listed on the 503A bulks list, only that particular salt or ester may be used. But it's not just regulations. Next slide, please. These definitions and distinctions are important in compounding, just as they are in drug product manufacturing. It's not just a matter of regulations or definitions. Those different salts, those different esters will have different pharmacological properties, different physiochemical properties, different pharm tox profiles and PK/PD profiles. And all of that matters, all of that is relevant to patient safety and efficacy of the product. Next slide, please. Also for our evaluation, what you see on the screen is the rule that we're supposed to follow in OPQ when we're trying to figure out if something is well characterized. I'm supposed to be looking at the properties and toxicities of the bulk drug substance that's going to be used in compounding and comparing it to the properties and toxicities of the test subject, of the test material that showed it to be safe and effective, or ideally shows it to be safe and effective. If those properties and toxicities are different, I cannot consider it well characterized. So when we're conflating different salts and esters in our nominations, it almost forces me to conclude it's not well characterized. Next slide, please. Should also mention, in terms of related information, there are unique identifiers and databases that are generally used to identify bulk drug substances or APIs. GSRS, or the Global Substance Registration System, is one of them. It's the home of the unique ingredient identifier, or known as a Unicode. You'll hear us talk about those a little bit today. There's also the Chemical Abstract Services, and that's the home of the CAS registry number. But please note, manufacturers and suppliers populate these databases, not FDA or any other regulatory agencies. The manufacturers and suppliers provide the structure and related information and request that unique identifier. Regulators like FDA don't own that data or police the data in it, and we don't have the authority to just unilaterally make changes to that data. That's owned by those various companies. Next slide, please. So, in conclusion, a bulk drug substance is defined as the same as an API in our regulations. The free base form of a bulk drug substance, as well as each of the salt forms, are distinct bulk drug substances, each with unique physical, chemical, PK/PD, form tox profiles, et cetera, et cetera. All of that impacts on patient safety and product efficacy. Nominators, bulk drug substance manufacturers, and compounders need to be aware of what single bulk drug substance they're nominating, manufacturing, and using to formulate a compounded product, and support that specific BDS in their nomination packages. Unicodes and CAS numbers are unique identifiers for APIs and BDSs, but they're not controlled by the FDA. And finally, our physical and chemical characterization assessment and conclusion is specific to a unique BDS. Next slide, please. Some final thoughts. I'm not going to spend any time or much time on botanical bulks, but for those of us who are following along or might want to nominate a botanical drug substance, please note that they are not like typical drug substances that are a pure substance. Botanicals tend to be mixtures of compounds, and care must be taken to identify a single bulk drug substance. We have guidances on this, and we encourage nominators to look at those guidances if they plan to submit a botanical. Also want to talk a little bit about the use of common names, because that's going to impact us a lot today. The use of common names as opposed to USAN names can be problematic and confusing. This will be highlighted as a concern for the 503 bulks evaluation list evaluations that we present today. Allow me to try to explain this issue using a hypothetical example so we don't need to reference any company confidential information. Imagine all of you are now suddenly the CEO of the ABC Discovery Company, and you have synthesized a new compound that you think will show promise as an anti-cancer agent. Let's call that compound ABC-123, consistent with your company internal naming convention. You get a Unicode for this, you get a CAS number for this, and you file an IND, or maybe you begin your early clinical trials overseas. During formulation development, you typically vary the salts and counter ions and evaluate the solubility and pharmacological effects of the compound. And as the story continues, your early clinical trials identify some significant negative safety signals associated with ABC-123 and its various salt forms. In response to those safety signals, you modify ABC-123 free base form and add a binding affinity ligand complex. Let's call it BAL. Now, this is structurally distinct compound and a different active moiety from the original ABC-123. But because you're in an early development phase, you just keep calling it ABC-123. You do some additional studies with this new form of ABC-123 and find a lack of efficacy, so you cease development. This is not unusual in a clinical program, in any development program, right? Most drugs fail in the clinic. At this point, you, the ABC company, are done with ABC-123. You're not going to make it anymore. You're not going to study it anymore. You have no reason to update GSRS or the chemistry abstract services, and you certainly have no reason to seek a USAN name for any of the compounds or variations that you studied. And at this point, there is conflicting information in the public domain, including in the clinical literature, about what ABC-123 actually is. Is it the compound without BAL, or is it an entirely different BDS, an active moiety that does contain the BAL structure? Which salt form or which active moiety is ABC-123? The truth is, there really is no right or wrong answer to this question. Now, if interest in clinical development of the compound was rekindled in the U.S., a sponsor would have to fix this problem. They'd have to submit information to us and let us know, hey, this is exactly what we plan to dose patients with. But if instead of development being reinitiated by a sponsor, a compounder decides to market ABC-123 prior to the substance being in the bulks list, and I am not commenting or suggesting there is any basis for that, but you as PCAC and we as FDA and even healthcare providers have absolutely no way to know what an RX for ABC-123 actually means. We don't know the right chemical structure, the BDS form, or even the active moiety form. It's entirely possible that physician thinks they're writing the script for one thing, while the BDS supplier thinks it's something else, and while the compounder thinks it's something else again. Well, this is a fictional example, It shows what can happen when nominators, BDS manufacturers, and compounders use common names as opposed to seeking USAN names and resolving naming conventions discrepancies. This is a challenge we're all going to encounter today and have to work our way through. Also want to spend a little bit of time on synthetic pathway considerations for complex BDSs. And this builds on Daniela's presentation about immunogenicity and impurities. For a complex BDS, such as long-chain peptides, where there's a greater immunogenicity risk, the synthetic pathway and related impurities can matter a lot in terms of product safety, and Danielle just spoke very eloquently to that. For such peptides, there may be as many as 3 broad categories of viable API manufacture, including 1, isolation from a natural source, 2, chemical synthetic pathways, and 3, biosynthetic pathways. And each of these broad approaches can lead to different impurity profiles, which can have downstream impacts on safety and efficacy. There are, of course, further variations to each one of these broad synthetic pathways, and even different in-process controls that might be used for the exact same pathway. The point being, there are many permeations possible in terms of synthesis and potential variations in BDS quality as a result. It is for those reasons that in the case of an approved drug product, there is a specific synthetic pathway and very tight controls in terms of API manufacturing to ensure the safety and efficacy of the product. And those specific technical details are company confidential information and not publicly available, nor can we disclose them to the committee. It's important to note that no such manufacturing constraints or controls can be assumed for a BDS that can be purchased on the open market and used for compounding. And those BDSs can and do have different characteristics and levels of quality. That's less of a concern for a small molecule like aspirin, for example. However, it's a greater concern for a complex BDS where the lack of any ability to ascertain what impurities is in there compared to what impurities were in the tested product really impacts dramatically on whether or not we can consider it a well-characterized substance. As the last slide of Daniela's immunogenicity presentation, actually the next to last slide of Daniela's immunogenicity presentation makes quite clear, these differences in BDS quality are not theoretical. They do have real-world implications on patient safety and product efficacy. Thank you so much for your time.
Thank you, Mr. Westdyke. We will now take clarifying questions to the presenters. When acknowledged, please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know and the slide number if possible. Finally, be helpful to acknowledge at the end of your question with a thank you. At the end of your follow-up question with a that is all for my questions, so we can move on the next panel member. Are there any clarifying questions for the presenters?
Hi, Kathy Gura. Thank you first for some excellent presentations. My question is to Mr. Westdyke. Where do excipients and adjuvant products fit into this? Because they're not API and they're just added sometimes to the final finished formulation. What kind of review do those go through? Thank you.
Gail and others might want to chime in as well from PCRT and or OCQC, but the evaluation that we do within OPQ is strictly related to the bulk drug substance. Do we think about the formulation? Yes, there can be formulation considerations, but in terms of the evaluations that are presented to you today, our focus is on the BDS.
Gail Bormel, I agree.
We have a question online. Dr. Gupta.
Good morning. My name is Dr. Anita Gupta. This question is for Dr. Varatelli's presentation on immunogenicity. My question is regarding the adjuvant reaction that you had mentioned, that there might be an immune response to these adjuvants. and that there might be a physiological or immune response. Can you clarify exactly what kind of response that might demonstrate clinically in patients or individuals that might use these peptides?
So, so any impurities that would activate the cells of the innate immune system, and those cells are not only in our blood, but are embedded in all of our tissues, can lead to an increase in the efficacy or the efficiency with which these peptides are taken up by the cells and presented to what we call the professional antigen-presenting cells. They present these antigens or these peptides to the lymphocytes in the immune system. That is critical to induce an immune response. And that's what adjuvants do in vaccines, right? When we have a vaccine, for example, there is a peptide. It is we use something to alert the immune system that it needs to make a response. That's called an adjuvant. Impurities in peptides can act in that same way, alerting the immune system that this is a substance that they need to make a response to. Does that address your question?
Yes, thank you. You had mentioned that there was anaphylaxis in one of your slides. I was just looking to better understand what kind of clinical response, if individuals are taking these, what kind of response? Is it only anaphylaxis, or are we looking at other kind of advanced kind of responses in patients or individuals taking these peptides?
There's different events that can happen if antibodies develop. Anaphylaxis is one type of adverse event. But just the mere development of antibodies can lead to loss of efficacy, can lead to antibodies that cross-react, that bind to similar structures that are endogenous, right? So we're administering peptides that have a certain structure. Those structures can emulate similar structures in the body of the patient. And if antibodies bind to those, it can alter their physiological function. So if you were to develop antibodies to those, you could reduce their activity. If that activity is non-redundant, it's a unique activity, that could lead to a deficiency syndrome. All of a sudden, that patient cannot perform that function that they used to be able to do because they have antibodies that are neutralizing that capacity.
Thank you.
Are there any more questions for our presenters? Dr. Kenneth Berman.
Ken Berman, just a question of what are the requirements for biologic effectiveness for the reason for the specific disease entity or for physiology to show effectiveness in these regulations?
Hi, this is Gail Bormel. For compounded drugs under the Act, there's no pre-market review for safety, effectiveness, or quality. So what we do as part of the bulk substances that are nominated for use by compounders under 503A, which are state licensed pharmacies, federal facilities, or licensed physicians, is we bring those bulk drug substances to this committee, and we review the information that's been submitted as well as conduct our own review of the literature. But that's the type of information that we look at. There's no pre-market review of these, formulations or of these bulk substances.
Thank you.
Are there any more questions? Yes.
Robin Bogner, if I were to purchase a peptide from two different companies, both of them being lyophilized peptides, and one company uses stabilizers and another company doesn't, are those two different bulk drug substances? Because I'm starting with two different things, one with peptide with perhaps sucrose or trehalose, and the other, without a stabilizer. Thank you.
Without knowing more information, it would be hard to answer that question, but generally I think the answer would be no, unless there was some interaction between the stabilizer and the peptide that actually changed the structure of the active moiety.
Thank you.
Welcome.
Are there any more questions? Okay. We will now proceed with the FDA presentation on CJC-1295 related bulk drug substances from Drs. Mary Ann San Antonio and Mai Tu.
Good morning. My name is Mary Ann San Antonio, and I'm a physician in the Office of New Drugs. Mai Tu is a senior pharmaceutical scientist in the Office of Pharmaceutical Quality. We will discuss the evaluation for CJC-1295 related bulk drug substances for possible inclusion on the 503A bulks list. Next slide. I would like to recognize the entire evaluation team, as well as the contribution of many other FDA colleagues who helped with this evaluation. Special thanks to the Division of General Endocrinology. Next slide. CJC-1295 related BDSs were nominated for inclusion on the list of bulk drug substances that can be used in compounding under Section 503A of the FD&C Act. CJC-1295 related BDSs were evaluated for the treatment of growth hormone deficiency. The proposed product route of administration is for a subcutaneous injection in a 2,000 microgram per milliliter concentration. The nominations were withdrawn, and FDA is evaluating the substances at its discretion. Next slide. We have evaluated publicly available data on the physical and chemical characterization, historical use in compounding, safety, and effectiveness of this substance. Next, my colleague from OPQ will speak about the physical and chemical characterization. Next slide.
Hello. Thank you, Mary Ann, for the introductions. I would like to start with noting that there appears to be inconsistent naming conventions associated with CJC-1295 related BDSs. The CJC-1295 related BDSs are analogs of growth hormones releasing hormones, or abbreviation is GHRH. There have been many modifications to GHRH over time, and we believe that Conjugam Biotechnologies may have developed CJC-1295 with drug affinity complex, or DAC, originally. DAC is a malay-imidopropionamide lysine, or MPA lysine unit, added at the C terminus, which we refer to as CJC-1295 DAC freebase in the memo. However, there are other modifications of CJC that may have been studied, including version without the DAC complex, and we want to note that CJC-1295 are common names and not USAN name, as discussed in the BDS compounding presentation by Russ. The common names do not follow the current systemic naming conventions and are being used for CJC-1295 related BDSs, as found in literature reports and other public sources. It's not possible to know which compound or structure is intended when referenced as common names, and the use of common name can be confusing and potentially cause medication errors. This represents a safety risk for patients, as they may be dosed with an incorrect medication compared to what the physician ordered, and it may also introduce error in chemical testing due to potentially inconsistent reference standards being used. Next slide, please. there is inconsistency in the material that provided to us, and for this reason, we evaluate 5 BDSs within the scope of the nominations and literatures due to significant safety concern mentioned. For the purpose of this evaluation, we will refer to 5 known CJC-1295 related BDSs as shown in the table, including CJC-1295 freebase, CJC-1295 acetate, CJC-1295 DAC freebase, CJC-1295 DAC acetate, and CJC-1295 DAC trifluoroacetate, or TFA. The 5 structure are distinct BDSs for the purpose of compounding with unique chemical structures and physical chemical properties. The 5 BDSs consist of 2 distinct active moieties, including CJC-1295 freebase and CJC-1295 DAC freebase. The information of Unicode CAS number, if available, are summarized in the table, and the molecular weight, molecular formula are unique to each BDS. Supplier is available for the first BDS in the table. Next slide, please. Due to the common name being used, it's not clear which BDS is being nominated from the nomination package received, which was then withdrawn. The first nominator nominates CJC-1295 freebase. However, the Unicode and the active moiety reference in clinical references refer to CJC-1295 DAC freebase that is different from the nominated BDS. The COA CAS number, molecular weight, molecular formula was not provided, and chemical name does not correspond to any of the CJC-1295 related BDSs being evaluated. The second nominator nominated CJC-1295 acetate, and the COA provided is for the same BDS. However, the Unicode and active moiety in clinical references refer to CJC-1295 DAC freebase that is different from the nominated BDS. The CAS number was a deleted CAS, and the molecular weight, molecular formula, chemical name match with CJC-1295 freebase that is also different from the nominated BDS. Next slide. The physical and chemical characterization is similar in all CJC-1295 related BDSs, and any difference will be discussed. For CJC-1295 acetate is the acetate salt of CJC-1295 freebase that is synthetic 29 amino acid of GHRH. Most of the properties are similar in all BDSs, and any difference are pointed out below. The BDSs are supplied as white lyophilized powder. CJC-1295 acetate, it's water soluble at 5 milligram per ml. There is no USB drug substance monograph for the BDSs. In terms of stability and storage condition, manufacturer recommend long-term storage at 2 to 8 Celsius in fridge or freezer, and it would remain stable up to 3 years when stored at negative 20 Celsius. The peptide in general is sensitive to product formulations, process, and environmental conditions, which might lead to aggregations and degradations. The potential impurities are peptide-related impurities and peptide synthesis process-related impurities, such as starting material, residual solvents, coupling reagents, activators, or catalysts. Next slide. The potential for immunogenicity, it's similar in CJC-1295 related BDSs and was discussed in more detail in the presentation on immunogenicity by Daniela. For CJC-1295 acetate, the COA was provided from a nominator, and it includes testing for peptide purity, largest single impurity less than 2%, but there's no information regarding the nature of individual impurities, aggregates, and bioburden or endotoxin level. There's lack of information on the potential of peptide aggregations, especially when formulated in an injectable dosage form for subcutaneous administration. We conclude that CJC-1295 acetate is not well characterized due to the concern arising from inconsistent naming conventions exist for the BDS. There's lack of certain critical characterization data, including impurity, aggregates, or bacterial endotoxin level. the potential for immunogenicity when formulated in an injectable dosage form for subcutaneous administration. We want to note that these concerns are also relevant for all CJC-related BDS. Next slide. For CJC-1295 freebase, it has limited solubility in water, about 1%, soluble in 1% acetic acid, which make it difficult to compound at the proposed solution concentration of 2 milligram per mil in water. Next slide. For this BDS, there is no COA provided in the nominations, and there's no information on impurity limits or testing result as critical attribute control in the COA that's reported in public domain. We conclude that CJC-1295 freebase is not well characterized. In addition to the reasons mentioned, CJC-1295 freebase has limited water solubility, which make it difficult to formulate in the proposed injectable dosage form. Next slide. And I think, okay, for CJC-1295 DAC freebase, it's CJC-1295 freebase with the MPA lysine unit added to the C terminus. And the BDS is soluble in water at 2 milligram per mil, which make it feasible to compound at the proposed concentration. Next slide. For this BDS, there's no COA provided in the nominations, and literature search show that most of the COAs for CJC-1295 DAC freebase only contain purity testing result, and there's no impurity profile available. We conclude that CJC-1295 DAC freebase is not well characterized. Next slide. And lastly, for CJC-1295 DAC acetate and CJC-1295 DAC TFA, a salt form of CJC-1295 DAC freebase. We have not identified publicly available information for these BDSs, and it appears that there's no supplier for these two BDSs, which likely contributes to the lack of data or any COA available. So we also conclude that CJC-1295 DAC acetate and CJC-1295 DAC TFA are not well characterized due to the reason previously discussed. Thank you for your attention. I will now hand over to Marianne, who will walk us through the next section of our talk.
Thank you, Mike. Next slide, please. Now we will discuss the historical use of CJC-1295 related BDSs in compounding. The form of CJC-1295 discussed in the references used in this section is often unclear. Therefore, the information will be considered for all forms of CJC-1295 as appropriate. CJC-1295 with DAC appears to have first been identified in 2005 as part of a research program led by Conjachem Biotechnology, in which derivatives of human GHRH were being developed in an attempt to overcome the short half-life of GHRH. Studies conducted in healthy human subjects were first published in 2006. However, Conjachem withdrew CJC-1295 with DAC from clinical trials later that year. CJC-1295 without DAC appears to have first been referenced in the literature in 2010 with the identification of an unknown pharmaceutical preparation that was seized by Norwegian police and customs. There is evidence that there has been compounding with forms of CJC-1295 since at least 2018, but no studies were identified that discuss the use of a compounded formulation of CJC-1295. Next slide. An analysis of online discussion forums to identify trends in popularity of doping products found that CJC-1295 emerged as a topic of discussion after 2005, and the number of discussions regarding its use have continued to trend upward. Additionally, there are reports of illicit use in professional sports. CJC-1295 related BDSs are marketed online for use in weight loss and muscle building, as well as an anti-aging peptide. It is unclear if any of the products are compounded. CJC-1295 is not recognized in the European or Japanese pharmacopoeias or in any of the national medical registries searched. In conclusion, the extent to which CJC-1295 related bulk drug substances have been used in compounding is unclear. Currently available data is too limited to understand the historical use for compounding. Next slide. Now we will discuss the non-clinical safety of CJC-1295 related BDSs. Like GHRH, CJC-1295 DAC acts as a growth hormone secretagogue, or GHS. CJC-1295 DAC is more stable and has a longer half-life than GHRH, and researchers suggested that the DAC modification accounts for the greater stability of CJC-1295 DAC. In rats, a single subcutaneous injection of CJC-1295 DAC TFA increased plasma GH levels. After peaking at 30 minutes, plasma GH levels declined to baseline by 2 hours post-injection. CJC-1295 DAC levels could still be measured up to 72 hours post-injection. Researchers suggested that the loss of the GH secretagogue activity in the presence of CJC-1295 DAC in the plasma could be due to CJC-1295 DAC-induced downregulation of GHRH receptors in the anterior pituitary gland, a decline in pituitary GH content, and/or CJC-1295 DAC-induced activation of a negative feedback loop regulated by somatostatin and the insulin-like growth factor IGF-1. FDA did not identify pharmacological studies of CJC-1295 free base or CJC-1295 acetate. Next slide. Non-clinical studies of the acute toxicity of the unspecified form of CJC-1295 DAC showed that in rats, a single IV injection reduced food intake, increased soft mucoid stools, and decreased activity. In dogs, a single subcutaneous injection induced emesis and transiently decreased activity. In repeat dose toxicity studies of the unspecified form of CJC-1295 DAC, rats were treated with IV doses and dogs were treated with subcutaneous doses for 14 days. Safety signals included, but were not limited to, reduced food intake and water consumption, increased soft mucoid stools and decreased activity in rats, emesis and decreased activity in dogs, reduced hemoglobin at all tested doses in both species, increased levels of cholesterol in both species, and injection site irritation with evidence of inflammation, hemorrhage, and minimal to mild necrosis in both species at all doses. Next slide. The genotoxic potential of CJC-1295 DAC unspecified form has been assessed through in vitro and in vivo experiments. These studies showed that exposure to CJC-1295 DAC unspecified form induced DNA damage in the pituitary cells. No embryo-fetal toxicity was observed in pregnant rats treated with subcutaneous CJC-1295 DAC unspecified form. FDA did not identify studies assessing potential effects of CJC-1295-related BDSs within a complete reproductive cycle and on peri- and postnatal development. FDA did not identify non-clinical 2-year carcinogenicity studies of CJC-1295-related BDSs. Next slide. In conclusion, while CJC-1295 DAC-related substances act as GHSs, it is unknown if CJC-1295 free base and CJC-1295 acetate are pharmacologically active. Safety signals reported in non-clinical toxicological studies of CJC-1295 DAC unspecified form included, but were not limited to, local irritation signals characterized by different degrees of hemorrhage, inflammation, and necrosis at the site of injection in rats and dogs, and genotoxicity signals characterized by DNA damage in vitro and in vivo. Due to lack of carcinogenicity studies, the potential for pituitary gland hyperplasia and tumors to develop due to overstimulation of somatotrophs by different forms of CJC-1295 DAC cannot be ruled out. There is a lack of non-clinical studies to inform safety considerations for potential clinical uses of CJC-1295 free base and CJC-1295 acetate. Next slide. Now we will discuss clinical safety. There was no PK data for children or for adults with growth hormone deficiency. PK data in healthy adults was discussed in 2 articles. The articles do not clearly identify the form of CJC-1295 related BDS that was administered. It appears that both references refer to CJC-1295 DAC free base as the active moiety, but they do not specify a salt. Teichmann et al. Describes 2 studies. The first was a study in which subjects received either a placebo or a single subcutaneous injection of CJC-1295 DAC unspecified form. The second was a repeat dose study in which participants received either placebo or 2 to 3 subcutaneous injections of CJC-1295 DAC unspecified form. In the study by Ionescu and Froman, 12 healthy adult men received a single subcutaneous injection of CJC-1295 DAC unspecified form. Next slide. CJC-1295 DAC unspecified form had a half-life of up to 8 days, and drug concentration was measurable for 10 to 13 days after administration. Elevated serum GH and IGF-1 levels compared to baseline were measured after administration of the study substance. IGF-1 levels exceeded normal levels in subjects who received 250 microgram per kilogram dose. Next slide. A search of the FAERS database for reports of adverse events for CJC-1295 related BDSs retrieved 2 reports which were both excluded. A search of the CIFSAN database retrieved no cases. Next slide. In study 1 by Teichmann et al., 94% of subjects reported an adverse event after a single injection. Injection site reactions occurred in approximately 70% of subjects receiving CJC-1295 DAC unspecified form, and rarely in subjects receiving placebo. Reactions tended to be more severe and/or prolonged after higher doses. Headache, diarrhea, and systemic vasodilatory reactions were also observed. In study 2 by Teichmann et al., injection site reactions were reported in all subjects who received CJC-1295 DAC unspecified form. Flushing, headache, nausea, abdominal pain, transient involuntary leg muscle contractions, and some loss of coordination, transient dizziness, and hypotension were also reported. In the study by Ionescu and Froman, increased heart rate and transient redness and tenderness at the injection site were observed after a single dose of CJC-1295 DAC unspecified form. Next slide. Anecdotal reports of a phase 2 clinical trial of CJC-1295 DAC GRF in subjects with HIV lipodystrophy were found via an internet search. Subjects were randomized to receive once-weekly injections of a 3-week escalating low or high dose of CJC-1295 DAC GRF or a placebo, and then continue for 9 weeks. The report states that 2 hours after receiving an 11th weekly dose of CJC-1295 DAC GRF, 1 subject complained of chest discomfort, and an ECG confirmed an acute myocardial infarction. The subject died approximately 1 hour later. The attending physician stated that the most likely explanation for the event was that the patient had asymptomatic coronary artery disease with plaque rupture and occlusion. The study was terminated, and the data from that study has not been published. No further information about the other study subjects or about adverse events was available. Next slide. Although 2 of the literature articles reviewed did not report major adverse events, these studies were in healthy adults, and most of the subjects only received 1 dose of CJC-1295 DAC unspecified form. Study authors reported that increased GH and IGF-1 levels were observed following administration of CJC-1295 DAC unspecified form. There are known potential risks associated with elevated GH and IGF-1 levels, and these risks are included in all FDA-approved recombinant human growth hormone product labeling and are listed here. FDA has not identified data or information to suggest that the CJC-1295 related BDSs would not present similar risks. Next slide. Teichmann et al. 2006 assessed the presence of antibodies to CJC-1295 DAC unspecified form in the PK study in healthy subjects and stated that no significant antibody formation was detected in subjects who received the active study drug. However, the observed incidence of anti-drug antibodies is highly dependent on the sensitivity and specificity of the assay, and the authors do not discuss this. Additionally, it is not known how this assay was validated, making it difficult to interpret the assay findings. Although the authors state that significant antibody formation was not observed, they do not specify whether there was no antibody formation. And this does not mean that there is no risk for antibody formation because data for long-term repeated exposure is lacking. Next slide. In conclusion, the available clinical safety information is derived from studies conducted in healthy adults and anecdotal reports of exposure in subjects with HIV lipodystrophy. Injection site reactions were observed in the majority of subjects who received CJC-1295 DAC unspecified form. Other adverse events are listed here. Although the authors of one study reported the lack of significant anti-CJC-1295 antibodies in subjects who received 1 to 3 doses of CJC-1295 DAC unspecified form, this does not mean that there is no risk for antibody formation because data for long-term repeated exposure is lacking. The CJC-1295 related BDSs are nominated to treat a chronic condition, but their long-term safety profile in humans is unknown, and there were no data to inform safety for the use in the pediatric population. Next slide. Next, we will discuss effectiveness. GHD is a disorder characterized by inadequate secretion of GH from the pituitary gland. GHD onset can be congenital or acquired during childhood or adulthood. Some cases of GHD have no known cause or diagnosable cause. GHD can be complete or partial. Diagnosis of GHD is based on a combination of criteria, including signs, symptoms, and GH stimulation tests. Next slide. Multiple recombinant human growth hormone preparations are approved for children with growth failure due to inadequate secretion of endogenous GH and adults with GHD. In pediatric patients with open epiphyses, GH therapy is used to normalize annual growth velocity and final adult height. In adults with GHD, GH therapy offers benefits in body composition parameters, exercise capacity, and quality of life. Next slide. No articles discussed the effectiveness of CJC-1295 related BDSs in humans with growth hormone deficiency. Patients with GHD will most likely not respond to GHSs including CJC-1295 related BDSs, unless these patients have partially preserved pituitary function or partial GHD. Patients with complete GHD will not respond to GHSs. Studies conducted in humans were in healthy adults, and it is unclear which substance was used. These studies did not measure endpoints such as changes in body composition in adults or height velocity in children. Next slide. In Teichmann et al. 2006 and Ionesco and Froman 2006, the authors stated, GH has also been used for therapy of disorders in children and adults in which pituitary function is either intact or only slightly impaired, or used in conditions with presumed functional GH deficiency. The authors hypothesized that CJC-1295 DAC unspecified form might be studied as an alternative to GH for conditions such as short stature associated with Prader-Willi syndrome, Turner syndrome, small for gestational age, and idiopathic short stature, or for treatment of HIV-associated lipodystrophy, wasting syndrome, and severe burns. However, there is no information concerning effectiveness to support use of subcutaneous CJC-1295 related BDSs for the treatment of these conditions. An anecdotal report of a trial in humans with HIV lipodystrophy who received CJC-1295 DAC-GRF was reportedly stopped early after a subject who received CJC-1295 DAC-GRF died, and the data were not published. Next slide. In conclusion, there is no information concerning effectiveness to support use of subcutaneous CJC-1295 related BDSs for the treatment of GHD. Professional society guidelines do not discuss the use of CJC-1295 related BDSs for GHD, and there are FDA-approved therapies with established efficacy for GHD. Next slide. In summary, for physical and chemical characterization, the CJC-1295 related BDSs are not well characterized due to inconsistent naming conventions, lack of certain critical characterization data, and potential for immunogenicity in injectable dosage forms. For historical use in compounding, although CJC-1295 related BDSs have been compounded, the extent to which they have been used in compounding and what they have been used for is often unclear. Currently available data is too limited to understand the historical use for compounding. Next slide. There are no peer-reviewed references with non-clinical or clinical data to assess safety and effectiveness of CJC-1295 free base or CJC-1295 acetate. Safety and effectiveness assessments discuss CJC-1295 DAC-related BDSs. The salt forms of the CJC-1295 DAC-related BDSs are not specified in the clinical references. Next slide. For non-clinical safety, while CJC-1295 DAC-related substances act as GHSs, non-clinical studies were not identified to establish whether CJC-1295 free base and CJC-1295 acetate are pharmacologically active. Safety signals induced by CJC-1295 DAC unspecified form included but were not limited to local irritation signals and genotoxicity signals characterized by DNA damage in pituitary cells. Due to lack of carcinogenicity studies, the potential for pituitary gland hyperplasia and tumors to develop cannot be ruled out. Next slide. The available clinical safety information is derived from published studies conducted in healthy adults and anecdotal reports of exposure in subjects with HIV lipodystrophy. Adverse events, especially injection site reactions, were reported in almost all study participants who received CJC-1295 DAC unspecified form. CJC-1295 related BDSs are nominated to treat a chronic condition but their long-term safety profile in humans is unknown. There were no data to inform safety for use in the pediatric population. Next slide. There is no information concerning effectiveness to support use of subcutaneous CJC-1295 related BDSs for the treatment of growth hormone deficiency. Professional society guidelines do not discuss the use of CJC-1295 related BDSs for GHD, and there are FDA-approved therapies with established efficacy for GHD. Next slide. After considering the information currently available, a balancing of the 4 evaluation criteria weighs against the following CJC-1295 related BDSs being added to the 503A bulks list: CJC-1295 freebase, CJC-1295 acetate, CJC-1295 with drug affinity complex or DAC freebase, CJC-1295 DAC acetate, and CJC-1295 DAC trifluoroacetate or TFA. Next slide. Thank you. This concludes my presentation.
Thank you, Doctors Tu and San Antonio. Slide 6, please. We will now take clarifying questions to the presenters. When acknowledged, please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. And if you wish for a specific slide to be displayed, please let us know in the slide number if possible. Thank you. Are there any clarifying questions for the presenters? Does anyone have questions who are participating online? We will now begin the open public hearing session. Both the FDA and the public believe in a transparent process for information gathering and decision making. To ensure such transparency, at the open public hearing session of the advisory committee meeting, the FDA believes it's important to understand the context of an individual's presentation. For this reason, the FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement, to advise the committee of any financial relationship that you may have with the product, and if known, its direct competitors. For example, this financial information may include the payment by a bulk drug supplier or compounding pharmacy of your travel, lodging, or other expenses in connection with your attendance at this meeting. Likewise, the FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. The FDA and this committee place great importance in the open public hearing process. The insights and the comments provided can help the agency and this committee in their consideration of the issues before them. That said, in many instances, for many topics, there'll be a variety of opinions. One of our goals for today is for the open public hearing to be considered in a fair and open way, where every participant is listened to carefully and treated with dignity, courtesy, and respect. For those presenting virtually, please remember to unmute and turn on your camera when your OPH number is called. For those presenting in person, please step up to the podium when your OPH number is called. As a reminder, please speak only when recognized by the chairperson. Thank you for your cooperation. To begin by calling speaker number 1 to the podium. Please state your name and any organization you're representing for the record, and you have 4 minutes.
Good morning. My name is Dan Denai. I'm the Chief Executive Officer of Avexis Health Solutions, which is a national network of over 10,000 medical providers, all representing many different disciplines of medicine. To be clear, We're all sitting here today because of a settlement offer made by the Department of Justice in response to a lawsuit filed against the FDA by our organization. The offer was made by the Department of Justice as they quickly and correctly recognized the FDA's overreach, as well as their failure to follow their own guidelines by incorrectly categorizing certain peptides as too dangerous to compound. My question is based on what? Why are these 17 peptides suddenly too dangerous to compound? Do we have people suddenly dying because they are overdosing on AOD? Do we have a rapid rise in hospitalizations because of the widespread use of ipamorelin or Thymosin alpha-1? Are there centers for addiction for CJC-1295? And why now? Why now is the FDA further restricting?
Oh, can I start over? My question is based on what? Why are these 17? Check. You guys hear me? I'd like to reclaim some time. My question is based on what? Why are these 17 peptides suddenly too dangerous to compound? And why now? Why now is the FDA further restricting a medical practitioner's freedom to practice medicine with compounds he or she believes, based upon his or her own clinical training, would benefit a patient? Medical providers practice the Hippocratic Oath, which is to first do no harm. Unfortunately, it would seem, especially in the case of compounded medication, the FDA is more concerned about protecting other interests rather than looking for ways to help patients live happier, healthier lives. Let us not forget that every approved recalled drug was at one point deemed safe and effective by the FDA. Many times, the only difference between medicine and poison is the dose. Many of the peptides that have been placed on Category 2 have been used successfully by thousands of our practitioners, treating hundreds of thousands of patients who utilize these compounds to energize cellular function and give the body what it needs to help address sickness and disease, including obesity, diabetes, and addiction, especially when commercially available products have simply not worked. The real-world result in the FDA's recent reclassification are that thousands of these patients have turned to the black market to obtain peptides. The black market includes fake, online, completely unregulated pharmacies, as well as other pharmacies advertising that they are a research facility, many of whom provide little or no direction on how a patient should use a compound. The agency's action now places patients into harm's way versus protecting patients' rights to a prescription provided by a medical practitioner through a licensed, regulated compounding pharmacy. Is the FDA now practicing medicine? Is the FDA interfering with doctor-patient care? Is the FDA infringing upon medical practitioners' freedom to practice medicine? Or is the FDA preventing patients from opportunities to get healthy? This may be a good time to acknowledge that at no time in the history of our country have we ever spent more money on health care. And although we have spent trillions of dollars, our population is sicker and under net profit on an annual basis. Something seems drastic. During the last PCAC hearing, there were concerns about side effects, understand the value and even the rules around compounding. Our trade association alliance to act on any proposal. It should also be noticed that our compounders do currently provide channels for patients and doctors to report adverse events. Again, something that the committee seemed unaware of. It really is no mystery why the citizens of this country are so sick and tired of being sick and tired. It is time for sick care to change to true health care in this country. And I am proud to do my part to make America healthy again. Thank you for your time.
Thank you. Now, speaker number 2, please state your name and the organization you represent to the right. You have 1 minute.
Yes, Jim Lavelle, Chair, International Peptide Society, co-chair for the American Academy of Anti-Aging Medicine. Let me get to that one. So on CJC acetate, the first step I wanted to make was that on the dosing and all the clinical trials, they are drastically higher than what was seen clinically in the hundreds of thousands of prescriptions that have been written from the 9 pharmacies that we surveyed. The dosing was typically at 100 micrograms once or twice a day. If you look at that on a 100 kilo person, it's drastically lower than the 25 milligram dose that was shown in the animal studies and some of the human studies. Let's take a look here. I think that the characterization of the CJC was well done by the presenter. I do know that if we can go on to the next slide. What are the current uses for? How do we see it in the preventative side? It's more from the standpoint of looking to restore circadian growth hormone as people age. Yes, they make growth hormone, they don't release it, can disrupt their sleep, can lead to loss of muscle mass as they're aging. Improving performance and recovery. I think it was well noted these cannot be used in any type of athletics where they're being tested. That is correct. And then at least on clinical reporting from practitioners across, I know what Dan had mentioned with his thousands of practitioners that people report getting improved sleep by restoring that circadian rhythm. I think we can move on to the next slide. This is what we do at the International Peptide Society. We write monographs and then we categorize what the level of evidence is for the studies that are available. And for us, this is what we've currently categorized. There's 6 randomized controlled trials reported. Level 3, evidence obtained from the randomized control. Level 4, evidence obtained without randomization. Of course, level 7 and 8, lower on the pecking order. Evidence obtained from either laboratory animal studies or evidence obtained from opinions or reviews. We do regular education and certification for practitioners to fully notify them of what the dosing should be, as well as provide all of the full text studies for the practitioners. And our practitioners would include medical doctors, MDs, DOs, nurse practitioners, or the primary people that get educated through International Peptide Society. Go ahead, next one. And these were already characterized. I don't think I need to go over these again. Yes, there is CJC with DAC and then CJC acetate. Very accurately talked about earlier. So when we looked at the, in this particular study at the 30 and 60 microgram per kilogram, which is still a very high dose in relationship to what is actually prescribed clinically, safely well tolerated, particularly at those doses. Noted that we do not have long-term studies. I think that that's the purpose of why International Peptide Society is trying to educate, create a forum, as well as be able to collect data so that we can see more and more how clinicians are utilizing these products in their, in their practices clinically. Go ahead and go to the next slide. Most frequent adverse events, I think that was it was fairly well characterized of, transient pain, swelling, or induration accompanied by some local urticaria. No serious adverse event reactions in either study reported. Estimated half-life of 6 to 8 days. I think there were two data points that were shown. Thank you. Thank you.
Please state your name and any organization you're representing for the record. You have 4 minutes.
My name is Tom Wind, and I'm a pharmacist at Pharmaceo, and I'm here today on behalf of all pharmacists to speak today about next slide, please. Next slide. So if you looked at with the briefing documents, there was a lot of talk of uncontrolled processes going on in the pharmacies as far as how they manage these bulk substances when they get them in. So when we talk about a bulk powder, it can be a active pharmaceutical ingredient, like an API, as they discussed before. It can be an excipient, preservative, sweetener, and allows us to compound certain entities without adding extra allergens. And that's sometimes what we are asked to do, because there's a reason why we're going to compound. It might be because they actually have an allergy to whatever is out there currently. Everything we're going to do that is going to be in sterile has to be from an FDA registered facility. So we have to get the bulk powder from an FDA registered facility. Next slide. And so when we're reviewing the bulk powders, these are just a few things that we're going to look at. We first are going to be able to check if they're FDA registered by going to the FDA drug establishment list. We then can ask them if they've been inspected. We can also check on the FDA warning and see if possibly they have any warning letters showing any 483s or something of that nature that we may want to review that'll give us a clue to some of their processes and what they're actually doing. We do ask about CGMP practices. Although I may not be able to get SOPs from all of them, I will get a table of contents so I can at least review what their policies and procedures are, getting an idea of if they're actually following CGMP in their practices. And again, FDA inspected. We want them to be FDA inspected. Although with our current guidelines through United States Pharmacopeia, it's only required that they be FDA registered, not inspected, although we want them to be inspected as well. Next slide. So this is just an example, and I apologize that the name of the actual company's not on there because I wanted to make it large enough that you could see, but in the briefing documents, again, the FDA commented on some C of A's that they got. This particular one was actually from D'America, is one that they mentioned. And on here, you'll see that they do peptide purity. They are doing microbial limits, so they're looking at some bacterial bioburden and bacterial endotoxins. Next slide. Oh, I think we missed one. But so there was, I think there was another one. That's right. We'll jump ahead. I had another one on there again showing similar items. And what I was getting at was the other one was going to be from Biopeptech. Both of those are FDA registered facilities, and they're both FDA inspected facilities. So if the FDA has a problem with the C of A's, then why don't they reach out to them and have them address that and put more information on there? They list the impurities, but they're right, they don't say specifically what those impurities are. But I think they would if they were asked to do so. So manufacturers are submitting information, though, for the processes. When they say that they're uncontrolled and they don't know what they are, it's not necessarily true because they can submit a drug master file. A drug master file is a confidential, detailed information about the facility's processes, articles used in manufacturing, processing, packaging, storing of human products. And I know some peptides have been submitted, not the ones we're looking at today, but they have submitted some GLP ones. Some companies have submitted those to the FDA to review. So that doesn't mean that they've approved it or denied it. They just review it. But that gives them an idea of the process that these companies use to go ahead and manage those peptides before they actually send them out to be utilized. So my question is, why can't we go ahead for any of the FDA registered and FDA inspected facilities say they have to submit a drug master file? Then you'd get all the information you want on the processing of what they do with these particular bulk powders. Next slide. So strategies with stability of peptides. I know that there was some talk about that too in uncontrolled processes, but usually buffers, pH modulators used to stabilize formulation. So, okay, thank you very much.
Can I get my slides up? I'm Dr. Terry Denai. I'm a clinician with 2 clinics in the Dallas-Fort Worth area. I'm representing myself and 3 other peers, all internal medicine, advanced endocrinology, and a pain management clinic. Next slide. I'm going to present some real-world data. This is from 3 internal medicine clinics. We had 508 patients, not prescriptions. 77% of these patients were on therapy for more than 6 months, greater than 40% of them longer than a year. You can see the breakdown of age and gender and ethnicity. Next slide. And this is data from a large pain management clinic as well. They were using it primarily to get their opioid patients off opioids, and quite very successfully. There's a few clinical case studies at the end regarding that. If I don't get to it, it can be found in the public documents. But they were using it primarily because it mimics natural body signals for pain control. It's non-addictive, accelerated healing, promoted tissue regeneration in several cases, and actually avoiding surgery in several cases, minimally invasive, very well tolerated, especially as compared to opioids and surgeries. days. And the patients also had a bonus that they experienced better sleep, enhanced quality of life. Next slide. The primary diagnoses in these 4 clinics we used for joint pain, sarcopenia, which is highly linked to increased morbidity and mortality in Americans, probably globally as well, fatigue, insomnia, unspecified bursitis, weight, and of course, chronic pain syndromes. These are the primary use in our internal medicine and pain management practices. Next slide. The dosing regime has already been noted, much lower doses than some of the negative outcomes in the studies that were presented previously. You know, I would go on record to say aspirin is lethal in the wrong dose, but we don't pull it off market. These doses were 100 micrograms sub-Q once a night in all these patients presented. Next slide. Now, I want to distinguish between side effects and adverse events because these are very different, and they are often interchanged incorrectly. And this comes from the VA Center for Medication Safety and VHA Pharmacy Benefits Management. This was a advisory panel that wanted to note adverse events are unintended pharmacological events that occur when a medication is administered correctly, while a side effect is a secondary unwanted effect. So that is a big distinction, and I'm noticing in several presentations they're used simultaneously. Next slide. So in these patients, side effects and AEs, this was our process. These patients were followed up 2 to 4 weeks after therapy initiated. They got injection training for at-home use. And also the pharmacies provided to the patients a QR code with a link to report any side effect or adverse event that the patient may experience. Next slide. What we found, side effects reported, which again, different from AEs, 19% of these patients presented with this flushing, this vasodilatory effect within the first 10 minutes of injection, which resolved within 20 minutes or less. This is a common side effect. No action was taken in most cases, but 2 patients did stop the medication because of it. Next slide. Adverse events, there were 3 adverse events, so 0.006% of the patients being reported. 2 patients reported itching. 1 patient had itching and subsequent anaphylactic response after several months of using them. All patients stopped the medication. The anaphylactic patient did seek medical care in the emergency room. After investigation, it was deemed by the pharmacist and the clinicians taking care of the patient that there was a preservative that could mimic an egg allergy, and this patient had a severe egg allergy, and the outcomes were resolved and, of course, reported to the pharmacy. Next slide. These are 2 pain management cases. One was a 67-year-old female. She had 3 prior failed surgeries on her right shoulder, was using CJC-1295. And in all of these case studies, it is the acetate form that was used. She also was using BPC 157. The outcome for her, and these patients were on these for greater than 6 months, avoided right shoulder reconstruction surgery. Her pain was significantly reduced, which led to a decrease in reliance on her pain medications, and she reported no side effects or adverse events. Thank you very much.
Any organization you're representing for the record?
Sure. My name is Lee Rosebush. If we could go to OPH slides number 1, please. For the record, I'm a doctor of pharmacy, a PharmD. I'm also a JD and represent the pharmacies inside that lawsuit against the agency. I apologize for the brevity of this. The reason why we have to go so fast today, if this was truly about patient safety, we'd actually have a true hearing from this. We'd have more than 3 to 4 minutes each. That is basically what we were assigned from that perspective, hence the brevity aspect of this. If I can, next slide. What you've seen right here is the 4 standards or the 4 requirements that are actually supposed to be reviewed when it comes to putting these substances on. There was a question earlier related to the amount of data or efficacy that would be required for that. Notice that there is not a standard for that. It's just simply supposed to be that there's an efficacy aspect to this. Next slide. Don't take my word for it. Don't take the statute's word for it. You can take FDA's word for it because they got their own regulation on it. This is the FDA's own regulation on what's required to get approval for the 503A bulks list. Notice there are 4 requirements. Notice also you heard a whole lot about products, about immunogenicity and risks associated with that. Notice here, the only thing that's supposed to be talked about products is when something's about efficacy. Everything else is about the drug substance. I would note that because if you actually look at the products that FDA has put on the 503 bulks list through PCAC, there's never been an injectable product ever put on there. And notice it's not supposed to be based on the products aspect of this. It's supposed to be based on the substance of these substances moving forward. Next slide. I've tried to give you a very quick history, a summary as to each of these substances. I will note, as was mentioned earlier, FDA does have a GSRS. NIH also has a PubChem. Both of the listings for CJC are on there. Next slide. This COA was not mentioned, if you noticed, in the presentation earlier. The reason being is the nominations were withdrawn. We weren't able to give a nomination associated with this, so this COA has not been included. Do notice in that perspective that you will see substances such as solvents included, things such as purity included, things such as the amino acid substance and sequences included on this. It is possible to figure out what these substances are, and it can be done. Next slide. Again, if there really was truly a concern about these products, it would be simply just release a guidance document as to what's needed for the APIs. Next slide. These are FDA standards. Yes, these are for approved products. Yes, these could also be required to be done for compounded products. And in fact, these products, which you have not heard, FDA conveniently left out, which is included in their deck, these have been made by outsourcing facilities. For those that don't know, CJC was made by a 503B. It was made under CGMP criteria, which includes the CGMP criteria for these substances. For those that want to learn about adverse events, you will see there was one adverse event reported to this. 503Bs are required to report their adverse events to FDA. Hence, there was one. I would also note that FDA inspected that 503B facility in the same year that these were reported being made. FDA did not issue any type of citation or warning or observation in their 483 associated with these products. Real quickly, if you can go to the history, and I'll be done. Next slide. Solvents, next slide. The reason why I'm going to this is keep going. As it had been historically used, you will see there's been over 450,000 prescriptions dispensed for this out of pharmacies. That's not including the 503B, which is on top of that. The estimated is over 1 million doses of prescriptions of this have been made historically from that perspective. If this is removed, you are taking away physicians' use of this for over 1 million times. Next slide. I would also note, and you heard this at the presentations at the very beginning, there is no expanded access potential program for this because there's no ongoing study for that, so it's irrelevant. The other thing I would hear is from an IND. FDA has reviewed CJC for an IND. In fact, it is listed on clinicaltrials.gov. One of the reviews for INDs, as was noted earlier, is safety. So if this product is too unsafe to be used now, that means FDA, when it put forward its IND, put forward a product that was too unsafe to be studied at that point as well. If it was okay to be studied at that point, should be allowed to be used now as well. Next slide. Here's the actual data that is used from FDA's own materials. Next slide. I would also point, as you just heard, real world evidence in this perspective has been used to be approved. I will note real quick, Charlie Ganney last time read what is real world evidence and accepted by the FDA. We made sure on that last slide, real world evidence was met what Charlie Ganney read. Thanks.
The committee will now turn its attention to address the task at hand. The open public hearing portion of this meeting is now concluded, and we will no longer take comments from the audience. The committee now will turn its attention to address the task at hand, the careful consideration of the data before the committee as well as the public comments. We will now proceed with the question to the committee and panel discussions. I would like to remind public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel. After I read each question, we will pause for any questions or comments concerning its wording. Slide 2, please. We will proceed with our first question, which is a voting question. We'll be using an electronic voting system for this meeting. Once we begin the vote, the buttons will start flashing and will continue to flash even after you've entered your vote. Please press the button firmly that corresponds to your vote. If you are unsure of your vote or you wish to change your vote, you may press the corresponding button until the vote is closed. After everyone has completed their vote, the vote will be locked in. The vote will be then displayed on the screen. The DFO will read the vote from the screen into the record. Next, we will go around the room, and each individual who voted will state their name and vote into the record. You can also state the reason why you voted as you did, if you want to. We'll continue in the same manner until all questions have been answered or discussed. Are there any issues or questions from the panel about the wording of the voting question? So the question is, Section 503A, bulk drug substances lists CJC-1295 related bulk drug substances. FDA's evaluation addressed 5 CJC-1295 related bulk drug substances, which include 2 active moieties, CJC-1295 free base and CJC-1295 DAC free base, and 5 different BDSs. FDA proposes to use a single voting question to address them as a group. Do committee members agree to use a single vote to address the group of CJC-1295 related bulk drug substances discussed today? Yes or no? Okay. If any member of the committee votes no, the FDA will take separate votes on each of these substances. If voting yes, the committee members will vote on the substance as a group and will proceed to answer 1 additional voting question. If voting no, committee members will vote on each of the substances separately and will proceed to answer 5 additional voting questions. Are there any issues or questions from the panel about the wording of the voting questions? Does anyone online have any questions regarding the voting? If there are no further questions or comments concerning the wording of the question, we will now begin the voting process. Please press the button on your microphone that corresponds to your vote. You will have approximately 20 seconds to vote. Please press the button firmly. After you've made your selection, the light may continue to flash. If you're unsure of your vote or wish to change your vote, please press the corresponding button again before the vote is closed.
Good morning, Takia speaking, DFO. For the record, there are 12 yeses, 1 no, and 0 abstentions. Thank you.
Okay. Now that the vote is complete, we'll go around the table and have everyone who voted state their name and vote into the record.
Todd Durham, I voted yes.
Hi, Alan Veda. I voted yes.
Robin Bogner, I voted yes.
Brian Serumaga, I voted yes.
Elizabeth Rebello, I voted yes.
Kathleen Gura, I voted yes.
Linda McElhaney, I voted no.
Tim Fensky, I voted yes.
Ken Berman, I voted yes.
Kurt Jensen, yes.
For the panel members that are online who voted for this session, Dr. Goulart, please state your name and your vote for the record.
Adnan Goulart, I voted yes.
Dr. Goulart, Takia speaking. You may be muted. Please state your name and your vote for the record.
I've unmuted myself. Are you able to hear me? Hello?
I can hear you, Dr. Gulor.
Thank you. Yes, Padma Gulor, and I voted yes.
Okay. One moment, please. Okay, continuing on to Dr. Gupta. Please state your name and your vote for the record.
Dr. Anita Gupta, and I voted yes.
For the people who are in the room, I believe the public can hear those who are speaking. Sorry, one moment, please. Hi, Takia speaking. I do apologize for the audio issues inside the room. I do believe that the audience outside the room can hear what the virtual panel members are saying. I'm sorry, I'm hearing an echo inside the room. We will continue to Dr. David Cook. Please state your name and your vote for the record.
David Cook, I voted yes.
Great, thank you so much.
Since one or more panel members voted no, we will proceed with questions 1B through 1F. Slide 4, please. Section 503A, Bulk Drug Substances List, CJC-1295, related bulk drug substances. FDA is proposing that CJC-1295 free base not be included on the 503A bulks list. So CJC-1295 free base be placed on the list. If voting yes, you're recommending FDA should place CJC-1295 free base on the 503A bulks list. If voting no, you're recommending FDA should not place CJC-1295 on the 503A bulks list. Are there any questions or issues from the panel about the wording of the voting question? Okay. If the substances Okay. If voting yes, you're recommending FDA should place CJC-1295 free base on the 503A bulks list. If you vote no, you're recommending FDA should not place the bulk drug substances on the 503A bulks list. If the substance is not on the list when the final rule is promulgated, compounders may not use the drug for compounding under Section 503A unless it becomes the subject of an applicable USP or national formulary, monograph, or a component of an FDA-approved drug. Are there any issues or questions from the panel about the wording of the voting question?
Oh, Dr. Berman.
I'm sorry, maybe it's just me, but that's different from what the question says in Part B. Part B says FDA is proposing free base not be included on the bulk list.
Question 1B is FDA is proposing that CJC-1295 free base not be included. So if you're voting yes, you're recommending FDA
not included.
That's correct. Yeah, so we need to change that. It's
correct as written. The question is, should CJC-1295 free base be placed on the list? So if you vote yes, you're recommending it should be placed on the list.
Yeah. But actually, you read the question here, it says not. Right. We give our this is Gail Bormel. We give the recommendation of FDA first. Right. That's just what we are recommending. But because there's a lot of confusion when there are negatives in a question, we then go ahead and ask the question, are you recommending that FDA place CJC-1295 on the bulks list? If yes, that's what you're recommending. If you're saying no, then you're recommending it not be placed on the list. You have to read the question separate from our recommendation. Got it. Thank you for the clarification. Are there any further questions regarding the vote? If there are no further questions or comments concerning the wording of the vote, the wording of the question, we will now begin the voting process. Please press the button on your microphone that corresponds to your vote. Reminder, if you are unsure of your vote or wish to change your vote, please press the corresponding button again before the vote is closed.
Takia Stevenson, DFO. For the record, there are 0 yeses, 13 noes, and 0 abstentions. Thank you. Hand it back to the chairperson.
Now that the vote is complete, we will go around the table and have everyone who voted state their name, vote, and if you want to, you can state the reason why you voted as you did into the record.
Todd Durham, I voted no.
Alan Veda, I voted no. It just seemed like there really wasn't enough evidence to prove it.
Robin Bogner, I voted no.
Brian Sarumaga, I voted no.
Elizabeth Urbello, I voted no.
Kathleen Gura, I voted no.
Linda McElhaney, I voted no.
Tim Fensky, I voted no.
Ken Berman, I voted no.
Kurt Jensen, no.
We'll now take our Dr. Galore.
Padma Galore, I voted no.
Dr. Gupta.
Anita Gupta, I voted no.
Dr. Cook.
David Cook, I voted no.
Thank you. We will proceed with question 1C. Slide 5, please. Question 1C. Section 503A, Bulk Drug Substances List, CJC-1295, related bulk drug substances. FDA is proposing that CJC-1295 acetate not be included on the 503A bulk drugs list. The question is, should CJC-1295 acetate be placed on the list? If voting yes, you're recommending that FDA should place CJC-1295 on the 503A bulks list. If voting no, you recommend that FDA should not place CJC-1295 acetate on the 503A bulks list. If the substance is not on the list when the final rule is promulgated, compounders may not use the drug for compounding under Section 503A unless it becomes a subject of an applicable USP or National Formulary Monograph or a component of an FDA-approved drug. Are there any issues or questions from the panel about the wording of the voting question? If there are no further questions or comments concerning the wording of the question, we will now begin the voting process. Please press the button on your microphone that corresponds to your vote. Reminder, if you're unsure of your vote or wish to change your vote, please press the corresponding button again before the vote is closed.
Takia Stevenson, DFO. For the record, there is 1 yes, 12 noes, and 0 abstentions. Thank you. Hand it back to the chairperson.
Now that the vote is complete, we will go around the table and have everyone who voted state their name, vote, and if you want, you can state the reason why you voted as you did into the record.
Todd Durham, I voted no.
Alan Veda, I voted no.
Robin Bogner, I voted no.
Brian Serumaga, I voted no.
Elizabeth Rebello, I voted no.
Kathleen Guerra, I voted no.
I voted yes.
Tim Fensky, I voted no.
Ken Berman, I voted no.
Kurt Jensen, no.
Dr. Galore?
Padma Galore, I voted no.
Dr. Gupta?
Anita Gupta, I voted no.
Dr. Cook?
David Cook, I voted no.
Thank you. We will proceed with question 1D. Slide 6, please. Question 1D, Section 503A, Bulk Drug Substances List, CJC-1295, related bulk drug substances. FDA is proposing that CJC-1295 DAC freebase not be included on the 503A bulks list. The question is, should CJC-1295 DAC freebase be placed on the list? If voting yes, you're recommending FDA should place CJC-1295 DAC on the 503A bulks list. If voting no, you're recommending FDA should not place CJC-1295 DAC on the 503A bulks list. If the substance is not on the list when the final rule is promulgated, compounders may not use the drug for compounding under Section 503A unless it becomes the subject of an applicable USP or national formulary monograph or a component of an FDA-approved drug. Are there any issues or questions from the panel about the wording of the voting question? If there are no further questions or comments concerning the wording of the question, we will now begin the voting process. Please press the button on your microphone that corresponds to your vote.
Takia speaking, DFO. For the record, there are 0 yeses, 13 noes, and 0 abstentions. Thank you. Hand it back to the chairperson.
Now the voting is complete. We'll go around the table and have everyone who voted state their name, vote, and if you want to, I encourage you to state the reason why you voted as you did into the record.
Todd Durham, I voted no.
Alan Veda, I voted no.
Robin Bogner, I voted no.
Brian Serumaga, I voted no.
Elizabeth Rebello, I voted no.
Kathleen Guerra, I voted no.
Linda McElhaney, I voted no.
Tim Fensky, I voted no.
Ken Berman, I voted no.
Kurt Jensen, no.
Dr. Galore?
Padma Galore, I voted no.
Dr. Gupta?
Anita Gupta, I voted no.
Dr. Cook?
David Cook, I voted no.
Thank you. We will proceed with question 1E, slide 7, please. Question 1E, section 503A, bulk drug substances list, CJC-1295 related bulk drug substances. FDA is proposing that CJC-1295 DAC acetate not be included on the 503A bulks list. The question at hand is, should CJC-1295 DAC acetate be placed on the list? If voting yes, you're recommending FDA should place CJC-1295 acetate on the 503A bulks list. If you're voting no, you're recommending FDA should not place CJC-1295 DAC acetate on the 503A bulks list. If the substance is not on the list when the final rules are promulgated, compounders may not use a drug for compounding under Section 503A unless it becomes the subject of an applicable USP or National Formulary Monograph or a component of an FDA-approved drug. Are there any questions or issues from the panel about the wording of the voting question? If there are no further questions or comments concerning the wording of the question, we will now begin the voting process. Please press the button on your microphone that corresponds to your vote.
Takia Stevenson, DFO. For the record, there are 0 yeses, 13 noes, and 0 abstentions.
Now that the vote is complete, we'll go around the table and have everyone who voted state their name, vote, and if you want to, state the reason why you voted as you did into the record.
Todd Durham, I voted no.
Robin Bogner, I voted no.
Brian Serumaga, I voted no.
Elizabeth Rebello, I voted no.
Kathleen Guerra, I voted no.
McElhaney, I voted no.
Tim Fensky, I voted no.
Ken Berman, I voted no.
Kirk Jensen, no.
Dr. Galur?
Dr. Galur, I voted no.
Dr. Gupta?
Anita Gupta, I voted no.
Dr. Cook?
David Cook, I voted no.
Thank you. We will now proceed with question 1F. Slide 8, please. Question 1F, Section 503A Bulk Drug Substances List, CJC-1295 related bulk drug substances. FDA is proposing that CJC-1295 DAC trifluoroacetate not be included on the 503A bulks list. The question at hand is, should CJC-1295 DAC trifluoroacetate be placed on the list? If voting yes, you're recommending FDA should place CJC-1295 DAC trifluoroacetate on the 503A bulks list. If you're voting no, you're recommending FDA should not place CJC-1295 DAC trifluoroacetate on the 503A bulks list. If the substance is not on the list when the final rule is promulgated, compounders may not use a drug for compounding under Section 503A unless it becomes the subject of an applicable USP or National Formulary Monograph or a component of an FDA-approved drug. Are there any issues or questions from the panel about the wording of the voting question? If there are no further questions or comments concerning the wording of the question, we will now begin the voting process. Please press the button on your microphone that corresponds to your vote.
Zacchaeus Davidson, DFO. For the record, there are 0 yeses, 13 noes, and 0 abstentions. Thank you.
Now that the vote is complete, we'll go around the table and have everyone who voted state their name and vote. And if you want to, you can state the reason why you voted as you did into the record.
Todd Durham, I voted no.
Alan Veda, I voted no.
Robin Bogner, I voted no.
Brian Sarumaga, I voted no.
Elizabeth Rebello, I voted no.
Kathleen Gura, I voted no.
Linda McElhaney, I voted no.
Tim Fensky, I voted no.
Ken Berman, I voted no.
Kirk Jensen, no.
Dr. Galur?
Padma Galore, I voted no.
Dr. Gupta?
Anita Gupta, I voted no.
Dr. Cook?
David Cook, I voted no.
Thank you, everyone. We will now take a quick 10-minute break. Panel members, please remember that there should be no discussion of the meeting topic during the break amongst yourselves or with any member of the audience. We will reconvene at 11 o'clock Eastern Time for the AOD 9604 related bulk drug substances topic. Thank you. Good morning, everyone. Thank you for resuming your seats. Before we begin the AOD 9604 related bulk drug substance topic session, panel members who will be in this topic will introduce themselves by stating their names and affiliation. We'll begin with Dr. Billington.
Good morning, Charles Billington. I'm Chief of Endocrinology and Metabolism at the Minneapolis VA Healthcare System and Director of Obesity Programs at the University of Minnesota and the Minneapolis VA Healthcare Systems.
Thank you. Dr. Yanofsky.
Susan Yanofsky. I'm co-director of the Office of Obesity Research at the National Institute of Diabetes and Digestive and Kidney Diseases.
Thank you. We will now proceed with the FDA presentation on AOD 9604 related bulk drug substances from Dr. Emily Neerum and Benny Matthew.
Good morning. My name is Emily Neerum. I'm a clinical analyst with the Pharmacy Compounding Review Team in the Office of New Drugs, and I will be presenting AOD 9604 related bulk drug substances with my colleague Benny Matthews from the Office of Pharmaceutical Quality. Next slide. I would like to recognize the evaluation team as well as the contributions of many other FDA colleagues. Our special thanks to the Division of Diabetes, Lipid Disorders, and Obesity in OND. Next slide. AOD 9604 related bulk drug substances were nominated for inclusion on the 503A bulks list. The nominations provided inconsistent information regarding the specific BDS proposed, AOD 9604 free base or AOD 9604 acetate. Later in our presentation, these different forms will be presented in detail. These are being evaluated for the treatment of obesity. The proposed products were oral capsule, subcutaneous injection, and transdermal topical cream. The nominations were withdrawn. However, due to safety concerns, which will be later explained, FDA is evaluating the substances at its discretion. Next slide. We have evaluated publicly available data on the physical and chemical characteristics, historical use, effectiveness, and safety in the compounding of these substances. Next slide. I will now turn it over to Benny to discuss physical and chemical characterizations of AOD 9604 related substances.
Thanks, Emily. Next slide. As Emily mentioned, there are two AOD 9604 related BDS conflated within the two nomination packages. They are AOD 9604 free base and AOD 9604 acetate. This table highlights the differences between these two forms. Unicode and Cas number are only available for AOD 9604 free base. Molecular formula, molecular weight, and chemical structure are unique for each BDS. However, the active moiety is same for both BDS, which is AOD 9604 free base. Next slide. This table highlights the detailed information submitted by the two nominators with the relevant information identified by the FDA. These nominations were later withdrawn. First nominator nominated AOD 9604. Unicode is not provided. Certificate of analysis is provided for AOD 9604. Cas number, molecular formula, molecular weight, and chemical name provided in the nomination package or certificate of analysis match with the free base form. All this information refer to same BDS. On the other hand, we notice inconsistent information in the second nomination package. AOD 9604 was nominated. However, certificate of analysis accompanied with the nomination package was for AOD 9604 acetate, which is not the same nominated BDS. Unicode, Cas number, molecular formula, molecular weight, and chemical name provided in the nomination package or certificate of analysis match with the free base form. Since AOD 9604 free base and AOD 9604 acetate are two distinct BDS, we evaluated both forms individually due to the safety concern. Next slide. I start with AOD 9604 free base. It is a hexadecapeptide. Thyrosin is linked at the N-terminal end of 15 amino acids fragment of human growth hormone. Two cysteine amino acids are cyclized to form a disulfide bridge, which may lead to peptide degradation. AOD 9604 free base is reported as a white solid powder, and it is soluble in water up to 2 mg in 1 ml. There is no USB drug substance monograph for AOD 9604 free base. It is recommended to store in a dry place to protect against water and moisture. It should be stored in closed containers and stable for 3 years at -18 degrees centigrade, 1 year at 0 to 7 degrees centigrade, and 6 months at 7 to 30 degrees centigrade. In general, peptides are sensitive to product formulation, process, and environment conditions, which may lead to aggregation and degradation. Next slide. There is potential for impurities present in AOD 9604 free base, such as peptide-related degradation impurities and peptide synthesis process-related impurities. For example, starting materials, residual solvents, coupling reagents, activators, or catalysts. There is a potential for immunogenicity risk when formulated in an injectable dosage form. The COA included tests such as appearance, solubility, identification, peptide purity, water content, and assay. Testing results for the control on impurities, aggregates, bioburden, and bacterial endotoxins were not included. In addition, these nominators propose other dosage forms such as transdermal cream and oral capsule. However, critical attributes associated with these dosage forms were not included in the submitted COA. Here we conclude that AOD 9604 free base is not well characterized due to lack of certain critical characterization data specific to AOD 9604 free base, potential immunogenicity risk when formulated in an injectable dosage form for subcutaneous administration, and lack of information on critical attributes associated with other proposed pharmaceutical dosage forms such as transdermal cream and oral capsule. Next slide. AOD 9604 acetate. It is the acetate salt of AOD 9604. Similar to AOD 9604 free base, the disulfide bond between the two cysteine amino acids of AOD 9604 acetate may also lead to degradation. It is reported as a white solid powder, and its solubility in water is very similar to its free base form. It has no USB drug substance monograph. The acetate salt form is recommended to store in a sealed container at 2 to 8 degrees centigrade for less than 6 months storage and at -20 degrees centigrade for more than 6 months storage. As similar to its free base form, AOD 9604 acetate may lead to aggregation and degradation. Potential impurities can be present, including peptide-related degradation impurities and peptide synthesis process-related impurities. Next slide. As similar to its free base form, there is a potential for immunogenicity risk associated with AOD 9604 acetate. The submitted COA controls several attributes, including related substance. Largest single impurity is controlled at less than or equal to 1%, and total impurities at less than or equal to 2%. However, the nature of the individual impurities that can be present at less than or equal to 1% is not provided. All other information is similar to AOD 9604 free base. In conclusion, AOD 9604 acetate is not well characterized due to the reasons previously discussed for the free base form. Next slide. I conclude my presentation here on the first evaluation criteria, which is physical and chemical characterization. Thank you. I will hand over to Emily.
Thank you, Benny. Sorry, can we go back one slide? Thank you. The non-clinical and clinical references reviewed do not clearly identify whether the substances discussed in the salt is the salt formulation or the free base. Therefore, in the next sections, the substance will generally be referred to as AOD 9604. Next slide. Here's what we found on historical use in compounding. AOD 9604 was first developed and patented by Metabolic Pharmaceuticals in the late 1990s in Australia for use in obesity. Based on outsourcing facility reports, none reported compounding any product containing AOD 9604. The extent to which it has been used in compounding is unclear. However, it has been widely advertised by medical spas and wellness clinics. One clinic indicated that peptides are prepared and delivered to your doorstep. Next slide. These are marketed online for various uses listed here, and in combination with other APIs as injectables and oral formulations, and have been used in sports as a doping agent. These substances are not recognized in the national medical registries or other foreign pharmacopoeias. Next slide. In conclusion, there is some evidence of compounded AOD 9604's use. Compounders have prepared injectable and oral formulations for a variety of uses, and they are marketed by medical spas and wellness clinics. Next slide. Now I'll discuss pharmacology and PK information. Based on non-clinical data, AOD 9604 decreases lipogenesis and increases lipolysis in obese rodents. In studies in obese rats and mice, authors noted a decrease in weight gain. It has no effect on plasma glucose or insulin levels or glucose oxidation in rats and mice. The mechanism of action is unknown, but unlikely to involve growth hormone receptors. Next slide. Based on non-clinical information, in vitro study found AOD 9604 was no longer detected after a 1-hour incubation in human serum. An in vivo study in pigs, IV had a half-life of 3 minutes, and oral had high bioavailability. FDA did not identify clinical studies in humans assessing pharmacokinetics. Next slide. I'll now provide a brief overview of obesity. This is a chronic condition that increases the risk for heart disease, diabetes, and cancer. Diagnosis is based on medical history and a high BMI. Treatment may involve the various FDA-approved options as listed on the slide. Next slide, please. I will now present studies on effectiveness for obesity. Wilding described 3 studies comparing AOD 9604 with placebo. These were part of the drug development by Metabolic Pharma in adults with obesity. One study via IV route once weekly for 4 weeks in 23 subjects. Another was via oral route once weekly for 4 weeks in 16 subjects. The third study, route and dosing were not specified, but administered for 1 week in 36 subjects. In all studies, authors noted AOD 9604 did not show statistically significant weight loss compared to placebo. Study limitations include insufficient details about methodology and study results, a small sample size, and unclear study parameters. Next slide. HERD was an abstract on a study in 300 adults with obesity who received oral AOD 9604 or placebo for 12 weeks. Authors state weight loss over 12 weeks was greater with AOD 9604 than placebo with a nonlinear dose response. We know this claim is based on small differences between groups, and interpretation of this study is limited by the minimal data in the abstract, insufficient details about the study methodology, and their results. Next slide. Next will be the option study. It is important to note this was conducted by Metabolic Pharma during drug development for AOD 9604 for the treatment of obesity. A double-blind, placebo-controlled study in 502 adults with obesity received oral AOD 9604 or placebo once daily for 24 weeks, along with a diet and exercise program. Primary efficacy endpoints included weight loss after 12 weeks. Study results reported no significant difference in weight loss between placebo and AOD 9604. Per the company, weight loss compared to placebo was too low to reach statistical significance. Next slide. Continuing with the option study. In 2007, the study summary and results were publicly announced by Metabolic Pharma. Quote, the phase 2B trial results for its drug, AOD 9604, do not support the commercial viability of the drug as a treatment for obesity. Development of the drug for this condition is terminated, end quote. We did not find details of the study design and preliminary efficacy results in the published medical literature. Next slide. In conclusion, in most of the studies we identified, AOD 9604 failed to show benefit for weight reduction when compared to placebo. Drug development was terminated for obesity because of a failed study. Therefore, there is a lack of evidence to support effectiveness for the treatment of obesity. There are multiple FDA-approved drug products for use in weight reduction in patients with obesity. Next slide, please. We will now switch gears to discuss safety. For non-clinical safety, Moore and Kenley summarized 3 toxicity studies with AOD 9604. A 4-week study in rats via IV showed decreased weight gain, thymus mass, and thymic cortical width. A 6-month study in rats via oral route showed decreased lymphocytic count, decreased osteocalcin levels at week 13, and increase at week 26 in females, increased creatinine and triglycerides in males. A 9-month study in monkeys showed vacuolation in hepatocytes. Note that the publication does not provide the underlying data. Therefore, it is difficult to interpret the author's conclusions. Next slide. In conclusion, for non-clinical safety, the molecular target and the mechanism of action underlying the pharmacological effects remain unknown, making it difficult to assess the biologic plausibility of the pharmacological effects reported in the different studies. Findings suggest that clinically relevant safety signals may develop with systemic exposures. Non-clinical studies available at the time of the evaluation were too limited to inform safety considerations for the potential clinical uses of AOD 9604. Next slide. The FAIRS and CARES retrieved no adverse events. Next slide, please. For clinical safety, Steyer et al. Presented a summary of IV and oral AOD 9604 in 6 placebo-controlled studies. Safety monitoring included interview of subjects for AEs, measurement of vital signs, laboratory parameters, ECG, anti-AOD 9604 antibodies in the blood, and serum levels of IGF-1. It is important to note these studies were funded by Metabolic Pharma. Next slide. For the IV route, there were 2 studies in a total of 38 subjects given single doses of AOD 9604 and or placebo. Reported were 3 AEs, severe in intensity: feeling of tightness in chest, deemed possibly related to AOD 9604 per authors, and 2 AEs in the placebo group with no additional details provided. AEs most reported included hypoglycemia, headache, euphoria, fatigue, dizziness, nasopharyngitis, and cough. Authors concluded safety profile of AOD 9604 was comparable in all treatment groups. Next slide. Continuing with Steyer, for the oral route, in 4 studies, patients received AOD 9604 for up to 24 weeks. Serious AEs reported in subjects receiving AOD 9604 include the following. After single doses, diarrhea and pneumonia were reported. Diarrhea was deemed possibly related to the substance. In a 12-week study, basal cell carcinoma, moderate lipoma, and other cancers, per authors not related to the substance. In a 24-week study, distribution of SAEs were similar among the groups with no details provided. Other AEs reported are listed here. Authors concluded safety profile of AOD 9604 was comparable in all treatment groups. Next slide. Steyer noted that for all studies, no statistically significant changes in laboratory parameters, ECG changes, or vital signs in any treatment group. No statistically significant differences in IGF-1 levels among the treatment and placebo groups. Anti-AOD 9604 antibodies were not detected in the selected patients who received oral doses for up to 24 weeks. We note the limitations of the study include lack of sufficient details, such as detailed description and breakdowns of AEs, intervention and outcome, and/or a short-term study duration. Next slide, please. For these substances, an additional safety concern is immunogenicity. The AOD 9604s consist of 16 amino acids. Peptides may elicit an immune response. This response may be enhanced when peptides are given via the subcutaneous route. They pose a risk for immunogenicity, potentially amplified by aggregation as well as potential peptide-related impurities. The nomination did not include, and FDA did not identify, information about these substances to suggest that they do not present these risks. Next slide. To conclude on safety, based on available studies in humans, on oral AOD 9604, serious AEs include diarrhea, chest tightening, and various types of cancers. FDA did not find information on the proposed subcutaneous and transdermal use in humans. Obesity is a chronic condition which may need long-term repeated treatments. There is insufficient information to support the long-term use in these patients. There is limited information to assess immunogenic safety risk for oral administration and no information on subcutaneous and transdermal topical routes. These are peptides containing 16 amino acids, and sequences of this length have the potential to be immunogenic. There are multiple currently available FDA-approved drug products indicated for weight reduction in patients with obesity. Next slide. On balance, physiochemical characterizations, information on historical use, lack of evidence of effectiveness, and safety information weigh against both AOD 9604 free base and AOD 9604 acetate being added to the 503A bulks list. Although available data suggests that these substances have historically been used at compounding, FDA's proposal is based on lack of data related to physical and chemical characterization, concerns related to potential immunogenicity risks, lack of evidence of effectiveness for use in obesity, and insufficient safety information. The lack of evidence of effectiveness and limited safety data in the existence of FDA-approved drugs for use in obesity, particularly in light of the fact that obesity increases the risk for many serious diseases and health conditions, weigh against these BDSs being added to the 503A bulks list. Next slide. After considering the information currently available, a balancing of the criteria weighs against AOD 9604 free base and AOD 9604 acetate being added to the 503A bulks list. Next slide. Thank you very much. This concludes my presentation.
Thank you, Drs. Miriam and Matthew. Slide 12, please. We will now take clarifying questions to the presenters. When acknowledged, please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. Are there any clarifying questions for the presenters? Dr. Billington.
I have a couple. So for Dr. Matthew, oh, there she is. I think you pointed out that for the acetate form, the drug should be kept frozen and specific criteria for how to be frozen and such. But when the drug is going to be made up for use, it's going to have to not be frozen. So once that happens, how long does it last? Do we have any information at all? You provided some evidence that said in serum, it's gone right away. And in the bloodstream, we have nothing, no pharmacology, nothing. So we don't know if this drug exists at all in the body once we give it. Am I right?
Actually, we know the storage condition at for 6 months of storage. Actually, it is 2 to 8 degrees centigrade, and for the more than 6 months storage, it is minus 20 degrees centigrade. So we don't have much information on that one other than that.
Right. So once we bring it out of storage and make it up into
no, we don't have
a form to be given to a person, we don't know what happens to it.
No, we don't have much information.
And it might well all be gone based on what we know. All right, which is interesting. Now, for Dr. Nero, my question was about the mechanism of action. In the briefing document, it mentions, first of all, that this material, AOD numbers, was intended to be looking like the active part of growth hormone. But if I read the document correctly, the evidence is that it doesn't bind a growth hormone receptor and it doesn't increase IGF-1, which would suggest that it's not doing anything in the growth hormone line, which you properly concluded in your slide. So here we have a drug, a peptide that was created thinking it was going to work like growth hormone, but it doesn't do what growth hormone normally does, except that it has these effects on lipolysis, which now seem to happen for a drug that we don't know how long it lasts for no reason that we know of. Am I characterizing this fairly accurately?
Everything you said was correct, yes. To our understanding.
All right. Well, I guess I have nothing further to say.
Thank you. We have a question from Dr. Yanofsky.
Yes, thank you. Thank you for very clear presentations. I think the data on efficacy or lack of efficacy are very clear. I did notice that in the rodent model, the weight loss was thought to be, at least in part, due to beta-adrenergic signaling. But am I correct that even though they measured vital signs in patients in the clinical trial, they didn't provide any data about changes in pulse or blood pressure? This would be something we'd be particularly concerned about in patients with obesity.
This is Dr. Nair. If I understand your question, you're asking if we saw any measures of blood pressure in any of our clinical studies? We did not.
they didn't report actual, they said no changes in vital signs, but they didn't report the actual data.
Correct. We don't have the raw data from any of the studies.
That would be a concern. Thank you.
Thank you.
Are there any further clarifying questions for the presenters? Being none, we will now begin with the open public hearing session. Both the Food and Drug Administration and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the open public hearing session of the advisory committee meeting, FDA believes it's important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement to advise the committee of any financial relationship that you may have with the product, and if known, its direct competitors. For example, financial information may be the payment by the bulk drug supplier or compounding pharmacy of your travel, lodging, or other expenses in connection with your attendance at the meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. The FDA and this committee place great importance in the open public hearing process. The insights and comments provided can help the agency and this committee in their consideration of the issues before them. That said, in many instances and for many topics, there'll be a variety of opinions. One of our goals for today is for this open public hearing to be conducted in a fair and open way, where every participant is listened to carefully and treated with dignity, courtesy, and respect. For those presenting virtually, please remember to unmute and turn on your camera when your OPH number is called. For those presenting in person, please step up to the podium when your OPH number is called. As a reminder, please speak only when recognized by the chairperson. Thank you for cooperation. We have 6 speakers, and I invite speaker number 1 to the podium. Please state your name and any organization that you are representing for the record. You have 3 minutes.
Good morning. My name is Dan Denai. I'm the Chief Executive Officer of Avexius Health Solutions, which is a national network of over 10,000 medical providers, all representing many different disciplines of medicine. In reviewing the recording from last PCAC session, I was a bit taken aback by the lack of understanding of what compounding is and what it does and how it is so vitally and beneficial to our patients and an intricate part of our healthcare system. During our last PCAC session, we continued to bring up this evidence, real-world evidence, continued to be mentioned in the last session. Allow me to read just one real-world testimonial submitted by one of our patients. In September 2022, our world stopped with a single devastating phone call from my daughter's school. Just shy of her 11th birthday, she had endured a harrowing 4.5-minute seizure in class, leaving us in a storm of fear and uncertainty. We stood on the precipice of a new reality, grappling with the unknowns of her health and the long-term implications of that terrifying moment. After months of tests, she was finally diagnosed with Rolandic epilepsy and was told that that was the good kind of epilepsy. However, in that moment, I found myself in a familiar but terrifying place, sitting across from a physician who lacked the insight and education to provide a comprehensive understanding of all options that were available. Instead, we were handed a choice of prescriptions, 2 medications accompanied by a daunting list of potential side effects we did not want for our daughter. Yet, as I had fought for my own health a decade ago, I was determined to advocate fiercely for my daughter. We forged a new path, rejecting the conventional treatment in favor of a functional medical protocol tailored to her unique needs. Central to her healing journey were 2 pivotal peptides. Then came the fall of 2023, a time when those 2 peptides were overnight reclassified, making it impossible for our family to continue to receive the medication from accredited and certified pharmacies. We could have used research only or not for human use peptides, but we chose not to risk our child's health and were forced to go without. My husband and I were engulfed by anger and a felt profound sense of betrayal. It became painfully clear that the very institution meant to safeguard patients were instead prioritizing their own interest over the well-being of children like mine. In that moment, I understood the cruel reality. This was not about protecting my daughter. This is about preserving a broken system. While we remain grateful for her aches for the countless parents who wander through misinformation, lack of options, and who are lost and desperate for answers, the constraints on our medical freedoms, the barriers preventing access to life-saving therapies is nothing short of criminal. This journey is not just ours. It is a call to arms for all who believe in the rights to choose the best path for their loved ones. Together, we must rise up and demand the freedom to pursue healing and to reclaim our power and to protect the future of our children. We've accumulated thousands of other testimonials like these, stories that truly are life-changing because of the power of peptides. Thank you for your time.
Thank you. Speaker number 2, please state your name and any organization that you're representing for the record. You have 3 minutes.
Yes, Jim Lavelle, Chair, International Peptide Society, Co-Chair, American Academy of Anti-Aging Medicine. Go ahead, next slide. So the main point being brought out about AOD is accurate. It's simply for lipolysis, does not have any effect on growth hormone. We've seen 159,000 prescriptions written in the last year that we categorized through the 9 pharmacies that we contacted from providers who thought that it must have been doing something, because patients don't buy things that cost money if it's not having some benefit for them. I would also add that it's typically used as an adjunct, so it's never just given alone, instructed at least with the International Peptide Society. It's given in conjunction with diet, exercise, and maybe other medication, so that makes the water a little bit murky. Let's go ahead to the next slide. 4 preclinical animal, 2 human clinical trials, very well done on the assessment of the trials. There were some shortings on all those, but it is reported to be GRAS status as a dietary supplement. I know that doesn't apply for exactly for 503A, but it is noted as being safe as a dietary supplement. And let's continue. Next slide, please. I do want to note that in the studies, they saw no immunogenicity changes. Of course, there was no effect on serum IGF-1 because it does not, it's a fragment of growth hormone. The fragment is specific to the lipolytic activity of growth hormone. It's not specific to the release of either release of growth hormone or mimicking growth hormone. So one of the big complaints, or at least that we heard at the last PCAC committee, is there's nothing on immunogenicity. In this particular case, the no anti-AOD 9604 antibodies were detected in any of the patients selected for the antibody assay. So the summary of this from the authors, at least in terms of safety and tolerability, that it was indistinguishable from placebo and had none of the adverse effects associated with the use of growth hormone. And of course, one of our biggest concerns is obesity, as was mentioned earlier. Everything we can do to try to keep people at a better body composition weight, less visceral fat, is important. Let's go to the next slide. Once again, what we do at International Peptide Society is look at level of evidence for, obviously it's limited with AOD 9604. It's limited. At the same time, 159,000 prescriptions have been written for by providers who have the right to write for a prescription. And instead of people going and getting...
Thank you. We're at time. Appreciate it. I invite speaker number 3. Please state your name and any organization that you're representing for the record. You have 3 minutes.
My name is Tom Wynn. I'm a pharmacist with Pharmaceo Pharmacy, and I'm here today just representing pharmacists. Next slide. Next slide. I'm going to blow through these first ones. We already went through this already when I talked about bulk powder, so next slide. Next slide. Next slide. Next slide. I want to stop here, and I have AO2 acetate up there only because that's the one I'm most familiar with, but it can apply to all. One of the things that was in the briefing was the FDA, again, was talking about aggregates, immunogenicity. The study that they used was one that was done in Europe, and it was 10 of the most frequently encountered falsified peptide drugs in Belgian market. And they investigated the peptides and acquired 3 different suspect illegal internet pharmacies. They took product from that and tested it. And using that product, that's where they came up with different aggregates and things that were there if they found within those peptides. AOD and CJC were both in there. So, of course, if we're talking about a counterfeit pharmacy, we're probably going to find a lot of different solvents in that we're not normally going to see. They found arsenic, they found lead. Next slide. Next slide, please. So that's the significance is that we don't know what the starting part was for those counterfeit pharmacies. Did they start with a crude peptide, or was it something like more what you'd use for a reagent, or is it one that we use in pharmacies? It's about 98%, as you saw in most of the C of A's that they were popping up earlier. And again, biopeptide North America are FDA registered and FDA inspected, and the purity is documented. Purity, peptides with a higher purity are less likely to vary from batch to batch and are less likely to aggregate than less pure peptides. So what I'm getting at is if you're taking that information from a counterfeit pharmacy and you're applying that to all peptides, you can't do that. We don't know the purity, what the starting point was. The other question I had is, I think it was slide 26 of the immunogenicity talk. There was a comparison of commercially available peptide to a compounded peptide. It was not stated what peptide that was. It can't be any of the, can't be CJC or AOD because there's not a commercially available product. Which one did they use? It wasn't stated. We also don't know how they got it. Did they get it from a counterfeit pharmacy? Did they have a pharmacy submit it? We just kind of brushed over that and went on, and everybody took it for granted. But that information, we need more information about it. If I was presenting information, I would have to probably also say, what lab was it at? Did they do a method validation? And all these other aspects that were not commented in that particular slide on immunogenicity. And we keep thinking about, you know, that can occur, and it's constantly going to be can occur. There's no talk of prevalence. You know, how prevalent is it? We don't know. I haven't seen it in any of the studies that they actually presented, both in the briefing and the ones that they're talking about in the original talk. So I don't really know how much that's actually going to occur is what I'm getting at. And usually it can be something to where it's going to be some type of...
We're at time. Thank you very much.
Thank you.
I invite speaker number 4. Please state your name and any organization that you're representing for the record. You have 3 minutes.
Hello, my name is Dr. Edwin Lee. I have no financial relationship. I'm a board certified endocrinologist in Orlando. Next slide. I want to start with a brief, next slide, leave it here, brief history of AOD 9604. And I'm going to basically just say AOD instead of saying AOD 9604. It's a tongue twister. So AOD is a 16 amino acid peptide. It comes from the very end of human growth hormone. It does not have any growth hormone effect. We've heard before, it does not raise IGF-1. It does not react to the growth hormone receptor. And in the late 1990s, Dr. Frank Ng and his colleague developed AOD. Next. So they were excited, Dr. Frank Ng, that the AOD had an effect on lipolytic effect and with weight loss in rats and mice. Next slide. In human studies, there was over $50 million investigated, poured into to see if this actually works in humans. And you've heard that it basically was ineffective, but it was very well tolerated in close to 1,000 patients. Next slide. So one positive spin is that there's no negative effect on carbohydrate metabolism. We also heard that there's no anti-AOD antibodies detected in any of the patients. Next slide. So this is something you can find on the internet. I typed in AOD and GRAS status, and this was in June of 2012. And it says AOD 9604 receives GRAS status. AOD receives pivotal GRAS status recognition to enter U.S. market, conditional on publication of our existing safety data. Next. I'm trying to basically get a copy of this because I cannot find in the FDA website this GRAS status that was issued. So I'm trying to reach out to the companies out there to see if I can get a copy of that letter. So I want you to focus on this picture here. You have two different men. They're exact same weight, exact same body mass index, same height. It's just that they have different body composition. One's leaner, the one's heavier. So think of AOD stimulating lipolysis and reducing lipogenesis, and that's what has been shown to do. Next. Unfortunately, we can't find that data with the 6 randomized control studies that were done in Australia and UK because they never published it.
Thank you very much.
Thank you.
I'd like to invite speaker number 5 to please come to the podium. Please state your name and any organization that you are representing for the record. You have 3 minutes.
Sure. My name is Lee Rosebush. If you go to OPH slides number 1 for number 1 presenter, please. I'm a PharmD, a pharmacist, doctor of pharmacy, and a lawyer, and represent some of the pharmacies associated with this lawsuit. I wanted to bring up 3 things very quickly as the slides are being brought up. We went through this earlier. There was 4 specific criteria that is supposed to be reviewed. Notice, based on some of the questions that were just asked, one of those criteria did not include mechanism of action. And in fact, if we had mechanism of action as one of the requirements, you could take off both OTC and FDA-approved drugs. For example, I'd ask you, what's the mechanism of action of Tylenol, acetaminophen? Or what's the mechanism of action of lithium, right? The mechanism of action of a drug does not necessarily apply to the efficacy associated with that drug. 2, from this perspective, we've also tried to get USP monographs. That was mentioned. We've met with Brian and the USP team to ask to develop a USP monograph for all 3 of these peptides, and were denied. The reason we were denied, because they're not FDA approved. If they were FDA approved, we wouldn't be here. Accordingly, you can't get a USP monograph, nor can you get FDA approval to compound the product. It's a solution that you could never be able to meet. Next slide. 3rd, from this perspective, as we've mentioned before, there is multiple criteria. If you look at FDA's own briefing documents, they say there's a long history associated with compounding this product. Accordingly, that is met. Next slide. We've gone through the criteria. Next slide. I would point out, number 3, in the history in this perspective, it's been used 158,000 times. Next slide. Here's the COA from that perspective, the immunogenicity aspects associated with this. It can also be used orally. That's been skipped over, so you'd also have that as taken care of. I would then ask the question, why is this necessarily being taken off now? If you can jump ahead a few slides down to where we say real-world evidence, you will see in this perspective that FDA has approved products previously using real-world evidence. In fact, keep going. FDA has approved, as we talked about this last time with Charlie, as little as 14 patients has been used before previously by the FDA to approve a product using real-world evidence. In this situation, we have 158,000 prescriptions. We have provided the data, again, that Charlie has mentioned in his readout from last time associated with that data. In addition, as was pointed out to here, there, in this case, a phase 2B clinical trial that has 536 trials. Here's the material with the footnotes showing that FDA has actually approved using real-world evidence, and as short as 14 patients, and it's done 13 times in the last 5 years. Next slide. Now, again, as we are appearing here, you'll see very little adverse events associated with those 158,000 prescriptions. Next slide. Next slide. Next slide. We talked about efficacy. That was the last question from the association here. I would point to that second bullet point. Notice in this case that, yes, there's been questions associated with the overall statistical significance, but there was weight loss. And in fact, females responded better. As I mentioned last time in the PCAC hearing, my understanding is from this administration, no federal regulator should ever stand between a healthcare provider and a female when she's making her healthcare decisions. Evidence number 1.
We're at time.
Thank you.
I'd like to invite our last speaker who will be participating virtually. Speaker number 6. Please state your name and any organization that you're representing for the record. You have 3 minutes.
Thank you. First slide, please. Good morning. My name is Dr. Brad Jordan, and I'm the Associate Vice President of Regulatory Policy for Eli Lilly and Company. I'm happy to deliver remarks today on behalf of Lilly in opposition of the addition of AOD 9604 to the FDA bulk substances list. AOD 9604 is a synthetic fragment of the C-terminal region of human growth hormone designed in the early 1990s. However, clinical development of this molecule for the treatment of obesity was terminated in 2007 because the molecule failed to show a significant therapeutic effect in clinical trials. In fact, the CEO of Metabolic, the company that initially tried to develop the drug, noted that their clinical program had been successful and that it clearly demonstrated that the molecule was not effective. Other attempts at studying this molecule for tissue repair and pain have similarly failed to demonstrate efficacy. AOD 9604 is a complex peptide, and the nomination of this molecule to the bulk list is essentially an effort to circumvent the FDA drug approval process. At the same time, false and misleading advertising by compounding pharmacies touting the effectiveness of this molecule for weight management and tissue repair is being used to deceive patients. Lilly urges the PCAC to vote against the replacement of this molecule, AOD 9604, on the bulk list. The nomination of AOD 9604 is part of a trend that is especially concerning to Lilly. Over the past several years, some compounding pharmacies have created unproven, untested, and unregulated protein and peptide-based therapies, including many at very large scale. Notably, we are now seeing entities marketing compounded versions of complex investigational drugs. We also continue to see entities mass marketing compounded versions of complex macromolecules in completely untested formulations, including sublingual versions of medicines that have only been approved and studied as injectables, and versions such as combination products created seemingly at random. These practices obviously circumvent the FDA approval process, are not grounded in science, and potentially present dangerous practices that could harm patients and threaten public health. We are also concerned that some compounding pharmacies are routinely making unsupported claims for their products and circulating them widely online. False and misleading advertising for compounding drugs has led to a massive black market alternative to authentic FDA-approved medicines. These marketing practices pose a significant threat to the integrity of the FDA approval system and put patients at risk. Lilly urges FDA and the committee to halt all mass compounding of protein and peptide-based therapies and to consider placing all such complex macromolecules on the difficult to compound list in order to gain control of the current situation and prevent further risks to patients. Thank you.
Thank you. The opening public hearing portion of this meeting is now concluded, and we will no longer take comments from the audience. As we will now take additional comments or questions from committee members. Do members of the committee have questions or comments regarding anything that was presented in this segment of the session? Dr. Sycamore.
Yes, Brian Serumaga from the USP. I would like to make a comment for the committee because USP was mentioned in one of the submissions from the general public. USP does create monographs, as you all know, for bulk drug substances, also monographs for FDA-approved products that are conventionally manufactured, and also monographs for compounded preparations. The guidelines for submitting materials for creation of monographs at USP are clearly elucidated on the USP website. So I'm reading to information that's in the public domain. USP encourages all entities to submit information either for the creation of a new monograph or the revision of an existing official monograph. However, it's important for the committee to also remember that USP is not a drug regulator. So it is USP policy not to create monographs for materials or products that are not FDA approved or that are indeed not legally marketed. So that might be one of the reasons why sometimes when we get nominations for monographs at USP, we decline. Okay, USP is not a drug regulator. So the monograph creation process at USP cannot be used to circumvent the new drug approval process. that is legally required in the Food Drug Cosmetic Act. Thank you.
Are there any further? Go ahead.
FDA would like to make 2 comments. This is Daiva Shetty. I want to comment on the real world data. The information on the use alone is not the same thing as real world evidence. Real world evidence is the clinical evidence about the usage and potential benefits or risk of medical product derived from analysis of real world data. Various sources of real world data can be analyzed in non-interventional studies, including registries, electronic health records, and medical claims. The information provided by the presenters are simply numbers of prescriptions filled by unidentified pharmacies that do not identify the use, dose, route of administration, and duration of exposure. And most importantly, it does not provide any data related to the safety and effectiveness of the drug. Therefore, we consider this information as describing historical use and compounding. Thank you. And Emily.
Hi, this is Emily Nerium. I just wanted to make a comment on the GRAS status that we heard. AOD 9604 is not currently listed on FDA's GRAS list and has not been through FDA's GRAS determination based on our search. In the metabolic pharma company document that we saw projected on GRAS status, it appears if you read the whole document that this is a self-determination. Thank you.
Okay, Gabrielle Kozlov.
Hi, thank you very much. I just wanted to add an additional comment. Some access concerns were raised earlier, and I wanted to remind the committee of our presentation earlier today about the availability of the IND process for access. As was shared earlier, individuals may be able to receive investigational products containing AOD 9604 related substances under an IND provided applicable requirements are met. Individuals can receive investigational products containing this substance either through a clinical trial or through FDA's expanded access program. And expanded access is a potential pathway for a patient with a serious disease or condition to gain access to an investigational product for treatment outside of clinical trials. I just wanted to remind the committee of that as well.
Any further questions or comments?
And hi, Russ Westdyke, OPQ FDA. I should also comment on some of the impurities, impurity COAs, and Daniela's slide regarding the immunogenicity as that was raised. We do acknowledge the receipt of additional C of A's, and in some cases they provided additional information that was incremental to our own and helpful. However, in some cases it also included additional information that raised additional concerns. We need to continue our evaluations of that. It was mentioned that Daniela's slide highlighted a peptide, and that's true. I mentioned that too, her next to last slide. I can't name that peptide because it continues to be an active investigation, but it's not one of the 3. I would have thought that was immediately obvious because we were including marketed product. If it was marketed product, we wouldn't be sitting here talking about it. They could compound it. What we found in that case was marketed product had a very clean impurity profile and lower immunogenicity potential. And in the case of compounded samples, which we pulled, we found a much higher impurity profile and potential for immunogenicity. I can't name it, but it's a peptide that is similar to what you see here, a large molecule peptide. That's as much as I can say there. Thank you.
Additional questions or comments? Anyone online? The committee will now turn its attention to address the task at hand, the careful consideration of the data before the committee, as well as the public comments. We will now proceed with the question to the committee and panel discussions. I'd like to remind public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel. After I read each question, we will pause for any questions or comments concerning its wording. Slide 11, please. We'll proceed with our second question, which is a voting question. We'll be using electronic voting system for this meeting. Once we begin the vote, the buttons will start flashing and will continue to flash even after you've entered your vote. Please press the button firmly that corresponds to your vote. If you're unsure of your vote or you wish to change your vote, you may press the corresponding button until the vote is closed. After everyone has completed their vote, the vote will be locked in. The vote will then be displayed on the screen. The DFO will read the vote from the screen into the record. Next, we will go around the room and each individual who's voted will state their name and vote into the record. You can also state the reason why you voted as you did if you wish to. We'll continue in the same manner until all questions have been answered or discussed. Question 2. Section 503A Bulk Drug Substances List, AOD 9604 Related Bulk Drug Substances. FDA's evaluation addressed 2 AOD 9604 related bulk drug substances, which include 1 active moiety and 2 different BDSs. FDA proposes using a single voting question to address them as a group. Do committee members agree to vote on AOD 9604 related bulk drug substances discussed today and AOD 9604 as a group? Yes or no? If any member of the committee votes no, FDA will take a separate vote on each of these substances. If voting yes, committee members will vote on the substance as a group and will proceed to answer voting question. If voting no, committee members will vote on each of the substances separately and will proceed to answer additional voting questions. Are there any questions from the panel about the wording of the voting question? If there are no further questions or comments concerning the wording of the voting question, we will now begin the voting process. Please press the button on your microphones that corresponds to your vote. You'll have approximately 20 seconds to vote. Please press the button firmly after you've made your selection. The light may continue to flash. If you're unsure of your vote or wish to change your vote, please press the corresponding button again before the vote is closed.
Hello, this is Takia Stevenson's DFO speaking. Dr. Yanovsky, please, reminder to please, for voting members online to please submit their vote via email. Thank you.
I've done so. I can do it again.
Takia Stevenson, DFO speaking. For the record, there are 12 yeses, 0 nos, and 0 abstentions. Thank you. Hand it back to the chairperson.
Thank you. Now that the vote is complete, we'll go around the table and have everyone who voted state their name and vote.
Todd Durham, I voted yes.
Ellen Veda, I voted yes.
Robin Bogner, I voted yes.
Brian Seromaga, I voted yes.
Elizabeth Rebello, I voted yes.
Kathleen Guerra, I voted yes.
Linda McElhaney, I voted yes.
Tim Fenske, I voted yes.
Charles Billington voted yes.
Dr. Galur?
Padma Galur, I voted yes.
Dr. Gupta?
Anita Gupta, I voted yes.
Dr. Yanofsky?
Susan Yanofsky, I voted yes.
Since everyone voted yes, we will proceed with question 2A. Slide 12, please. Question 2A, Section 503A Bulk Drug Substances list AOD 9604 related bulk drug substances. FDA is proposing that AOD 9604 and AOD 9604 acetate not be included on the 503A bulks list. The question at hand is, should AOD 9604 and AOD 9604 acetate, freebase and acetate, be placed on the list? If voting yes, you're recommending FDA should add 9604 related bulk drug substances on the 503A bulks list. If voting no, you're recommending FDA should not place AOD 9604 related bulk drug substances on the 503A bulk list. If the substances are not on the list when the final rule is promulgated, compounders may not use the drug for compounding under Section 503A unless it becomes subject of an applicable USP or National Formulary Monograph or a component of an FDA-approved drug. Are there any issues or questions from the panel about the wording of the voting question? If there are no further questions or comments concerning the wording of the question, we will now begin the voting process. Please press the button on your microphone that corresponds to your vote. You will have approximately 20 seconds to vote. Please press the button firmly. After you've made your selection, the light may continue to flash. If you're unsure of your vote or wish to change your vote, please press the corresponding button again before the vote is closed.
Takia Stevenson, DFO. For the record, there is 1 yes, 11 no's, and 0 abstentions. Thank you.
Now that the vote is complete, we will go around the table and have everyone who voted state their name, vote.
I believe there was an error because I voted no on my email. For 2A.
Yeah, no, this is Takia speaking. One moment, please. Thank you for letting us know. Apologies for the delay. To Kia speaking. Just one moment. We'll continue in a moment. Apologies for the delay. Takia speaking. Correction for the total for the record, there were 0 yeses and 12 noes and 0 abstentions. We'll go around the table for everyone to state their name and vote into the record. Thank you.
Todd Durham, I voted no.
Alan Veda, I voted no.
Robin Bogner, I voted no.
Brian Sarumaga, I voted no.
Elizabeth Rebello, I voted no.
Kathleen Guerra, I voted no.
Linda McElhaney, I voted no.
Tim Fenske, I voted no.
Charles Billington voted no.
Dr. Padma Galur,
Padma Galur, I voted no.
Dr. Anita Gupta,
Anita Gupta, I voted no.
Dr. Susan Yanofsky.
Susan Yanofsky, I voted no.
Thank you. Since one or more panel vote. Okay, so we'll now break for lunch and we'll reconvene at 1 o'clock Eastern time. Please take any personal belongings with you. Remember that there should be no discussion of the meeting topic during the lunch break amongst yourselves or with any member of the audience. Additionally, those panel members participating in the remaining topic discussions are planned to rejoin at 12:45 Eastern time. Those that are joining us virtually are also included in that to ensure you are seated before we reconvene at 1 o'clock. Thank you. Enjoy lunch. Welcome. My name is Elizabeth Rebello, and I'm serving as acting chair for this meeting. Before we begin, I'd like to remind everyone to silence their cell phones and devices. And we'll begin with the Thymosin alpha 1 related bulk drug substances topic session. Panel members who will be in this topic will introduce themselves by stating their names and affiliation, and we'll begin by those that are here in person, followed by virtual participants. Dr. Clark.
Nina Clark, Loyola University Chicago. I'm an adult infectious disease doctor.
Dr. Zaha.
Mark Saja, Emory University School of Medicine.
Dr. Ganny.
Mark Ganny, NIH, Bethesda, Maryland.
Dr. Chris.
Mark Chris, Thoracic Medical Oncology from Memorial Sloan Kettering.
Dr. Powell.
Alice Powell, NIID, NIH.
Dr. Siberry.
George Siberry, Pediatric Infectious Diseases and Chief Medical Officer at Office of HIV/AIDS, USAID.
And for those of us that are joining virtually, Dr. Brian Lee.
Brian Lee, a hepatologist at University of Southern California.
Dr. Janet Lee.
Janet Lee, Washington University, St. Louis, Pulmonary Critical Care Medicine.
Dr. Corbett.
Hi, Amanda Corbett, a pharmacist at the University of North Carolina, Eshelman School of Pharmacy.
Dr. Gans.
Hi, Dr. Haley Gans, Pediatric Infectious Disease at Stanford.
Dr. Monge, if I missed you, apologies.
Cecilia Monge, Medical Oncology, National Cancer Institute, NIH.
Great, thank you. We will now proceed with the FDA presentation on Thymosin alpha 1 related bulk drug substances from Drs. Elizabeth Hankla and Jing Lee.
Good afternoon. My name is Elizabeth Hankla. I'm a clinical analyst with the Pharmacy Compounding Review Team in the Office of New Drugs, and I will be presenting Thymosin alpha 1 or TA1 related bulk drug substances with my colleague Jing Lee from OPQ. Next slide. I would like to recognize the evaluation team, as well as the contribution of many other FDA colleagues. We would like to give a special thanks to the 7 review divisions that we worked with across the Office of New Drugs and in the Center for Biologics and Research. Next slide. TA1-related bulk drug substances were nominated for inclusion on the 503A bulks list. However, the nomination was subsequently withdrawn. FDA is evaluating the substances at its discretion. The product proposed in the nomination is a 3 mg per ml injection for subcutaneous administration. Next slide. TA1 freebase and TA1 acetate were evaluated for the 12 uses listed in this slide. Next slide, please. The criteria we consider in our evaluation for the 503A bulks list are physical and chemical characterization, historical use and compounding, safety, and available evidence of effectiveness or lack of effectiveness. Next slide. I'll now turn the presentation over to my colleague, Jing Lee.
Thank you, Ben. Due to conflicting information about the different TA1 BDSs in the nomination package, it's unclear which TA1-related BDS was nominated. Therefore, first we summarize the basic information on 2 BDSs, including TA1 freebase and TA1 acetate in this table, to show some differences between them. From the table, you can see the unichord Cas number, molecule formula, and the molecule weight are different between these two substances, but they have the same active moiety, which is TA1 free base. Next slide, please. This table shows detailed information submitted by the nominator, as well as relevant information identified by FDA. As you can see, the nominated BDS is referring to TA1 free base, and the Unicord matches TA1 free base. But the COA provided is for TA1 acetate. However, the basic information in the COA, including Unicord, Cas number, molecule formula, molecule weight, and the chemical name, are all referring to TA1 free base. Because TA1 free base and TA1 acetate are two distinct BDSs, their different physical and chemical properties can impact patient safety and product efficacy. We evaluated both TA1 free base and TA1 acetate, and will present them in the following slides. Next slide, please. Let's first talk about the TA1 free base. TA1 free base is N-terminal acetylated 28 amino acid peptide. In 1977, isolation and the purification of TA1 free base from thymosin fraction 5 of calf thymos was reported. Later, chemically synthesized TA1 free base named thymophysin has been produced. TA1 free base is a white to off-white lyophilized powder. It's soluble in water up to 2 milligrams per ml. There is no USP drug substance monograph for TA1 free base. In terms of storage and stability, manufacturer recommends storing TA1 free base below -18 degrees Celsius. Upon reconstitution, TA1 free base in solution is stable between 2 to 7 days at 4 degrees Celsius. And for future use, the solution should be stored below -18 degrees Celsius. Like other peptides mentioned in the morning, TA1 free base is also sensitive to product formulation, process, and the environment conditions, which may lead to aggregation and degradation. Next slide, please. In addition, there may be some peptide-related and peptide synthesis process-related impurities presented in TA1 free base, such as starting materials, residue solvents, coupling reagents, etc. Because there's no COA provided in the nomination, FDA also could not find information from public domain regarding potential impurities and peptide-related aggregates that may be presented in TA1 free base. Therefore, it's difficult to rule out the potential for immunogenicity associated with peptide-related impurities and aggregates, especially when formulated in the injectable dosage form for sub-Q administration. In conclusion, TA1 free base is not well characterized because lack of certain critical characterization data, including impurities, aggregates, bioburden, and bacterial endotoxins. In addition, there's a potential for immunogenicity, especially when formulated in injectable dosage form for sub-Q administration, due to potential for aggregation as well as peptide-related impurities in the BDS. Also, no information was provided regarding how to formulate proposed injectable dosage form at the concentration of 3 milligrams per ml using water as a solvent due to limited water solubility of TA1 free base. Next slide, please. TA1 acetate is an acetate salt form of TA1 free base. As shown in this slide, the physical and the chemical properties, including stability and the potential impurities for TA1 acetate are similar to those for TA1 free base. Next slide, please. The COA provided only includes identification, assay, water content, and acetate content. However, no critical characterization data for impurities, aggregates, bioburden, and bacterial endotoxins. In addition, the water solubility of TA1 acetate is 1 milligram per ml. Due to the same issues as mentioned in previous slides for TA1 free base, in conclusion, TA1 acetate is not well characterized. That concludes my presentation. Now I hand over to Beth.
Thank you, Jane. Next slide, please. Here's what we found on historical use in compounding. As mentioned previously, TA1 was first isolated from calf thymus in 1977. However, the earliest and extent of TA1 free base or TA1 acetate use in compounding is unknown. We found no evidence of compounded drug products containing TA1 free base or TA1 acetate in the published literature or in outsourcing facility reports. Compounded drug products containing TA1 have been marketed online and in the US as an injectable and nasal spray for use in numerous conditions as those listed in the slide. Next slide, please. According to a 2014 annual report from Cyclone Pharmaceuticals, which is a pharmaceutical company previously associated with the development of Zidaxin, or TA1 free base, TA1 is approved for use in countries in the Asia-Pacific region, Latin America, Eastern Europe, and the Middle East. According to the annual report, approvals are primarily for the treatment of hepatitis B and as a vaccine adjuvant, with additional approvals in certain countries for the treatment of hepatitis C or as a chemotherapy adjuvant. FDA is unable to independently verify these claims of approval in the specified countries. TA1 is not approved in the United States, Japan, or in Europe except for Italy. Additionally, TA1 is not recognized in the European or Japanese pharmacopoeias. Next slide, please. We will now discuss safety information. This slide presents some of the non-clinical safety information we identified from a Zidaxin product monograph. Of note, the monograph did not provide the underlying data for these safety conclusions. In terms of acute toxicity, a single sub-Q injection of TA1 up to 20 mg per kg generated no drug-related safety signals in mice, rats, and marmosets. In a repeat dose toxicity study, treatment of mice, rats, and marmosets with TA1 sub-Q doses up to 6 mg per kg per day for 13 weeks or 1 mg per kg per day for 26 weeks generated no drug-related safety signals. According to the monograph, TA1 did not produce genotoxicity signals in vivo and in vitro. Next slide. According to a published review article, Cyclone Pharmaceuticals successfully completed a lengthy segment 3 reproductive toxicology study, which is a study conducted in rodents to evaluate drug effects during the last trimester of pregnancy and the period of lactation. FDA did not identify non-clinical carcinogenicity studies with TA1. In conclusion, summaries of non-clinical toxicity studies available in a product monograph for Zidaxin that contain TA1 free base 1.6 mg per ml appear to suggest that TA1 free base did not induce safety signals in non-clinical toxicity studies. However, the underlying non-clinical data generated to support the safety of TA1 are not available for review. At the time of evaluation, FDA did not identify published non-clinical toxicity studies to inform safety considerations for potential clinical uses of TA1 acetate. Next slide, please. Here we present some information on the pharmacology of TA1. TA1 has immunomodulatory properties mediated at least in part by its interactions with toll-like receptor 9 on dendritic and lymphoid progenitor cells. Per non-clinical studies, TA1 suppressed tumor growth, suppressed viral infections, and decreased sepsis in vivo rodent models. Additionally, as post-treatment, TA1 increased the antibody titer generated by some vaccines in mice. We note that most non-clinical studies were conducted with fixed doses that, according to body surface area, translate to human equivalent doses higher than those used in most clinical studies. Finally, it's difficult to determine the minimal TA1 dose required to induce a pharmacological response due to lack of studies assessing dose-response relationships. Next slide, please. Here we present some information on the pharmacokinetics of TA1. In rats that received an IV injection of TA1, the half-life ranged from 1.9 to 3 minutes. The non-clinical PK profile of TA1 delivered via the nominated route of administration is unknown at this time. After sub-Q administration, in healthy men, TA1 was absorbed rapidly with a Tmax of approximately 2 hours and a serum half-life of approximately 2 hours. There was no evidence of accumulation using daily dosing for 5 days. Next slide, please. In terms of clinical safety, FDA search of the FAERS database of AEs for TA1 retrieved one report from a 46-year-old male receiving PEG interferon alpha 2A and TA1 for about 12 weeks as part of a clinical trial. The subject was hospitalized with anxiety, atrial fibrillation, and a transient decrease in his TSH levels. Interpretation of causality in this case is confounded by concurrent use of PEG interferon, as all 3 of the reported AEs are potential AEs described in US labeling. Next slide, please. In clinical trials, TA1 has been administered in daily doses ranging from about 1 to 16 milligrams, usually via sub-Q administration on a bi-weekly schedule, for treatment periods ranging from 1 day to 12 months. The most common dose in clinical studies was 1.6 milligrams via sub-Q administration. The most common AEs reported include local irritation, redness, and injection site discomfort. Next slide, please. On this slide, we note a few potential AEs reported in the medical literature in our review of the studies. These AEs included ALT flares in subjects with chronic hepatitis B, TSH abnormalities in subjects with chronic hepatitis C, nipple pain, fatal immune hemolytic anemia, and engraftment failure in hematopoietic stem cell transplant, or HSCT, recipients. Next slide, please. There are potential safety concerns with administering TA1 in patients who are undergoing deliberate immunosuppression. For example, in patients who are undergoing HSCT, TA1 could develop or worsen acute or chronic GVHD and lead to engraftment failure. Safety data for use of TA1 when given as a vaccine adjuvant with preventative influenza vaccines are insufficient to evaluate safety or assess the optimal TA1 dose and regimen and associated risks. Adding an immunomodulatory product such as TA1 to any vaccine could pose unknown safety concerns that warrant further evaluation. Next slide, please. We searched for products containing TA1 as an active ingredient licensed and marketed outside of the United States with publicly available labels. We identified 2 such labels. Information obtained from these 2 labels include warnings and contraindications when used in children, pregnant and lactating women, patients with autoimmune diseases, and immunosuppressed populations. The label for Indonesia includes information about transient increases in liver enzymes, characterized as flares, and further TA1 administration. Next slide. As described earlier this morning, immunogenicity is a concern for peptide products. This immunogenetic response may be enhanced when they are given via the sub-Q route of administration. The consequences of triggering an immune response may range from antibody responses with no apparent clinical manifestations to life-threatening and catastrophic reactions. TA1 may pose a significant risk for immunogenicity, potentially amplified by aggregation and peptide-related impurities. Importantly, we are unaware of data in humans to support the proposal for a 3 mg per ml solution, and it's possible that a more concentrated solution could lead to aggregation and therefore increased immunogenicity potential. Next slide. In conclusion, the use of TA1-related bulk drug substances in compounding may raise safety concerns. In most clinical studies, TA1 has not been associated with significant adverse events attributable to TA1 when administered in doses in the range of 1 to 16 milligrams via the sub-Q route of administration for up to 12 months. The most common adverse reactions reported included local irritation, redness, or discomfort at the injection site. Although TA1 has not been associated with significant AEs attributable to TA1 in the literature, there may be concerns about its clinical use in compounding. For example, it's not clear whether the administration of TA1 in patients undergoing HSCT could lead to the development or worsen GVHD and/or lead to engraftment failure. In addition, safety data are insufficient to evaluate the risks associated with the use of TA1 as a vaccine adjuvant with influenza vaccines licensed for use in the United States. Finally, as a peptide with 28 amino acids that is administered through the sub-Q route, it may pose a significant risk for immunogenicity. Importantly, the highest strength identified to date in clinical studies was 2 mg per ml, and it's possible a more concentrated solution could lead to aggregation and therefore increased immunogenicity potential. Next slide. Now I will present information on the effectiveness for each of the 12 uses we evaluated. Here we note that studies discussed in the next set of slides for effectiveness administered TA1 via sub-Q administration unless otherwise noted. As we need to cover a lot of information in our effectiveness discussion, we will take a brief 5-minute break after our discussion of COVID-19. Next slide, please. First, we will discuss hepatitis B. Hepatitis B infection can lead to chronic HBV, which can cause liver damage, cirrhosis, hepatocellular carcinoma, or HCC, and death. In clinical practice, sustained HBV DNA suppression and clearance of HBV surface antigen are associated with improved outcomes and used to assess therapeutic effect. Per professional guidelines, preferred therapies for chronic HBV include antiviral agents and PEG interferon. Oral antivirals, or NRTIs, entecavir and tenofovir, are administered long-term. 68 to 90% of patients achieve undetectable HBV DNA levels after 48 weeks. They have been shown to reduce the risk of chronic HBV complications. These newer NRTIs are generally associated with lower rates of resistance. PEG interferon is administered for a finite duration and is associated with lower HBV DNA suppression rates. Of note, TA1 is not mentioned in the treatment guidelines. Next slide, please. We identified several studies that administered TA1 as monotherapy compared to placebo, no treatment, or various doses of TA1. TA1 was administered at doses up to 1.6 milligrams twice weekly for up to 12 months. The 5 studies conducted from 1998 to 2005 reported mixed efficacy results. One randomized double-blind placebo-controlled trial showed no difference in undetectable HBV DNA rates measured 6 months after treatment. Other studies with less rigorous design showed undetectable HBV DNA rates of 15 to 30%. These studies were generally limited by small study sizes, lack of blinding, HBV DNA assays with a limit of detection much higher than current standards, i.e., assays used for quantification of HBV DNA were much less sensitive than current assays. And the interpretation of older studies was complicated by the use of composite endpoints. Next slide, please. We identified several studies that compared TA1 versus interferon alpha monotherapy, an older, currently non-preferred treatment for hepatitis B. In these 4 studies, authors generally observed higher rates of undetectable HBV DNA with the interferon group at the end of treatment versus TA1 group at the end of follow-up. Surface antigen data were not discussed. These studies had similar limitations to those discussed in the previous slide, as well as the use of an outdated comparator, interferon alpha. Of note, PEG interferon has replaced the use of standard interferon because it's more effective for serologic and virologic outcomes and is better tolerated with less frequent dosing. Additionally, standard interferon therapies have been discontinued from marketing in the United States. Next slide. The next 2 slides focus on the use of TA1 in comparison to current preferred therapies. There were 2 studies that compared PEG interferon monotherapy versus PEG interferon in combination with TA1. In a retrospective analysis from Song et al., the virologic response rates were not different between the 2 treatment arms. In the open label study from Kim et al., authors concluded that the addition of TA1 was not superior to PEG interferon alone. These trials had limitations as listed in the slide. Next slide, please. Finally, we identified 1 trial where TA1 was used in combination with entecavir. In this randomized open-label trial of entecavir monotherapy versus entecavir with TA1, undetectable HBV DNA rates were similar between groups at 52 and 104 weeks. Surface antigen clearance rates were also similar between groups. Per authors, results showed that combination therapy has a similar effect as entecavir monotherapy in mortality, decompensation rate, HCC incidence, virological response rate, and some additional measures. Next slide, please. In conclusion, we have insufficient evidence to determine the effectiveness of TA1 for HBV, which can be serious. Available studies are limited by design and the use of outdated assays. While information from available published studies suggest mixed efficacy results of TA1 monotherapy, we have limited information about its use with or an alternative to current preferred therapies. Available studies of TA1 with current preferred therapies, such as PEG interferon or entecavir, demonstrated unclear efficacy of TA1 and have limitations. There are FDA-approved drugs with established efficacy for the treatment of chronic hepatitis B. Next slide. Moving to hepatitis C. Chronic hepatitis C can lead to chronic infection, which can cause liver disease, cirrhosis, HCC, or death. The treatment goal is virologic cure, as evidenced by sustained virological response, or SVR. That is undetectable HCV RNA months after completing treatment. For treatment, currently recommended therapies are oral direct-acting antivirals, which are administered daily for 8 to 12 weeks. With direct-acting antivirals, SVR rates of greater than 90% are observed, with or without ribavirin. Older, less effective therapies include interferon, PEG interferon, and ribavirin. Next slide, please. We identified 1 study that used TA1 as monotherapy for chronic hepatitis C. In this early trial, no subject cleared HCV RNA. TA1 has also been studied in combination with older, non-preferred treatments. Trials of TA1 in combination with interferon, with and without ribavirin, were limited by study design, small size, older assays, and limited SVR data. In studies that reported SVR, rates ranged from 21 to 40%. 1 randomized blinded placebo-controlled trial of TA1 in combination with PEG interferon and ribavirin showed an SVR of 12.7% with the TA1 group versus 10.5% in the placebo group. Authors stated that the addition of Thymosin alpha-1 to the standard of care did not increase the on-treatment HCV viral response. Next slide, please. In conclusion, there are insufficient data to establish effectiveness of TA1 to treat HCV infection, which has the potential to be serious and life-threatening. TA1 was studied as an alternative to or in combination with older therapies with lower SVR than current standard of care oral direct-acting antivirals, for which SVR rates exceed 90% in many populations. There are available FDA-approved therapies with established efficacy for the treatment of chronic hepatitis C. Next slide. Next, I will discuss HIV. HIV is a virus that attacks the body's immune system. The key component of the immune deficiency associated with HIV is a marked reduction in CD4 positive T cells. Current professional treatment guidelines recommend initiating antiretroviral therapy as soon as possible after diagnosis to reduce HIV-related morbidity and mortality and to reduce transmission risk. Treatment-induced decreases in viral load have been shown to be highly predictive of meaningful clinical benefit and are validated in clinically meaningful surrogate endpoints. Thus, HIV viral load suppression is often a primary endpoint in HIV treatment trials, while CD4 T cell counts are included as secondary endpoints. Monitoring lymphocyte subsets other than CD4 T cell counts have not proven clinically useful and are not recommended. Next slide. FDA identified 5 published studies using TA1 in subjects with HIV. These studies are broken up into this slide and the next slide. Shuloff et al. Evaluated TA1 as monotherapy, while other studies evaluated TA1 as part of combination therapy with antiretroviral drugs. None of the 4 studies on this slide increased CD4 positive T cell counts when TA1 was added. Next slide, please. The 5th study by Garacci et al. Randomized subjects to 4 treatment groups. In the table, we can see the 4 treatment groups along with the mean CD4 T cell counts, pre-treatment and after 12 months. After 12 months of treatment, CD4 counts were significantly increased in group 4 and persisted for up to 18 months. However, small sample sizes, potential bias from the unblinded study design, and the unknown contribution of TA1 to the immunomodulatory effect, when combined with zidovudine and interferon alpha, limits the interpretability of the data. Next slide. In conclusion, there is insufficient evidence to conclude that TA1 is effective for the treatment of HIV infection. These published studies were not adequately designed. Additionally, CD4 T cell counts and/or laboratory-based immune parameters are not validated surrogate endpoints. None of these studies demonstrated statistically significant effects on clinical endpoints such as prevention of AIDS-defining illness or death, or on the validated surrogate endpoint of HIV viral load. Finally, there are numerous FDA-approved drug products for the treatment of HIV. Next slide. Next, I will discuss COVID-19. COVID-19 is the disease caused by SARS-CoV-2. Early in the clinical course, the disease is primarily driven by the replication of SARS-CoV-2. Later in the clinical course, it's driven by a dysregulated immune/inflammatory response to infection that may lead to further tissue damage and thrombosis. FDA was not able to find literature that discussed the use of TA1 in children with COVID-19 or adults with COVID-19 in the outpatient setting. Thus, our discussion focuses on the use of TA1 in adults with acute COVID-19 in the hospital setting. Next slide. This table lists the 4 meta-analyses included in the evaluation. Liu et al. 2022 reported that there was no association between TA1 treatment and mortality. Shang et al. 2023 reported that TA1 therapy had no statistically significant effect on mortality. Wang et al. 2023 reported that no differences were found in mortality or length of hospitalization between subjects who did and did not receive TA1. Sotaro et al. 2023 reported that use of TA1 was associated with a lower mortality rate, but treatment with TA1 did not reduce the need for mechanical ventilation, nor did it reduce the length of hospital stay. In conclusion, 3 of the 4 meta-analyses concluded that there was no decrease in mortality in subjects treated with TA1. Next slide. There were 3 additional references not discussed in the meta-analyses. Tuthill et al. 2023 was a prospective trial in adults with end-stage renal disease. This study has not been completed and results were not reported. Wang et al. And Wu et al. Were retrospective cohort studies in adults hospitalized with COVID-19. Next slide, please. Limitations for the studies include limited sample sizes and deficient study designs, as well as the use of other medications. In addition, most of the studies were conducted early in the pandemic, and it's unclear if the outcomes will be reproducible given the current circulating viral strain and changes in treatment since these studies were completed. There was heterogeneity between studies in the definition of disease severity and dose and frequency of exposure to TA1, making it difficult to compare study outcomes. Next slide. In conclusion, there is insufficient information concerning effectiveness to support the use of TA1 for the treatment of COVID-19. IDSA, NIH, and WHO treatment guidelines do not discuss the use of TA1 for COVID-19, and there are FDA-approved therapies with established efficacy for the treatment of COVID-19. Next slide, please. Thank you. This concludes this portion of our presentation. After a short break, we'll return to discuss the available evidence of effectiveness or lack of effectiveness for the remaining uses. Thank you.
Hi, this is Takia Stevenson, DFO. Just while we're on this brief break, just reminder, just for the panel members to please don't leave your seats or leave the Zoom room. We will resume shortly. Thank you.
Yeah. Yeah, I'm good to go. Okay, we just wanted to give everyone a couple minutes to digest, and I want to take a sip of water, so I appreciate the short break. Welcome back. We will continue our discussion. Oh, next slide, please. Perfect, thank you. We will now continue our discussion on the effectiveness of TA1. Next, we will discuss adjuvant to influenza vaccines. FDA identified 5 clinical studies evaluating TA1 as an adjuvant to influenza vaccine in elderly or patients on hemodialysis. Of these, 2 clinical studies were retrieved in abstract form and were not able to identify full-text articles. These 5 published studies administered TA1 at different time points in relation to vaccination with monovalent or trivalent influenza vaccines. Interpretation of effectiveness in these studies is limited, as will be discussed in the next slide. Next slide, please. Limitations of the studies for this use are listed. ELISA was used in most studies. However, ELISA is not an accepted methodology used by FDA to evaluate immune responsiveness. Studies had exploratory design without formal hypothesis testing and pre-specified immunogenicity or efficacy endpoints. Importantly, control groups were missing or inadequate, and data quality were difficult to interpret. For example, studies did not use influenza vaccines recommended in the US for use in the populations considered. Overall, the studies were not properly controlled and did not produce meaningful data on use of TA1 with influenza vaccines. In conclusion, there's a lack of sufficient evidence to determine any conclusions on the effectiveness of TA1 as a vaccine adjuvant. Next slide, please. Next, I will discuss malignant melanoma. We identified 4 published studies using TA1 in patients with metastatic melanoma, which is a subtype of melanoma. The first study, by Mao et al., is described in this slide. This study was an exploratory, multi-center, open-label randomized study in 488 patients with stage 4 melanoma and unresectable metastases. Patients with previous chemotherapy were not eligible for inclusion in this study. Patients were randomized to one of 5 treatment groups as shown in the table on the slide. Patients received various combinations of dacarbazine, interferon alpha, and TA1. The primary endpoint of the study was best overall response rate at 12 months. Overall response rates at study completion are shown in the table. The study failed to show a significant difference in the overall response rate with any TA1 containing regimen compared to the control group. Furthermore, in terms of the dose response, the lower response rate of group 3 compared with group 2 suggests that these findings may be due to chance. Importantly, the study was conducted when chemotherapy and either interferon or interleukin were the standard of care. Next slide, please. We identified 3 additional studies using TA1 in patients with metastatic melanoma. 2 single-arm studies where TA1 was used in combination with interferon alpha or interleukin 2, and 1 retrospective review. These studies were limited by their design, and it was not possible to identify the contribution of TA1 to the observed treatment effect. Next slide, please. In conclusion, the studies investigating the use of TA1 in melanoma to date are insufficient to demonstrate the effectiveness of TA1. Published studies use controlled data from inferior, outdated regimens. Statistically significant, clinically meaningful improvements in TA1-treated melanoma patients were not demonstrated. The existence of FDA-approved drugs to treat the disease and the lack of rigorous data demonstrating the benefit of TA1 for use in patients with malignant melanoma weigh against including TA1 on the list, particularly in light of it being a serious or life-threatening disease. Next slide, please. Next, I will discuss hepatocellular carcinoma, or HCC. We identified 2 studies that used TA1 with transarterial chemoembolization, or TACE. The first study was a single-arm study in 12 patients. However, the study had limitations including the study design and small sample size. The second study, from Gish et al., was a randomized pilot study in 25 patients. Patients were randomized to receive TACE plus TA1 or TACE alone for 24 weeks. Authors did not find a difference in the response rate or median overall survival between the treatment and control groups. The study was designed primarily as a safety study, and authors recognized the need for a larger phase 3 trial in this setting. Next slide, please. Next, we identified 1 study that administered TA1 after hepatectomy and with TACE to reduce recurrence. Chang et al. 2004 was a randomized controlled trial in 57 patients, 18 of which received TA1. Authors did not find a difference in the 1-year recurrent rate, but did find a difference in the median overall survival. Of note, the overall survival rates in the control arm are much shorter than would be expected based on other published reports, which raises concerns over the reported data. upon reanalysis of the data from this study, a Cochrane review published in 2011 did not find a statistically significant difference in either the overall survival or disease-free survival. Next slide, please. We identified 5 additional studies using TA1 in patients with HCC that are listed on this slide. 4 studies evaluated TA1 post-hepatectomy to reduce recurrence, and 1 study evaluated TA1 after liver transplantation to reduce recurrence. These studies were limited by their design, and it was not possible to identify the contribution of TA1 to the observed treatment effect. Next slide, please. Limitations of these studies include the retrospective design, small patient numbers, studies were not adequately designed to assess the study endpoints, and in some studies, patients received other therapy in combination with TA1 and were not designed to demonstrate the contribution of TA1. And they were mostly single-center studies that were geographically limited, thus the results may not be directly applicable to patients in the U.S. Next slide, please. In conclusion, there is insufficient evidence to demonstrate the effectiveness of TA1 as a treatment option for HCC. In the 2 randomized controlled trials identified to date, authors did not find a difference in the 1-year recurrent rate or response rate and median overall survival between the treatment and control groups. There are no FDA-approved products for HCC in the adjuvant setting. However, there are FDA-approved products for the treatment of unresectable HCC. Next slide. Next, I will discuss non-small cell lung cancer. 2 randomized controlled trials evaluated TA1 in non-small cell lung cancer. These studies are nearly 40 and 30 years old, respectively. Although there were improvements in both relapse-free survival and overall survival for the TA1 treated groups when compared with the placebo group in the 1985 study by Shuloff et al., we agree with the study authors' conclusions that definitive conclusions regarding the impact of Thymosin therapy on survival can only be ascertained in large-scale multi-institutional trials. Furthermore, these studies are inconsistent with contemporary management of patients with non-small cell lung cancer with placebo controls and without genetic testing. In the study by Salvati et al., the median overall survival in the ifosfamide chemotherapy arm was 16 weeks, and in the combined chemotherapy plus TA1 plus interferon arm was 24 weeks. However, based on the 2-arm study design where several drugs were administered, it's not possible to determine the contribution of TA1 administration to the overall treatment effect of the regimen. Next slide. We identified 5 additional studies using TA1 in patients with non-small cell lung cancer that are listed on this slide. These studies were limited by their design, and it was not possible to identify the contribution of TA1 to the observed treatment effect. Next slide, please. The ability of these studies to establish the effectiveness of TA1 in patients with non-small cell lung cancer are limited by small sample sizes, advances in staging and genetic characterization since these studies were conducted, and changes regarding U.S. standards of care and available therapies. Furthermore, the control therapies received are not consistent with U.S. standards of care for patients with non-small cell lung cancer without actionable genomic alterations. It's unknown whether the addition of TA1 to contemporary treatment regimens would have resulted in clinical benefit for these patients. Next slide. In conclusion, there is insufficient evidence to demonstrate the effectiveness of TA1 as a treatment option for non-small cell lung cancer. It is unknown whether the addition of TA1 to contemporary U.S. standards of care would have resulted in clinical benefit for the patients included in the clinical studies. Non-small cell lung cancer is a serious disease. There are several FDA-approved drug products which have contributed to a sharp decrease in the disease-related mortality. Next slide, please. Next, I will discuss sepsis. We identified 4 full-text articles in English using TA1 in subjects with sepsis. This slide describes the first 2 articles. Wu et al. Conducted a single-blind, randomized, controlled study to evaluate the efficacy of TA1 in 361 patients with severe sepsis. The study showed that in the time-to-event analysis, patients in the TA1 group survived longer after enrollment than the control group. However, it did not show a significant difference in the overall absolute reduction in mortality, ICU mortality, length of ICU stay, and duration of ventilation. Authors concluded that larger multi-center studies are needed. Pai et al. Conducted a post hoc analysis of the study by Wu et al. However, there were no results for the subpopulation of patients who received TA1. Next slide, please. The third study was a randomized control trial in septic shock patients who received TA1 plus blood purification versus control. All subjects received ulinistatin, which is not approved in the United States. Although the TA1 treated group showed significant improvements in the T lymphocyte subsets, inflammatory cytokines, and myocardial function, it showed no difference in survival. The fourth study by Wang et al. Was a small double-blind pilot study in patients with severe acute pancreatitis that showed a significant reduction in the rate of positive blood and abdominal drainage cultures and the rate of surgery. The patient population with severe pancreatitis is representative of only a subset of patients with sepsis. Next slide, please. We note several limitations. All clinical studies were conducted in China. Based on the studies reviewed in this section, treatment of sepsis in China may not be reflective of the U.S. medical practice. Due to several limitations, we conclude that there is insufficient information to support the effectiveness of TA1 for the treatment of sepsis. Published clinical trials show that TA1 may affect biomarkers of immune function. However, the majority do not provide evidence of meaningful clinical benefit of TA1 in the treatment of sepsis, reduction in mortality, or need for organ support. Next slide. Next, I will discuss infections after hematopoietic stem cell transplantation, or HSCT. HSCT can be defined as the transfer of hematopoietic stem cells from one individual to another, or the readministration of previously harvested cells to the same individual. Major causes of early morbidity or mortality for patients who undergo stem cell transplant are disease relapse, acute graft versus host disease, infection, regimen-related toxicity, and graft failure. Endpoints that can translate to meaningful clinical benefit and survival include decrease in the infection rate and hematopoietic recovery endpoints. For example, time to neutrophil recovery in addition to decrease in infection rate. Next slide. We identified 2 published studies and 1 abstract using TA1 in subjects who were stem cell recipients. The first study from Peruccio et al., 2010, was a single-arm study in 14 stem cell recipients who received TA1 for 16 weeks post-transplant. Authors concluded that TA1 may favorably affect immune function, but recognize that larger number of subjects and longer follow-up are needed to assess its impact on survival. The second study was published as a meeting abstract. Peruccio et al., 2011, was a study in 30 stem cell recipients who received TA1 from the day of transplantation for 16 weeks. Authors reported a potential improvement in non-relapse mortality, but the details needed to assess the impact of TA1 are lacking because it was a meeting abstract. The third study by Ding et al. Was a case series of 8 stem cell recipients that received TA1 or standard of care for 4 weeks. The infection rate did not decrease in this small study, and it appears that the infection rates were increased in the TA1 treated group. Next slide, please. Limitations of the studies include heterogeneous populations, small sample sizes, limited duration of follow-up, lack of clinically meaningful endpoints, and unclear clinical relevance of the measured markers, among others. In conclusion, these studies do not provide evidence that TA1 reduces infection or infection-related mortality after HSCT. Next slide. Next, I will discuss COPD. We identified 2 published studies using TA1 in patients with COPD exacerbations. The first was a double-blind randomized trial in patients with acute exacerbation of COPD who received unspecified routine complex treatment and either TA1 or placebo for 4 weeks. Authors state that pulmonary function and blood gas indicators improved in both groups, but were more pronounced in the experimental group. However, no details of the results were provided. The second study was a study in elderly patients with an acute attack of COPD with respiratory failure who received theophylline sustained release or theophylline sustained release and TA1 IV for 4 weeks. Authors state that both groups improved after treatment with better effects observed in the study group in terms of pulmonary function, blood gas indicators, and exercise ability. Authors noted that further trials are required prior to application in clinical practice. Next slide. Both studies had limitations including small sample sizes with short durations, lack of sufficient details about statistical methodology and other study design elements, lack of meaningful clinical endpoints, and a lack of details on other COPD medications. In conclusion, there is a lack of evidence to support the effectiveness of TA1 for the treatment of COPD. While authors claim that studies suggest there are better effects observed in those who receive TA1 compared to the control group, the available information has limitations as shown in the list above. Next slide, please. Finally, I will touch on myalgic encephalomyelitis and chronic fatigue syndrome, or ME/CFS. FDA did not identify any data to support the effectiveness of TA1 in the treatment of ME/CFS. Next slide. Considering all of the effectiveness information, we conclude there's a lack of evidence to support the effectiveness of sub-Q administered TA1 freebase and TA1 acetate for the evaluated uses. None of the clinical practice guidelines for U.S. health professionals recommend TA1 freebase or TA1 acetate. Studies on the serious and life-threatening conditions considered in the evaluation of effectiveness of TA1 were inconclusive and limited by small sample sizes and study design deficiencies. There are multiple FDA-approved drug products indicated for use in the treatment of many of the conditions evaluated. Next slide. On balance, the physiochemical characterization, information on historical use, lack of evidence of effectiveness, and safety information identified for both TA1 freebase and TA1 acetate weigh against them being added to the 503A bulks list. In particular, FDA's proposal regarding these substances is based on the fact that TA1 freebase and TA1 acetate are not well characterized from a physiochemical perspective, and it's unclear how it would be possible to formulate the proposed injectable form as an aqueous solution with a concentration of 3 mg per ml. There is insufficient safety information on the use of the substances and of lack of information about immunogenicity risks. There's also a lack of evidence of effectiveness of these substances for use in the conditions evaluated, and the existence of FDA-approved drugs to treat most of these conditions, particularly in light of them being serious and/or life-threatening conditions. Next slide, please. After considering the information currently available, a balancing of the 4 evaluation criteria weighs against TA1 freebase and TA1 acetate being added to the 503A bulks list. Next slide. Thank you very much. This concludes our presentation.
Thank you, Drs. Hanklin and Lee. Slide 3, please. We will now take clarifying questions to the presenters. When acknowledged, please remember to state your name for the record before you speak and direct your question to the specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. Finally, it'd be helpful to acknowledge the end of your question with a thank you and the end of your follow-up question with that is all for my questions. We can move on to the next panel member. Are there any clarifying questions for the presenters? Dr. Gura.
Yes, hi. Thank you for a great overview. I'm kind of confused because there is a product known as Adaxan. Where does that fit in to the whole scenario? Because that is FDA approved. And this does have orphan status, correct? I'm just wondering how these all fit together. Thank you.
Oh, okay. Okay, so Zadaxin or Thymosin alpha-1, any products containing Thymosin alpha-1 are not approved in the United States by FDA. There are foreign marketed products. As mentioned in the historical use section, Zadaxin is approved in other countries. In terms of orphan drug designation, there are 4 orphan drug designations for Thymosin alpha-1. But just a note here that designation as an orphan drug qualifies sponsors for certain incentives, but it's a separate process from FDA approval. So the drugs for rare diseases must still go through the same rigorous scientific review process as any other drug for approval or licensing. Does that help?
Well, like I said, yeah, I was just looking online and I see a lot of conflicting information. That's why I'm trying to double check what the real deal is. Because I mean, right now I'm seeing like so many citations saying it is FDA approved. And I haven't had a chance to go into my, LexiComp databases, but I'm just curious, that's because I am confused.
Yeah, Elizabeth Hankley again. It is not FDA approved.
Thank you.
We have
if I may, this is Donette Stoss from Jazz. If you go to the Drugs at FDA website, there's a really nice database where you can actually look at drugs that are actually approved by the FDA.
Thank you. Dr. Gans online has a question. Dr. Gans?
Thank you so much. I just really do want to applaud that amazing review of the literature that is out there. So thank you for that. I did have one quick question, maybe just for broadly for the FDA. I was unaware of why the submission to the FDA was actually withdrawn, and I just wondered if we could have any additional information on that to think about what we're voting on today.
This is Gail Bormel. I mean, the agency doesn't generally comment publicly on litigation, so I'm not really able to talk much about that. But it is important that even though the nominations were withdrawn, we can elect to review the information and bring it to the PCAC. We have another question online from Dr. Corbett. Dr. Corbett?
Hi, Samantha Corbett. Along those lines, just checking if, since this was a withdrawn, the nominator withdrew this application, assuming if it were not allowed to be on the bulk list now, is it a possibility that any of this product could have a nomination in the future? Is there anything to preclude that from happening in the future?
This is Gail Bormel. I mean, we are bringing the bulk drug substances to the committee for their recommendation. And so the process after that would be to conduct rulemaking. So those bulk drug substances, that were addressed would either be proposed or not proposed for the rule. There's nothing that in the future would preclude, an entity, someone from, asking us to address it again. I'm not sure it would be a nomination. It might be either a nomination or another type of vehicle like a citizen petition.
Okay, thank you.
Dr. Gainey, did you have a question?
I did. Yes, Mark Ganny. My question is to Dr. Hankler. In the application, did they outline how they would expect to use this? What was the regimen, in particular, the dosing frequency and the duration? And would it be different for different diseases?
Elizabeth Hankla. No, unfortunately, we don't have that information. In the nominations that we get, we usually don't get dosing information, how long it'll be given, post dosage, that sort of information.
If I could just add to that, one of the things that came in the nomination that caught our attention was that they were proposing a strength that was twice what we've seen in any of the previous clinical studies and all the history on. So almost everything up there was 1.6 mg per mil, if I recall. What was proposed was a 3 mg per mil. That's as much as we know.
Dr. Galore online has a question. Dr. Galore?
Thank you. If I could just get a clarifying answer to this. Once a substance is placed on the bulks list, is there regulation? Is there any indication there or direction on what dose can be used, et cetera? Or is it just that it allows people to compound and the clinical regimens are left to the prescriber?
This is Gail Bormel. Once a bulk drug substance is placed on the 503A bulks list, it will be left up to the prescriber for the route of administration and the dosage.
Thank you.
Dr. Clark?
Yeah, I have two questions. One was on the Dr. Henkel's presentation, slide 63. The improvements in immune function, were those in vitro responses, do you know, or what led them to conclude that TA1 was helpful for immune function?
Elizabeth Hankla, can you bring up slide 63?
Sorry, this was Perugia, 2010.
Okay, apologies. Can you repeat your question again?
Yeah, it was just the author's conclusion was that it may favorably affect immune function, and there wasn't data in the background information about this either. I was just wondering what assays you used, or if you knew what they were referring to there, if it was just in vitro assessments or how they were measuring that.
Elizabeth Hankla. I believe they were in vitro, like T cell responses to specific viruses. I would have to look up specifically what it was.
Okay. Yeah. I have one other unrelated question. On the COAs that we've been talking about, for the drugs that have been made it to the bulk drug substance list, are those COAs significantly different than what we've seen today, like in terms of detail, a list of the impurities, or how do nominators know what to include there?
Excuse me, Russ Westlake, FDA. It will vary from drug substance to drug substance, right? I mean, for something like, I'm going to go back to my old example, for something like aspirin, it's extraordinarily well known. We're less concerned with certain aspects of it. For a large peptide where there's immunogenicity concerns and impurities can have such a dramatic impact, you're looking for more information on impurities in order to ensure patient safety. So it will vary from product to product.
And can the FDA require exactly what's put on those so that the nominators know what you're looking for?
Gail, I'm not sure. That's more of a legal question. I'm not sure.
If we can impose certain COA requirements, is that what you're asking?
Yeah, I guess, and even on the suppliers to know that what the pharmacies are getting is pure.
Yeah, I mean, I think that that what's in the statute now just generally talks about a valid certificate of analysis. We haven't further defined that. Is it possible that there could be in the future a definition of that? Yes. But again, yeah, I mean, again, part of the issue is going to be standardizing different elements, different substances for one standard. And so that may not be as clear as when there is, for example, we're not in this realm, but when you're talking about like a USP monograph, which has, it's a public standard. So I think, just to answer your question simply, it's possible that that there could be a further definition, guidance about what a valid COA is.
Thank you. That's it.
Just a friendly reminder to state your name before you ask the question. We actually have a queue. So the next person is actually Dr. Gans online, followed by Dr. Chris, followed by Thomas Lupton. But I'll call you on, Javi. Dr. Gans.
Thank you.
State your name.
Thank you. Haley Gans, Stanford. I had a general question again, and it goes along the lines of, I think, what we're slightly grappling with, because, I mean, with that amazing literature review, which we're now have to use that as opposed to some kind of nomination, is that it's been used in many different ways and doses and all that sort of stuff. So I guess my general question is, and I'm assuming the answer to this is no, what we decide here today really does not inhibit people further studying the use of TA1, because I think in certain populations, given in certain ways, typically it's been used later than we would have liked, that there actually may be some benefit, but that's a very specific study question. And I guess my question is, there's nothing that we do today to discourage people from continuing to look at ways in which it might be helpful to certain disease states. Like, for instance, primary immunodeficiency wasn't evaluated, but there are some suggestions that it might be helpful in people who are deficient in TA1.
So this is Gail Bormel, if I can respond. No, it doesn't. The decisions today are separate from whether a study could continue to occur. But, there are parameters for studies. And so they involved informed consent and a protocol, et cetera. And, we had talked a little bit about the IND process and expanded access, et cetera. What this process today is about whether to place Thymosin alpha-1 and this substances on the 503A bulk drug list. And what that means is, if it's placed on the list, then pharmacies can use that as a substance in compounding, pursuant to a valid patient-specific prescription. But there's no studying that would need to occur formally, and no other parameters on that.
Great, that's what I thought. Thanks.
Next, we have Dr. Chris.
Mark Chris. Are there any INDs in existence now for this?
This is Gail Bormel. I don't believe we can comment on that right now.
Okay.
Next, Dr. Lupton.
Thomas Lupton, a quick comment regarding the COA process. We have the number of COAs in the packets, and I appreciate that, but those COAs reflect the supplier's QC testing of their product. That does not mean that that item is sufficient for the compounding formulation that pharmacy is completing. So it's up to that pharmacy to demonstrate that COA meets their internal requirements based off their formulation and data.
Question online from Dr. Corbett. Dr. Corbett?
One of my questions was answered about the IND can't be addressed, it sounds like, but in addition to that, are you able to answer if an NDA was ever submitted as a pharmaceutical agent to the FDA at any time? And I assume not approved, but I'm just curious if that had been done, since it's approved in at least in other countries, if it was ever presented to the FDA as a pharmaceutical agent.
Well, diversity FD. We may have had several communications with different proposals for TA1 in development, but we are not aware of any NDA that was submitted and approved or not approved. We don't have approval for TA1 for any condition.
Okay, thank you.
Next, we have Dr. Gura.
Sorry if I could just add to that.
Oh, sure, go ahead.
And Russ Westlake, FDA, the only thing I would add, because this is definitely the land of DIVA, but Cyclone's press releases have made clear, so this is public information, that they were initiating a phase 3 trial in the US. There's been no communication since from Cyclone.
Kathy Gura, Boston Children's. If we decide as a group that this does not make the list, would clinicians still have access to this product under an expanded access, or could they import, using Zadaxin, for example, now that I've been educated that it's not available in the US, but they can import it using expanded access or, EIND route or something like that. So patients who need it or might benefit, as a last ditch effort, they'd still have access to it.
Yes, diversity FDA. Yes, this is always possible availability through expanded access or the IND route.
Next, Dr. Powell.
Kind of similar to your question, Dr. Gura. This is Alice Powell from NIH. It's my ignorance about, early on in one of the presentation was that you can get it online in injectable or nasal spray. My question is, so whether the online products are from another country, therefore, one can get it online, or whether those are companies that are making them in the US without having any kind of approval, bulk compounding, or any other approval. This is my ignorance. I just didn't know how that happens.
This is Gail Bormel. I mean, there's with the internet, there are always products that are available that are, sometimes counterfeit, sometimes illegal. A lot of different products that would fall into that category. Sometimes they're from foreign manufacturers, manufacturers abroad or foreign entities. So, there's a lot of things on the internet that may not be legal. I mean, so that's sort of a separate aside from what we're talking about now, which is, the legitimate practice of pharmacy compounding, which falls within, Section 503A. And so to the extent that pharmacies are compounding in compliance with the applicable federal provision as well as their state laws, that would be, legal. But what people sell, I mean, there's a lot of things online that are available through, websites that may very well be not legitimate. So,
Dr. Janet Lee online.
Oh, thank you. Washington University in St. Louis. I just had a question in reference to one of the statements that was previously made that one aspect of one aspect was the concentration that was proposed for the injectable product is greater than what had been shown or in the literature review. So am I to understand that none of the studies, it was a very comprehensive review, so thank you very much, the clinical studies had ever used that concentration sub-Q? Thank you.
Hi, Elizabeth Hankla. In terms of the clinical studies, the highest concentration we saw used was 2 mg per ml.
Thank you.
Are there any other questions, either from our in-person group or online? Oh, Brian Lee. Brian Lee.
Hi, Brian Lee from University of Southern California. And just to clarify then, so placing a drug on the 503 list, that makes it legal for pharmacies to compound that drug. If a drug is not on that list, does that make it illegal for pharmacies to compound that drug with enforcement?
Hi, this is Gail Bormel. So if a bulk drug substance is placed on the 503A bulks list, that is one of the conditions to in the act that allows you to be exempt from certain provisions, like getting a new drug application or having adequate directions for use, or in the case of compounders, pharmacy compounders, from complying with current good manufacturing practices. So it makes it a essentially, yes, a legal practice as we define it in, if all the other parameters of that provision are met. If the bulk drug substance is not placed on the 503A bulks list, it means, and a pharmacy would compound it with it, it means they would no longer be exempt from certain provisions of the Act. Like they'd have to get a new drug application, they'd have to have adequate directions for use, and they would have to comply with CGMPs. So it's a little bit of a twist on what you said. And remember, when we place something, after the committee makes its recommendation, we go through a rulemaking process. And so there's a proposed rule for which the public can provide comments before there is a final rule to actually finalize the list. So I just wanted to point that out as well.
Thank you.
Are there any other comments or questions from the group? Yes.
Hi, Elizabeth Ankla. I just wanted to loop back Dr. Clark. I looked at that article. They measured T cell counts and functional pathogen-specific T cells responses by a limiting dilution assay. Thanks.
Thank you for that additional information. Any other questions or comments? Online, any questions or comments? I want to thank everyone for their questions, comments, and FDA for their responses. I will now take a 10-minute break. So we'll reconvene at 2:32 to continue the thymosin alpha-1 related bulk drug substance topic. Panel members, please remember that there should be no discussion of the meeting topic during the break amongst yourselves or any member of the audience. And we'll see you all at 2:32. Thank you. I just make one clarifying comment before we? Absolutely, yes.
I think we are we live? I'm sorry. We're not quite live yet.
Okay.
No, it's okay.
Okay. Before we resume, FDA has a comment. Go ahead. Thank you. This is Gail Bormel. So I was just taking a look at the final rules and the proposed rules for the 503A bulk substances. And I have noticed that for many of the bulk drug substances that were, well, some that were finalized and many that were proposed, that we have limited it for route of administration. And in some cases, there's like percentages that have also shown up as proposed limits. And so if that's in part of what was evaluated at the time by the agency and then brought to the PCAC, they made it into the proposal. So I didn't want to leave with the impression that it couldn't be limited at all, the bulk drug substance, because many of them in the proposed rule and in the final rule are at least limited by route of administration. Thank you. Thank you for providing that information.
And well, two of them that I see are in the proposed rulemaking are also limited in concentration.
Great, thank you. Any other? Okay. So we will now begin the open public hearing session. Both the Food and Drug Administration and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the open public hearing session of the advisory committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement to advise the committee of any financial relation that you may have with the product, and if known, its direct competitors. For example, this financial information may include the payment by a bulk drug supplier or compounding pharmacy of your travel, lodging, or other expenses in connection with your attendance at the meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have such financial relationships. If you choose not to address the issue of this financial relationship at the beginning of your statement, it will not preclude you from speaking. The FDA and this committee place great importance in the open public hearing process. The insights and comments provided can help the agency and this committee in their consideration of the issues before them. That said, in many instances, for many topics, there'll be a variety of opinions. One of our goals for today is for this open public hearing is to be conducted in a fair and open way where every participant is listened to carefully and treated with dignity, courtesy, and respect. For those presenting virtually, please remember to unmute yourself and turn on your camera when your OPH number is called. For those presenting in person, please step up to the podium when your OPH number is called. As a reminder, please speak only when recognized by the chairperson. Thank you. So we'll begin with speaker number 1. Please state your name and any organization that you represent. For the record, you will have 3 minutes.
Good afternoon. My name is Dan Denai. I am the Chief Executive Officer of Avexius Health Solutions, which is a national network of over 10,000 medical providers, all representing many different disciplines of medicine. To clarify to our panel, if TA1 is not on the list, patients and practitioners will lose access, and that's what leads to patients seeking out online fake pharmacies. The real-world results of the FDA's recent decision around TA1 is that thousands of these patients have turned to the black market to obtain peptides. The black market includes fake online, completely unregulated pharmacies, as well as other pharmacies advertising that they are a research facility, many of whom provide little or no direction on how a patient should be using the compound. Strangely enough, the agency became extremely active in policing thymosin alpha-1 compounding when a group of physicians reported high rates of success in addressing COVID-19. And this was far earlier than any of the studies that were cited earlier in the presentation. The agency even put out a special notification that expressly prohibited compounders from making this extremely beneficial peptide, demonstrating again and again, their willingness of the agency to overreach and interfere with a medical provider's freedom to practice medicine, which is truly a travesty. There seems to be a massive disconnect between medical practitioners and the FDA. To quote our soon-to-be HHS Commissioner, RFK Jr., the FDA's war on public health is about to end. This includes its aggressive suppression of psychedelics, peptides, stem cells, raw milk, hyperbaric therapies, chelating compounds, ivermectin, hydroxychloroquine, vitamins, clean food, sunshine, exercise, nutraceuticals, and anything else that advances human health that can't be patented by Big Pharma. I represent thousands of medical providers who are burned out on writing band-aided prescription after band-aided prescription for patients only to see their health and well-being continue to decline. Our practitioners want to help their patients. Our citizens long to live happier and healthier lives, to create a world that is positive and is uplifting to the human spirit. Ladies and gentlemen, we can do and must do better because it is our moral and ethical duty to do so. Anything less is unacceptable. Thank you for your time.
Thank you. Speaker number 2, please state your name and any organization that you're representing for the record. You have 3 minutes.
Yes, Jim Lavelle, International Peptide Society and American Academy of Anti-Aging Medicine. I'm not even going to go off of these slides. I have to address a few things here. I taught 18 years at the College of Pharmacy and Medicine, University of Cincinnati, currently adjunct professor at the George Washington School of Medicine and Health Sciences, Department of Integrated Medicine. And we teach at the International Peptide Society, we're trying to do the right thing. We're trying to help providers help people feel that they have a chance for a healthier life when they are in chronic illnesses and conditions. And there's a difference, and I have to say, the review of your information on Thymosin alpha-1 was incredible and thorough. And I can tell you that the majority of doctors that are in the kind of the integrative medicine or the what you want to call wellness space are not using it to treat hepatitis A or not using it to treat hepatitis B. What they're looking for is how can we help people not move down the path of metabolic inflammation, or as you well know from the literature since 2008, metaflammation leads to inflammaging. And the key process in that is the disruption of the immune system, the loss of homeostasis of the immune system, the loss of immunomodulation of the immune system. And where TA1 offers a potential benefit, and where at least 300,000 prescriptions have gone out and physicians have responsibly wanted to write prescriptions to pharmacies that want to comply, that can be inspected, that they can then see a result with their patient. That is what's being taken away. So I'm not in disagreement with what the assessment was for the more acute condition, but where peptides provide value, what we have seen with our 1,000 physicians in IPS, our 15,000 physicians at the American Academy of Anti-Aging Medicine, is they're using these type of compounds in order to be supportive as a part of a structured program that helps people to reduce that risk of moving towards acute conditions. And I could give you a bunch of slides of information. We know it's immunomodulatory. It doesn't seem like there's immunogenicity. At least it wasn't shown to be that way. Just as I would say a quasi-academic, I can't claim full-time as an academic, but I've directed a fair amount of my life towards it, as well as writing databases for Lexicomp. It was mentioned earlier. We have a chasm between acute care, disease care, and how we're going to keep people well. And that's where these compounds, I think, sit squarely in that gray area. And hopefully we're going to provide guidance from the FDA on what are acceptable excipients, how do we protect a compound, as well as be able to collect real-world evidence through doctors that are using EHRs so that we can provide that information to all of you so that we can make great decisions forward that help people to improve on their quality of life. Thank you.
Thank you. Speaker number 3, please be sure to state your name and any organization you're representing for the record. You have 3 minutes.
My name is Tom Wynn. I'm with Pharmaceo Pharmacy. Next slide. Next slide. Next slide. Next slide. We'll stop there. So one thing I wanted to comment on is somebody asked before about the C of A and what are the requirements that are on there. There is a USP guidance 1503, I believe it is, that's for synthetic peptides, and it does go through specifically what test should be done in that case. The FDA brought this up before. It is there and available so that there is something to move that along. The other thing I want to mention too is, again, we brought up the immunogenicity several times today. At every time it's been said, it can, it has potential, it might, maybe. There's never been one time we said it will cause immunogenicity. So we're kind of not really getting to the point of the immunogenicity. We're just saying it could, it's a possibility. Now, I will say one of the products that's currently available, commercially available, semaglutide, has not been brought up at any point. That's not what we're talking about today, but there is a preservative they're using called phenol in there. Phenol has been shown for sure to cause protein aggregation, and it's actually in the product that's commercially available. Now, as a compounder, we would move away from that. We'd probably use a different one, maybe benzoyl alcohol or something else. We would purposely stay away from phenol because, and I know it's in some vaccines, it's in a lot of different products, but in this case, how do we know that that is not causing some immunogenicity, some aggregation, if you will, from all the different kind of side effects we're having lately on semaglutide, whether it be the GI stuff or they're even getting different kind of lethargy and different things of that nature that you're hearing about if you Google it online. So, I mean, what I'm getting at is, you know, how serious are we about immunogenicity if we're putting something into products that are commercially available that will do that? And if you look here, I'm kind of going through again, uncontrolled manufacturing process, something they brought up. At a pharmacy, we do follow USP guidelines for preparations. And as far as USP goes, they have new revisions, 797 for sterile. The FDA was a part of that. They did have somebody there during the committee meetings, maybe not all of them, but I know there was an advisor there that was able to help put in place some of the particular aspects of that particular chapter. And I'm not against the chapter. It's fantastic. And we all want to, you know, abide by that chapter. But to say that we have uncontrolled processes isn't true, because you were there at the point when when that chapter was written, and proposed for us to actually use. Weren't a voting member. There's probably just an advisor, but still you had some play in what was going on there. Pharmacies do have controls in place as far as temperature controls. We're at
time.
Thank you.
Thank you. Speaker number 4, please state your name and any organization that you're representing for the record.
Can I have slide deck number 6, please? Can you stop the time?
Sure.
All right. My name is Dr. Edwin Lee. Thank you for letting me be here. I'm a board certified endocrinologist. And can you go to slide, not the next one, but the one after that, please? So 50 years ago, in 1974, the FDA approved a IND for thymosin fraction 5, which is a precursor of thymosin alpha 1, to be used in children with primary immune deficiency. Most of the kids had a dysfunctional thymus gland. They were very sick. The very first child was Heather, 5 years old, and she was critically ill, slowly dying, multiple infections, and thymosin fraction 5 was life-saving. She was titrated off it and was doing very well. Next slide. Unfortunately, she died in her early 20s from lymphoma, and she developed cancer from a weak immune system. Dr. Amen, involved in the thymosin fraction trial, told Dr. Alan Goldstein, who discovered thymosin alpha 1, his biggest regret was stopping the medicine. Next slide. So fast forward in 2024, Dr. Elliot Dentz and myself published an article titled Comprehensive Review of the Safety and Efficacy of Thymosin alpha 1 in Human Clinical Trials. We've tabulated over 11,000 subjects worldwide that received an average dose of thymosin alpha 1, 1.6 milligrams sub-Q 3 times a week. It was absolutely safe, and most of the studies showed positive results, not 100%. Next slide. A colleague of mine, Dr. Luis Martinez, did a study in his office to test if thymosin alpha-1 can improve the immune function in 10 of his patients, and you can see their age and their conditions there. He used the biomarker CD4-CD8 ratio. A ratio under 1 indicates a weak immune system. A ratio over 1 indicates a normal immune system. Using thymosin alpha-1, 1.6 milligrams sub-Q 3 times a week in 6 months showed 100% improvement of the ratio. Next slide. I have many patients that have used thymosin alpha-1 over 1 year. This is one of my patients. She has stage 4 colon cancer in 2021 before treatment of surgery, radiation, chemo. She started thymosin alpha-1, 1.6 milligrams sub-Q 3 times a week. And fortunately, she has had no complications of neuropathy or even lost her hair. There is a study published showing that thymosin alpha-1 with chemotherapy reduces neuropathy. Next slide. In conclusion, thymosin alpha-1 has been approved in 37 countries. It's naturally made, decreases as we age. It is safe, efficacious, it's life-saving. Thymosin alpha-1 should be used in adjuvant therapy with chemo or cancer therapy, and the American public deserves thymosin alpha-1. Please save it. Thank you.
Thank you. Speaker number 5, please come to the podium. State your name and any organization that you're representing for the record. You have 3 minutes.
Can I have the slides, please? My name is Dr. Terry Denai. I am an acute care nurse practitioner and research clinician. Next slide. I'm going to talk about some real-world data that was collected recently from our internal medicine clinics. This was looking at 38 patients. I'm going to go through this quickly because you can look at it, the demographics of the patients, the age ranges, et cetera, ethnic breakdown. Next slide. These are the primary diagnosis codes used, and these are the primary diagnosis codes in hundreds of my colleagues that have been through advanced peptide training, use it primarily for an autoimmune disorder in the early stages. Somebody mentioned earlier, a lot of these other studies in late stage disease. Early stage is what we're finding where it has the most benefit. Next slide. Concentration that were used in these 38 patients were 3 mg per mil, initially BID, so it's 300 micrograms twice a day, and then daily thereafter. Next slide. No side effects or adverse events were noted. This is just a slide from the VA distinguishing between a side effect and adverse event. They are 2 completely different things. Next slide. And again, none were reported. Next slide. And then I'm going to present a quick case study. This is a very common presentation of patients that we see. 67-year-old healthy female. She came to our office diagnosed with autoimmune hepatitis. When she was seen by her PCP, she had LFTs way up there, 1,400, 970. Couldn't find anything wrong with her. No cancer. Everything was negative. All her serology was negative. After 3 months on oral prednisone, she showed some improvement. They wanted to put her on methotrexate. She researched it. She refused. She wanted other options. Next slide. She came to us still with elevated liver enzymes. We started her on TA1 BID for 12 weeks with nutritional support, tapered off her oral prednisone, and then her over the course of 6, 12, and 24 weeks, she had normalized on her LFTs. No side effects, no adverse events for 3 years. She was on it. She was very afraid to go off and very devastated a year ago when it was taken off. And last I saw her a year ago, she was seeking off-market options, unfortunately. And this is a common clinical picture of patients that have seen massive benefit with thymosin alpha-1 to their immune status and their chronic condition that had no other treatment or other treatments had failed, and now this has been taken away from them. Thank you.
Thank you. Speaker number 6, please state your name and any organization that you're representing for the record.
Sure. Could we bring up slide deck 1, please, for Lee Rosebush? My name is Lee Rosebush. I'm a PharmD doctor of pharmacy. I'm also an attorney. I am, in this case, also here to represent some of the pharmacies associated with this, but this may be even news to some of the FDA folks, is that 20 years ago, I actually spent time in the office of Orphan Products. I was there underneath Marlene Hafner, Diane Centeno, DeShields, and Jeff Fritsch. And in fact, they actually wrote my letters of recommendation to get into law school. I say that because I am here not just as a pharmacist and an attorney, I'm also here as an OOPD patient advocate. And we're going to go through that in just a second, what that actually means. More importantly, I have a daughter that many of you know from this perspective, who has a rare disease and is life-threatening at Children's Hospital on a weekly basis. So this really does mean something. To answer some of the questions that were asked earlier, if you put this on the list, it will be taken away. It is illegal to compound from that perspective. Some of you asked about INDs. Many pharmacy compounders are the ones who supply those products. They will be taken away. And if you encourage expanded access of the foreign-based product, that means FDA is actually on record encouraging the importation of a non-FDA approved product from a non-FDA registered site over that product that could be made domestically and inspected today. And instead, it's going to be made in a facility that FDA would have no jurisdiction over. That's important to remember. Next slide, please. There's 4 specific criteria we've gone through multiple times. Is it well characterized? Obviously, this one is approved in 36 countries, including Italy. It is approved in the European from that perspective. Has it been used historically in compounding? If we can go to the next slide, you'll see we actually have, next slide. We actually have over 300,000 prescriptions that have been dispensed by this. There's a huge historical record associated with this. Are there safety concerns associated in effectiveness use? I'm going to read directly from FDA's own slide that was just given to you. On the safety side, in most clinical studies, TA1 has not been associated with significant adverse events attributable to TA1 when administered in doses of ranges of 1 to 16 milligrams. That's from FDA. On the efficacy perspective, this is just one indication, and we'll go through others. Study authors suggest that TA1 may favorably affect immune functions, period. That's all 4 criteria. I'm not sure why we're here from that perspective. If this was FDA approved, as was asked about Zydexin, we wouldn't be here from that side of it because we have the ability to compound. If this had a USP monograph, which we have approached USP to make a monograph, we wouldn't be here from that perspective. Unfortunately, USP will not allow us to make a monograph. On the orphan side, and the reason why I raise that, for those that don't know, in this situation, there is a specific regulation, 21 CFR 31620. And in it, it says, quote, a discussion of the scientific rationale, this is what's included in order to get orphan drug status, quote, a discussion of the scientific rationale to establish a medically plausible basis for the use of the drug for the rare disease or condition, including all relevant data from in vitro laboratory studies, preclinical efficacy studies conducted in animal mode for the human disease or condition, and clinical experience with the drug and rare disease or condition that's available. FDA has granted orphan status using that standard safety and efficacy 4 times in the last 15 years to this substance. As somebody who has a kid, a child who's potentially dying from a rare disease, if you say no to this and get put on a list, it will be removed, regardless of what is said. Practically from that perspective, if this says no, ask yourself, what wholesaler would ever carry this product? They won't. So in this case, you'd be asking for an expanded use product from a non-FDA approved source, from a non-FDA inspected facility, and you're taking away something that could be domestically made here that saves lives for kids that have rare diseases. And you know why it's not approved? Because there's no money to it. Don't let this get caught up in politics with COVID-19 and peptides that you just heard about from Lilly. That's why the other ones were there. Don't let this one get taken away because of politics, please.
Thank you. The open public hearing portion of this meeting is now concluded, and we will no longer take comments from the audience. We will now take additional questions and comments from those here. I want to open that up at this point. Are there any clarifying questions?
Yes, Russ Westake, OPQ, FDA. Dr. Rosebush and many of the speakers raised some really good points. One of the challenges for us in the FDA, and especially from the standpoint of characterizing a bulk drug substance, is that, going back to my presentation first thing this morning, there are dramatic differences in quality from bulk drug supplier to bulk drug supplier. So you'll see a lot of clinical trials without significant safety signals potentially, done with material that was manufactured by, in essence, branded companies under tight controls. For any approved product, there's an approved synthetic pathway. There's an approved set of methods and specifications for the drug substance, and an approved set of methods and specifications for the drug product. That's not the case here. Anybody can make it to any standard, and it can be imported and compounded. And that's part of what we're asked to do in OPQ, is take a look at what's out there, look at the C of A's and see, is it similar to what's been tested and shown to be safe and effective, if it was, or not? And that's our challenge. We don't have information to make that assessment. I talked this morning about 3 potential pathways, synthetic pathways for large peptides. You can go through a biochemistry standpoint. You can go from a chemical synthesis, or you can isolate it from natural sources. This can be made all three ways. It will have dramatically different impurity profiles. And out of that, you have dramatic differences in terms of immunogenicity potential. To the point, it's not to say it will happen, but certainly the potential is there. And that's why we come to the conclusion from an OPQ perspective that it's not well characterized. Thank you.
Thank you. Any other additional comments or questions? Yes. Please state your name.
Got it.
And affiliation.
Oh, and affiliation. Oh. Robin Bogner, University of Connecticut. I have similar concerns about some of the certificates of analysis that I've seen. They may be called valid, but I don't trust a lot of what's there. With this particular compound, drug substance, could a pharmacist in the US source it as the manufactured product from overseas and compound with the manufactured product that has presumably gone through not FDA, but some other agencies, I guess, regulatory approval and oversight? And that's my question, because if that's the case, I might look at it differently.
I think both, sorry, Russ Westlake, FDA, I think both Gail and I will likely comment. Could they? Yes, they could. Are they, but the important question to me is, are they constrained to do that? And the answer is no, they can buy it from anybody, anywhere. And I want to be clear, my criticism, there's an implied criticism here, is not so much aimed at the compound industry. It's a bulk drug substance industry that can make and do whatever they want. Some of the C of A's, it's kind of shocking. If you really dug into the materials you have on multiple substances here, this isn't a compound. This is a bulk drug substance manufacturer. And there's a C of A that has a name on it. And then you look at the tests and you're like, wait a minute, they didn't make that from these tests. You know they made something else, in fact. And then you look at the molecular formula, the molecular weight and chemical name. Wait a minute, it's a third thing entirely. And it's hard for us because I look at it and I'm like, do they even know what they're making? Do they even know what they're testing? That's not a compounder's problem. That's a bulk drug substance manufacturer's problem. But that's what we're seeing when we look at some of these C of A's. So it becomes challenging to say, hey, this is well characterized. Could it be? Yeah. Is it?
This is Gail Bormel. I think that the challenge is also that it's sort of that there could be a number of different manufacturers of an API, and if they register with the FDA and they list their product, they can they can ship it in into the U.S. for appropriate uses like compounding, research, et cetera. And I think what Russ is saying is that, it doesn't mean that every BDS that's the supplier that's registered and has listed a product, that every BDS is necessarily identical in its, quality. And so, again, it's a little different scenario than when we have a substance that's in the USP and, has a public monograph, and that's what is required to be adhered to. So I think that's the difficulty. There could be multiple manufacturers of the same, BDS, but it may not really be the same quality.
Yes, go ahead, Dr. Burke.
I can respond.
Yes.
I think my point is, because I agree with you, for many of the other peptides, I wouldn't know where to go looking, right, for a good bulk drug substance. Yes, you can get bulk drug substance from anywhere, but in this case, I know there's a product out there that has gone through somebody's regulatory review that I would feel more comfortable with. So while a compounder could go anywhere, maybe I, as a more careful compounder, would go to a very specific place that I know exists, right? A marketed product elsewhere, to be sure, but whereas with other peptides, it's not clear where I would go. That's my point, I think, the differentiation here.
So we have Dr. Gupta first, online, and then Jeanette Stays. Dr. Gupta?
Yes, hi, thank you. I had a question and perhaps a comment, on this. My concern is, I see a lot of patients that often come in on a lot of different supplements, some of them including these. And much of the concern I have is, where does the consumer go? We're talking about the compounder, we're talking about the individuals that ultimately have side effects that may perhaps be mild to severe. And no one has addressed the consumer and the patient. When they're having a side effect and I'm the physician managing it, or any physician in the United States, what are we supposed to do? And that is ultimately the question that seems to be going unanswered here. And it's frustrating because many of these individuals have no recourse. There is no solution, and there's no response from the companies that are developing these products. And no one really has an answer. So I would love to hear an answer today from somebody in the room on what we're supposed to do.
Hi, this is Gail Bormel. So I think the question, and correct me if I'm wrong, is about where to report certain adverse events. Is that the question? My understanding was, as a physician, how do you help the consumer? Is that correctly addressing your question, Dr. Gupta?
Yeah. I think that's basically it. There are so many side effects that we're seeing, to these products. And many times patients will come in with bottles of different, because they don't want to take traditional medications. They want to try these substances. So I think the question I have again is that, where do consumers or patients go for addressing these symptoms or these conditions? Usually the compounder doesn't, because they're not the prescriber. The prescriber doesn't know, which would often be, someone that's either doesn't know how to handle it, and the manufacturer may be foreign. So what are we supposed to do in that situation? And the evidence is lacking from what I'm understanding. So I'm trying to comprehend here today what we're supposed to do in those situations. If anyone can answer that, I would love to hear the answer.
Again, this is Gail Bormel. And so, let's just go through the process that we know about, which is for, drugs in general that either approved or compounded drugs that are not, for compounded drugs that are not made by outsourcing facilities. If there are, complaints about them or certain concerns, usually what happens is that there's a recourse to either, nothing's mandatory in our law, but, there's a recourse to, submit complaints to the State Board of Pharmacy. You can voluntarily report information to FDA through its MedWatch program. So it's a voluntary reporting thing of these, drugs that are, not FDA approved, like, either compounded drugs or other sort of drugs they may be obtaining from the internet. It is an issue that when people purchase drugs, and I'm not really sure if you're alluding to people buying things from the internet or they're buying things from just other sources, that they may be mail ordering things. That sort of, we still do get complaints through different systems in the FDA, even if they're not FDA approved drugs.
I mean, that's just a mechanism of reporting. But, you know, I'm not sure if you're trying to sort of figure out a way just to advise, you know, patients about practices, or, you know, you're trying to get information out there. I mean, the reason we have IND and expanded access programs is so that we can, you know, it's a way of formally getting drugs to patients with informed consent and sort of looking at the data in a controlled way. And it seems what you're talking about are sort of patients that are purchasing things on the internet, not even, you know, being sure that they're legitimate.
I believe Dr. Danette Stace had the next question.
Hi, Dr. Danette Stace from Jazz Pharmaceuticals. I actually just wanted to follow up on Dr. Bogner's question about sourcing outside of the U.S. Really just directing this to Dr. Westdyke and Dr. Bormel is, I guess the consideration is that if you were to go and get Zadaxin from outside the U.S., it would already be formulated as a drug product. So it wouldn't be the drug substance. So if you were to bring that in, I don't know, and that's a question I have, I'm not sure how you would then compound that. You would have to extract the drug substance from that and then reformulate it in the way that you would like. So perhaps maybe the thinking then would you'd be more targeting the drug substance supplier that you know, that company used to make the product. You would have to go to that source. If you were looking for something that was, you know, of high quality, if you will. Otherwise, I don't think it really helps you to go after the actual marketed product, but I'm happy to hear what the agency has to say about that. Thank you.
Russ Westdyke, FDA. Absolutely agree with everything you just said. With respect to my colleague, yeah, I don't think a compounder is likely to import a commercial drug product to compound. It would be much easier to go buy the bulk drug substance from somebody. And the whole reason you're compounding it is, in theory, you want to put something in it. You're concerned with an allergen in it. Otherwise, you would just frankly dose that product.
Yes, Dr. Bogner.
Actually, a lot of compounding is done from a marketed product and then compounded to a different dosage form. I work mostly in the large molecule biologics, and bulk drug substance is partially at least formulated so that it can be stored. So I think that you can compound from a drug product.
Russ Westdyke, FDA. Absolutely agree with you. Certainly. But in this case, we're talking injectable to injectable, right? The product on the market is an injectable. What they're proposing to make is an injectable. Why would you recompound it? It just seems like it would be going through a lot of extra steps and trouble. Not saying you couldn't do it. It just logically, I as a formulator, I'm where you're coming from. Like, why would I do that? But it could be done. Absolutely.
This is Gail Bormel. I sort of read it the way that Dr. Stas was reading it, that you would want to import the API from the reliable API manufacturer that was providing the drug to the the product that was approved in a foreign country. Once you start importing drugs that are approved in another country but not approved in the United States, there are other laws that take effect. So you can't necessarily bring in drugs that are approved in another country and bring it over, you know, to use in this country necessarily. So I just wanted to caution if you think that we could just, you know, allow that importation of the finished product that's approved in another country.
Any other comments? Questions? Online? Okay. The committee now will turn its attention to address the task at hand, the careful consideration of the data before the committee, as well as the public comments. We will now proceed with the questions to the committee and panel discussions. I'd like to remind the public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel. After I read each question, we will pause for any questions or comments concerning its wording. Slide 2, please. We will proceed with our third question, which is a voting question with subsections. We'll be using electronic voting system for this meeting. Once we begin the vote, the buttons will start flashing and will continue to flash even after you've entered your vote. Please press the button firmly that corresponds to your vote. If you're unsure of your vote or wish to change your vote, you may press the corresponding button until the vote is closed. After everyone's completed their vote, the vote will be locked in. The vote will then be displayed on the screen. The DFO will read the vote from the screen into the record. Next, we'll go around the room, and each individual who voted will state their name and vote into the record. You can also state the reason why you voted as you did if you want to. We'll continue the same manner until all questions have been answered or discussed. For question 3, Section 503A bulk drug substances list, Thymosin alpha-1 related bulk drug substances. FDA's evaluation addressed 2 Thymosin alpha-1 related bulk drug substances, which include 1 active moiety, Thymosin alpha-1 free base, and 2 different BDSs. FDA proposes using a single voting question to address them as a group. Do committee members agree to vote on Thymosin alpha-1 related bulk drug substances discussed today, Thymosin alpha-1 free base, and Thymosin alpha-1 acetate as a group? Yes or no? If any member of the committee votes no, FDA will take separate votes on each of these substances. If voting yes, committee members will vote on the substance as a group and will proceed to answer 1 additional voting question. If voting no, committee members will vote on each of the substances separately and will proceed to answer 2 voting questions. Are there any issues or questions from the panel about the wording of the voting question? If there are no further questions or comments concerning the wording of the question, we will now begin the voting process. Please press the button on your microphone that corresponds to your vote. You'll have approximately 20 seconds to vote. Please press the button firmly. After you've made your selection, the light may continue to flash. If you are unsure of your vote or you wish to change your vote, please press the corresponding button again before the vote is closed.
Takia Stevenson, DFO. For the record, there are 20 yeses, 1 no, and 0 abstentions. I'll hand it back to the chair.
Since everyone voted yes, we'll proceed with.
We got 1 no.
Oh, sorry.
That's good. We're going to do 3B.
Okay. Since 1 or more panel members voted no, we will proceed with questions 3B and 3C.
Oh, but can you go around the table and ask them?
Sure. Okay. Now the vote is complete. We'll go around the table and have everyone who voted state their name, vote, and if you want to, you can state the reason why you voted as you did into the record.
Todd Durham, I voted yes.
Ellen Veda, I voted yes.
Robin Bogner, I voted no.
Brian Saramaga, I voted yes.
Elizabeth Rebello, I voted yes.
Kathy Gura voted yes.
Linda McElhaney, I voted yes.
Tim Fensky, I voted yes.
Alice Powell, I voted yes.
Mark Ganny, I voted yes.
Mark Sasha, I voted yes.
Cecilia Monge, yes.
George Cyber, yes, because nothing in the presentation or discussion considered either of these differently from the other. Thanks.
Nina Clark, I voted yes.
Mark Chris, I voted yes.
Dr. Galur?
Padma Galur, I voted yes.
Dr. Corbett?
Dr. Corbett, I voted yes.
Dr. Gans?
I voted yes.
Dr. Gupta?
I voted yes.
Dr. Brian Lee?
Brian Lee, I voted yes.
Dr. Janet Lee?
Janet Lee, I voted yes.
Right, thank you. Since one or more panel members voted no, we will proceed with questions 3B and 3C. Slide 4, please. Question 3B, Section 503A, Bulk Drug Substances List, Thymosin alpha-1 related bulk drug substances. FDA is proposing that Thymosin alpha-1 free base not be included on the 503A bulks list. The question asks, should Thymosin alpha-1 free base be placed on the list? If voting yes, you're recommending FDA should place Thymosin alpha-1 free base on the 503A bulks list. If voting no, you're recommending that FDA should not place Thymosin alpha-1 free base on the 503A bulks list. Are there any issues or questions from the panel? Oh, sorry. If the substance is not on the list when the final rule is promulgated, compounders may not use the drug for compounding under Section 503A unless it becomes the subject of an applicable USP or national formulary monograph or a component of an FDA-approved drug. Are there any issues or questions from the panel about the wording of the voting question? Actually, Dr. Corbett has a question. Dr. Corbett?
I think it's just a clarification from the previous conversations. So just confirming this is a yes, it should be placed on the list, or no. And then let's just hypothetically say it's a yes. We are not voting towards the specificity in the code as far as indication, dosing, any of that. Is that true?
This is Gail Bormel. That's true. If you vote yes, you would be recommending placing Thymosin alpha-1 free base on the 503A bulks list, and there's no parameters of route of administration or dosage.
Thank you.
Are there any other questions from the panel about the wording of the voting question? If there are no further questions or comments concerning the wording of the question, we will now begin the voting process. Please press the button on your microphone that corresponds to your vote. Reminder that you'll have approximately 20 seconds to vote. Press the button firmly. After you make the selection, the light may continue to flash.
Takia Stevenson, DFO. For the record, there are 4 yeses, 17 noes, and 0 abstentions. Thank you.
Now that the vote is complete, we'll go around the table and have everyone who voted state their name, vote, and if you want to, you can state the reason why you voted as you did into the record.
Todd Durham, I voted no.
Alan Veda, I voted no.
Robin Bogner, I voted yes. I struggle all the time with access versus quality. And in this case, I fell toward access.
Brian Serumaga, I voted no.
Elizabeth Rebello, I voted no.
Kathy Guerra, I voted no.
Linda McElhaney, I voted yes.
Tim Fenske, I voted yes.
Alice Powell, I voted no.
Mark Ganny, I voted no.
Mark Sasha, I voted no.
Cecilia Monge, I voted no because of lack of evidence of efficacy.
George Cyber, I voted no for the same reason, lack of evidence of efficacy.
Nina Clark, I voted no, also due to the low quality efficacy data that I thought was well summarized by the FDA.
Mark Chris, I voted no because curative therapies are now available for some of these conditions, and I would definitely want those curative therapies to be offered first.
Dr. Galore?
Padma Galore, I voted no for reasons already stated.
Dr. Corbett?
I voted yes, and I too very much struggle with this decision, but the reason for voting yes was it was an excellent summary on the efficacy of the conditions that were presented. I would not necessarily support them in any of the clinical trials and data that was submitted, but being in the integrative health dietary supplement world, I do kind of struggle with availability, and I think similar to what Dr. Bogner listed around availability and purity, and just decide to trust my colleagues that are prescribers and compounding pharmacists to do the right thing. Realize I would probably be in the minority, but I appreciate this was a really great summary and presentation by the FDA. Thank you.
Dr. Gans?
Hi, I voted no, but that is not to say that I didn't really appreciate the presentations for those who are in support of this. I didn't see much of a way in which this vote for compounding would actually change since it's already in use. And I think that if there's all those prescriptions already being used, that should be studied in those cases. And given that that's not out in the scientific world, we really need to know more about this. Case reports are really important, but it looked like when there were larger studies, the efficacy of these, albeit in the ones that were studied, and I agree that chronic disease and primary immunodeficiency still need to potentially have access to this in other forms, but I want to make sure that people understand its value before just prescribing it, which it seems is being done at the moment. And so I really support the scientific investigation into important peptides such as TA1.
For those of you that I'm calling out, please state your full name and then your vote. Dr. Gupta.
Thank you. I voted no. I struggled with this answer. I really deeply am for patients and for access. But I do really believe that the rigor is necessary for patients. They deserve the due diligence for these products to ensure safety, to ensure efficacy of these products at various doses and formulations, given what we heard today. Thank you very much.
Dr. Brian Lee.
Brian Lee. So I voted no. I think that the public deserves to have access to drugs that have shown efficacy and that have been shown to be safe. And I think that the data demonstrated today has not met that bar.
Dr. Janet Lee.
Janet Lee, I voted no.
So to summarize, the majority of the votes were yes, based on a lack of evidence. And those that voted no cited two issues, one, access, and two, availability. Sorry, majority. The majority was no. Correct. Okay, next we'll proceed with question 3C. Slide 5, please. Question 3C. Section 503A, bulk drug substances list, Thymosin alpha-1 related bulk drug substances. FDA is proposing that Thymosin 1 alpha acetate not be included on the 503A bulks list. The question today is, should Thymosin alpha-1 acetate be placed on the list? If voting yes, you're recommending FDA should place Thymosin alpha acetate on the list. If voting no, you're recommending that FDA should not place Thymosin alpha-1 acetate on the 503A bulks list. If the substance is not on the list when the final rule is promulgated, compounders may not use the drug for compounding under Section 503A unless it becomes a subject of an applicable USP or National Formulary monograph or component of an FDA-approved drug. Are there any issues or questions from the panel about the wording of the voting question? If there are no further questions or comments concerning the wording of the question, we will now begin the voting process. Please press the button on your microphone that corresponds to your vote. Reminder, if you're unsure of your vote or wish to change your vote, please press the corresponding button again before the vote is closed.
Takia Stevenson, DFO. For the record, there are 4 yeses, 17 noes, and 0 abstentions. Thank you.
Now that the vote is complete, we'll go around the table and have everyone who voted state their name, vote, and if you want to, the reason why you voted as you did into the record.
Todd Durham, I voted no.
Alan Veda, I voted no.
Robin Bogner, I voted no.
Brian Serumaga, I voted no.
Elizabeth Rebello, I voted no.
Kathleen Guerra, I voted yes.
Linda McElhaney, I voted yes because I think there's some rare indications where patients need access to this drug.
Tim Fensky, I voted yes.
Alice Powell, I voted no.
Mark Ganny, I voted no.
Mark Sasha, I voted no.
Cecilia Monge, I voted no.
George Cyber, I voted no.
Nina Clark, I voted no.
Mark Chris, I voted no.
And online, Dr. Galore?
Thank you, Padma Galore. I voted no, and I would like to take a minute to just go over the decision. As many have stated, it was not an easy decision because access is something we would all like to ensure patients here in the United States have to drugs and compounds that could help them, especially with rare diseases. I want to thank the presenters who advocated strongly for this and brought data. However, the data was not adequate to ensure the safety and efficacy of this drug when compared to the FDA presentation. That said, we do applaud those efforts. And the argument that that we should be manufacturing this here to provide access to our patients is valid, but on the flip side, that is exactly what consumers here, and I think that's what Dr. Gupta was alluding to, consumers in the United States consume products that are available here, assume that they are safe. And while the compounders, I'm sure, are making every effort on their part to do so, as the FDA pointed out, this is really a bulk drug substance sourcing. And since most of the sourcing is not in the United States, that is concerning. So for those reasons, I voted no. And I also wanted to take this opportunity to thank Dr. Rubello for stepping in for me at the last minute. And so thank you very much.
Thank you. Dr. Corbett?
Hi, I'm Amanda Corbett. I voted yes for similar reasons as I'd mentioned before. Thank you.
Dr. Gans?
Dr. Haley Gans, I voted no again for many of the similar reasons. In addition, because putting it on bulk doesn't in any way help with decisions on how to use it. So we need all of that to be part of an application to the FDA.
Dr. Gupta?
Thank you, Dr. Gupta. I voted no for the same reasons already stated.
Dr. Brian Lee?
Brian Lee, I voted no.
Dr. Janet Lee?
Janet Lee, I voted no for the same reasons eloquently stated by others.
Great, thank you. And I just want to take the time to thank Dr. Takia Stevenson, who's gone above and beyond in making this meeting happen today. I want to thank all of you for being present, for the presenters, both on the FDA side and on both sides. And I just want to wish you safe travels. And before we adjourn, are there any last comments from the FDA?
This is Gail Bormel. I wanted to personally thank everybody for their for coming today and for their expertise and taking the time out to participate in this very important advisory committee meeting.
Well, with that, we will now adjourn the meeting. Thank you.